injectable. And this goes hand in hand with an increase in the pH of the tumors. So tumors with a complete knockout of lactate, they have a higher pH, which is associated with this transport by lactate transporters. And then we injected LDHA-B double knockout cells in mice. And Tobias Bob in Mainz, they performed a kind of PET scan of those tumors. You can see that the wild type tumors accumulate glucose very well. And there was a much lower accumulation of lactate in tumors without LDHA-B. And when those tumors then were exposed to checkpoint therapy, the tumors with low lactate levels, they responded very well. Only one did not respond. Accumulation of lactate compared to wild tumors without LDHA was very limited. And when those tumors, there seems to be really an interplay between lactate and the response to immunotherapy. But why is that the case? So why is tumor glucose metabolism important for patient prognosis? So let me show you some pictures of patients, biopsies. In this case, it was breast carcinoma. So we analyzed tumors, the glucose transporter expression, GLUT1, and in consecutive slides or biopsies, sections, we analyzed CD3, so T-cell infiltration. And you can see here that in the tumor site where we find a high glucose transporter expression, so high glycolysis is going on, there are almost no T cells present. So it seems that T cells do not like high glycolytic activity of tumors. So tumors, so-called Warburg tumors, seems to be cold tumors. This means there are almost no T cells around and also no NK cells around. But still, why is this the case? So we found out that lactic acid has a strong negative influence on immune cells, not only on T cells, CD4, CD8 T cells, but also on NK cells. And there's also a negative impact on antigen-presenting cells, dendritic cells. But surprisingly, Tregs, so regulatory T cells, which are more immunosuppressive, or macrophages, seem to have no problems with lactic acid. They can stand it and they even like it. So I will give you some examples. So we cultured human T cells, activated them, and added lactic acid or lactate, so without acidification. And then we analyzed proliferation of the lymphocytes, IL-2 expression and interferon gamma expression, and also perforin. Perforin is important for the kill of tumors by T cells. So here on the left-hand side, you can see proliferation of T cells exposed to lactic acid, the black bars. And you can see dose-dependent effect. So T cells stop to proliferate in the presence of lactic acid. This is not the case if you add lactate. So the effect is somehow acid-related. And you can also see here that perforin, this important effector molecule of T cells, is suppressed by lactic acid. And the same holds true for IL-2 and interferon gamma, important cytokines for the defense against tumors. And this is a complete, more or less, shutdown if you apply 20 millimolar lactic acid. If you can recall, in melanoma metastasis, we found levels up to 40 millimolar. So this is extremely physiological concentration. And what is also what does also happen is that lactic acid in a dose-dependent fashion leads to the apoptosis, so the kill of T cells. So the green cells are dead cells. With increasing concentrations up to 20 millimolar, almost no T cell is present. To prove the effect of lactic acid and or LDH on immune cells, we used a different B16 melanoma model where we decreased, not completely knocked out, LDHA to limit the lactate production in these melanoma cells. You can see here on the right side that LDHA in B16 cells with small hairpin LDHA, they show a decreased protein level of LDH, and the lactate level in those cells in vitro is decreased about 40%. We then injected cells with low LDHA and control cells subcutaneously in mice, and Wolfgang Müller-Klieser analyzed by a special bioluminescence technique. This was before mass spec imaging. He detected lactate in those sections, and you can see the control cells have approximately 15 to 18 millimolar lactate, and the LDH low cells about 7-8 millimolar. So what was the consequence of that? The tumor growth was clearly different. So tumor growth of lactate low cells was lower compared to the wild type, but this was not the case in vitro. So in vitro, the cells were able to proliferate in a similar way. And also in mice without immune cells, the proliferation was completely comparable. So this effect, this difference, seems to be related to the activity of immune cells. So we analyzed immune cells in the tumors with low and high lactate expression. And you can see here that the NK cells, there were higher levels of NK cells, there were higher levels of CD3, CD8 T cells, and it was not only that the cell number was increased, there was also an increase in the NK cell activity, so there was more interferon, and there was more interferon also in the CD8 T cells that we analyzed. So what I showed you here is that it seems that lactic acid really has a strong impact, especially on cells like T cells and NK cells related to the tumor defense. In patients, it's hard to do a genetic deletion of LDHA, B, or whatever. So you have to find pharmacological ways to target glycolysis. This is not so easy. There's a nice review of Vinit Kumar discussing the possible targeting of monocarboxylate transporters. And that's what we did. And we did it with diclofenac or Voltaren, which is an old drug that is in the clinic for the inhibition of COX, limitation of COX activity. And what we found is that diclofenac really targets monocarboxylate transporters. It targets MCT1 with a lower Ki than MCT4, but in the end, it blocks lactate production, which is shown here. It blocks, this is a melanoma cell line, a human melanoma cell line, MEL-IM, and diclofenac dose-dependently after 24 or 48 hours can reduce lactate levels of those cells. So we asked, so would it be possible if we now target glucose metabolism with diclofenac, could that improve checkpoint therapy in a murine model? So in this case, we used 41 tumor cells. This is a breast carcinoma cell line. And it was, we used a vehicle control, diclofenac checkpoint therapy with anti-PD-1, CTLR4, and a combination. And you can see here the tumor grows, the tumor volume of tumors treated without anything. This is the vehicle control. And diclofenac, then it delayed the tumor growth, but tumors still were growing very well. Checkpoint therapy alone delayed the tumor growth, but elimination of tumors really perfectly happened with diclofenac. And this was not a anti-COX effect. This is what people normally ask. So is this effect of diclofenac based on its anti-cyclooxygenase inhibiting? It's not, because we used as a control aspirin on the right-hand side. And if you add aspirin instead of diclofenac, you can clearly see the difference. There is no positive effect of aspirin on tumor growth. So, but still, diclofenac is not a specific drug for MCTs. It inhibits cyclooxygenase 1 and 2. So we cannot be sure that this is really the only effect or the only important effect. Therefore, we asked whether a specific inhibition with MCT1, 2, Fatima already showed you some results on that, could improve checkpoint therapy or MCT4 inhibition. And this is a collaboration with the Merck company. Here we used MC38 tumor, and we combined the Merck inhibitor, the MCT4 inhibitor, together with checkpoint therapy. These data were mainly generated by my PhD student, Nathalie Babel, with the help of Professor Renner Sattler. And you can see here the tumor grows in those mice. Compared to the vehicle control, the black line, MCT4 inhibition alone had no effect at all. It was perhaps a slight delay in the beginning. Checkpoint therapy, anti-PD-Link1, delayed tumor growth, the blue line, but only the combination of MCT4 and checkpoint therapy, here the green, has a clear effect. And adding MCT1 either alone or in combination with anti-PD-Link1 had almost no effect. So it did not improve the effect of MCT4 in this model. And the same here for survival of the mice. So the best effect is seen in the combination of MCT4 and checkpoint therapy, again, the green line. And if you add not only MCT4 inhibitor, but MCT1 and MCT4 inhibitor, the red line, this does not improve tumor control. So what I've been showing to you up to now is that tumors produce lactic acid, and lactate and protons are accumulated in the tumor site, and it seems to be important to limit this secretion. But there's not only lactate production by tumor cells. Also activated immune cells, for example, macrophages, and also sometimes fibroblasts can secrete lactate and lead to the accumulation of lactate and protons in the tumor environment. So, and this is work of Peter Ziska, a physician in our institute. So Peter asked the question whether T cells or macrophages are prognostic in clear cell renal cell carcinoma. And what he found is surprising, because he found that CD8 T cells are not prognostic for clear cell carcinoma. This is a TCGA, it's just gene analysis. But the the number or the expression of CD14, this is a macrophage marker gene, was associated with survival of patients. So if you have lower numbers of macrophages, it seems to improve survival. So macrophages, tumor-associated macrophages, seem to be not good for patient prognosis. And what we also found in this tumor is that MCT4 expression is important, and IL-6 expression. So IL-6 is a cytokine. This is mainly or often secreted by macrophages. So we wondered whether MCT4 might be also expressed in macrophages. And Malte Simon from the Deutsche Krebsforschungszentrum, he took some data, single-cell RNA-seq data from clear cell renal cell carcinoma patients, and he analyzed the MCT4 expression in different cell types. And what you can see here is that MCT4 is clearly and strongly expressed in myeloid cells in this type of tumor. It's also expressed in tumor cells, but there's a strong expression in macrophages. Based on these data, we wondered whether treating macrophages or tumor biopsies with diclofenac would reduce lactate and in turn also IL-6, which seemed, and this is what Natalie did. She used biopsies from renal cell carcinoma patients. She made a single cell suspension and then treated the cells either with diclofenac again and with aspirin. And what you can see here is that after 24 hours overnight culture, lactate levels are clearly decreased with diclofenac, but not with aspirin. And this in turn also leads to a reduced IL-6 production in those macrophages. We also did these experiments with MCT4 and MCT1 inhibition. So we see similar results. MCT4 and MCT1 is needed in this case to reduce lactate levels and IL-6. So to sum up these results, it seems that not only tumor cells, but also tumor-associated macrophages express high levels of MCTs, and inhibition of glycolysis reduces lactate levels in both types of cells. And in macrophages, this is associated also with a strong decrease in IL-6. So the conclusion that we draw from this is that lactic acid accumulation limits T cell infiltration and especially T cell function in murine and in human tumors. So it seems a good idea to target glycolytic activity, but not only in tumor cells, but also immune cells. And this may then in turn support immunotherapy. So this is the end of my talk, and I would like to thank my co-workers, especially Katrin Renner-Sattler and Natalie, who did most of this work. And I thank you for your attention. Thank you.
Thank you very much, Marina, for this complete demonstration of the way and the lactic acid. And now the question for all of us, of course, will be how we can exploit all this knowledge to treat related tumor. So this is open for any question now from the...
Thank you very much. I was wondering, does lactic acid modulate PD-1 expression on immune cells?
Yeah, this is a very interesting question. We analyzed that, but surprisingly, there was not a clear regulation on neither on T cells, tumor cells, or macrophages. Yeah.
Nor CTLA4 or any MHC expression?
MHC, you are right. There was an impact on MHC, decrease in MHC1 expression, but not on the checkpoints. Yeah.
Thank you.
Thank you.
Yeah, thank you for that. A question about the MCT1 expression, which I understand goes up in CD8 T cells on activation. Yeah, I just like your thoughts on how that would play out in the in the patient's environment when the tumor is developing.
Yeah.
And the second connected question is that do you think that now that CAR T cell and NK cell therapy is slowly developing, does it make sense to modify the metabolism of these cells as well when these in vitro manipulations are being performed?
Yeah, very good question. Yeah, so we analyzed whether MCT1 and or 4 inhibition limits T cell proliferation activation. Actually, MCT1 was developed as a T cell suppressive agent. And this is really true. So proliferation is decreased by MCT1, not so much by MCT4 because it's not so strong expressed. But surprisingly, if you look at effector molecules such as interferon, there's not much change. So it seems mainly the proliferation is changed. So there is a therapeutic window probably, especially also if you think about CAR T cells also. But second part of your question, yes, I think there is a need to harness T cells against this environment. And this is what we are currently doing. We tried that with LDHB overexpression so that the lactate that comes in is metabolized. So this shows some response, but it's not so perfect. Yeah, we are working.
Hi, thank you. Excellent talk. I wanted to know regarding this is it limited to melanoma or other cancers also you have explored and you see the similar kind of, is it like cancer dependent or is it like, a lot of cold tumors, let's say other cold tumors also have the similar kind of impact? Thank you.
So what you are asking is why the effect is cancer dependent?
Is it cancer dependent? Like,
So, it is the case that not each and every cancer is highly glycolytic. Most of them are, especially metastases are. And it's true that not each and every cancer shows a high expression of MCT4. So the cell line that I showed you in the end with this MCT4 inhibitor, the colon carcinoma is, for example, expresses higher levels of MCT4 in patients and also in this cell line. But this is not true for all tumor entities.
We have more questions? If not, yes, over there. Okay.
Hello, ma'am. Excellent talk. Ma'am, I had a question. You have shown the negative effect of lactate on NK cells and T cells. I had a doubt whether any co-culture experiments were done like T cells and NK cells like to replicate a more complex tumor microenvironment. And the effect was seen in the co-culture experiment, whether one of the immune cells like T cells is preferentially downregulated or not in terms of lactate.
So you are asking whether immune cells are more susceptible than other cells?
A particular immune cell is more susceptible or not. Like in a tumor, there is a collection of immune cells, right? Not a single immune cell.
So if I understood you right, so not all immune cells in the tumor react in a similar way. Yeah. So T cells and especially NK cells are very susceptible, but myeloid cells can stand it very good. So they do not care, do not mind about it.
Okay, thank you.
So is there any online participant questions? Apparently not. If not, we would like to thank you very much. Thank you, Marina.
Thank you, ma'am, for your enlightening us with your talk. I now request the session chairs Dr. Jack and Dr. Sushil Kumar, along with Dr. Prakash Baligar, to felicitate the speaker.
Very good. So, yeah, okay. So it's a pleasure to have the, as an associate professor, Chandra Pascal. Okay, no?
So let's.
You went for the presentation. Okay, okay. So, okay. Yeah, yeah, it's you. No, the speaker is. We have the next speaker now. Next speaker. Yeah. Oh, I see. Anna Preto now. Oh, I see. Where is she? Yeah. The next speaker here on this, Anna Preto. Is that you sure? I don't know what is the. These are not the problem. No? Yes, Shamra, Dr. Shamra. So you have to put on that. Yeah, I am Dr. Shamra. So the next presentation will be by Dr. Chandra Prasad. Okay. I hope it's here. Okay. So Dr. Chandra is Associate Professor in the Department of Medical Oncology, All India Institute of Medical Science, New Delhi. And his area of research, of course, is cancer cell biology, Wnt signaling, cancer metabolism, and therapeutics. Okay, so a pleasure to listen to you, Dr.
Thank you. Thank you, Josh. So good morning, everyone. First of all, I want to thank the organizers, EMBO and MIT University, for organizing this wonderful conference. And we are really happy to be here and presenting our work. So today I'll be speaking about lactate metabolism in triple negative breast cancer. So coming to this disease first, like worldwide, in India also, breast cancer is the most common cancer. So in 2022, if you look at the new registration of breast cancer cases, it is around 105 per 100,000. So it is really high. Now compared to West, in India, ER negativity is really high. And if you look at the prevalence of this disease, it's around 25 to 35%. In the West, it is around 15%. So yes, TNBC is an India-centric cancer. Now, I work in medical oncology department where we routinely treat patients, and we can find these TNBCs are more aggressive than other breast cancer subtypes. And the therapy for TNBCs are limited because, as it says, triple negative breast cancer. These cancers are negative for estrogen, progesterone receptor, and HER2/neu. So you can't have hormone therapy or HER2/neu targeted therapy like Herceptin for these cancers. So patients are mainly treated with the classical chemotherapy. And if you look at the data, it is very dismal because 50% of, it's around 50 to 70% of the patients with the early stage TNBC actually recur. The disease recurrence is there. And 37% of the patient experience 5-year mortality rate after surgery. So yes, treatment is required for this. So coming to this introductory slide on growth factors, we all know that growth factor is one of the hallmarks of cancers because many of the growth factors are overexpressed in cancers, which actually drive cancer development and progression, like EGF. EGF binds to its receptor EGFR, and you can see in many cancers, EGF as well as EGFR overexpression is there, which actually initiate or triggers the cascade, which are involved in the modulation of cancer cells, like it provide resistance to apoptosis, increased migration, increased cancer growth. So for the past few years, we are working on a growth factors known as WNTs. So these are cysteine-rich glycoproteins, and they have been shown to be deregulated in various cancer. And these WNTs basically regulate the critical characteristics of cancer cells, like proliferation, migration, and invasion. So it's a pictorial diagram where it shows that a WNT-producing cell is there, which is secreting WNT into the tumor microenvironment. And the WNT-responding cell should have a receptor for WNTs known as frizzles with a co-receptor LRP so that they can initiate the intracellular signaling. So WNT can work autocrine or in a paracrine mode. Now, if you look at the literature, there are 19 WNTs. And these WNTs are beautifully characterized that what signaling they can actually induce. So if you look at WNT pathway, you have like 3 pathways, canonical and non-canonical. In canonical pathway, which is known as or beta-catenin dependent pathway. And these non-canonical pathway can be calcium-induced pathway and planar cell polarity pathway, which we previously know that it doesn't associate beta-catenin or it is beta-catenin independent pathways. But now the things are changing. So I will talk about WNT5A signaling, which is actually a non-canonical pathway. And it induces G protein signaling and calcium signaling, basically involved in cell adhesion and migration. So the beautiful thing about this WNT5A is that WNT5A has opposing roles. In some cancer, it's a tumor suppressor, and some cancer, it's a tumor promoter. So when I was doing my postdoc in Sweden, they were working on WNT5A, and they have like 2 groups, one working on melanoma, where it was a tumor promoter, and one on breast cancer, which is a tumor suppressor, to understand better how this WNT5A actually is functioning in these cancers. Now, when WNT5A cells, basically they have very high, melanoma cells have very high WNT5A levels. These are A2058 cells, which have moderate level of WNT5A, so that can be induced with recombinant WNT5A. So when the recombinant WNT5A were given to these cells, the migration of these cells increased. When we profiled these cells with proteomics, we found that glycolysis is the one of the pathway which is upregulated when the WNT5A is given. And one of the genes that we validated was lactate dehydrogenase. And we found that in WNT5A treated cells, LDH levels are really high and activity is also high. We then look into lactate, and we also found that WNT5A actually induced lactate production in A2058 cells, and it was very well neutralized when the antagonist of WNT5A, which is Box5, was given. We further try to inhibit the lactate production in the endogenous cells A2058 and HTB63 cells, and we find that if we treat with WNT5A siRNA, the lactate production decreases. Through seahorse, we showed that WNT5A actually promotes extracellular acidification rate and the glucose consumption in melanomas. Now, it was very well defined what lactate can do. It can actually modify tumor microenvironment, mainly the immune cells, and as well as it has been shown that lactate can induce migration of cancer cells. So I'll not go into this Warburg effect because now we have discussed time to time. But now coming to breast cancer again, where Wnt5a is a tumor suppressor. In a highly invasive breast cancer cells, like triple negative breast cancer cells, they display glycolytic phenotype. So they produce too much lactate. So just give you a background about Wnt5a in breast cancer. It's a tumor suppressor gene. So it's just opposite to melanomas. And there is a loss of Wnt5a in invasive rectal carcinomas, like 40 to 50%. And in TNBCs, this loss is around 75%. What we have shown previously that if you reinstall Wnt5a signaling in these cancers like breast or colon, you actually inhibit the migration and invasion of the cells. So the question was, if Wnt5a in melanoma can actually provide a glycolytic phenotype and thereby making them more migratory, can Wnt5a reinstallation in breast cancer cells inhibit the lactate production? Indeed, it is. So what we did, because we used two cell lines, MDMB468 and MDMB231. So both the cell lines are negative for Wnt5a because Wnt5a is a tumor suppressor in breast cancer. So we transfected these cells with Wnt5a plasmid. As you can see here, that Wnt5a transfected cells have less lactate production as well as less migration. However, we found no effect on the tumor proliferation. To decipher more what is happening in these cells, we look at the glycolytic markers. And we found a protein. It's an isoform of PFK1. We call it phosphofructokinase P. It's a platelet isoform. It was downregulated in Wnt5a overexpressing cells. And in both the cell lines, MDMB468 and 231, where we overexpress Wnt5a, the PFKP expression was down. So we checked PFKP isoforms in breast cancer using online meta-analysis tool. And we found that it is only PFKP, not the liver isoform or the metabolism muscle isoform has been involved. It's only PFKP that is associated with the reduced breast cancer patient survival. So it is an important protein. So what we did next, we knocked out PFKP in both the cells, parental cells of TNBCs, and we found that there is a decrease in lactate production, as well as the migration of the cells. We also shown the reduction in the invasion also. So PFKP is an important modulator of lactate production in these cells. But how this PFKP is regulated? And at that time, a study came in colon carcinoma where they showed that PFKP is beautifully placed below beta-catenin. So we looked at beta-catenin in Wnt5a transfected cells, and we found that there is a reduction in active beta-catenin level, which is non-phosphorylated beta-catenin, in Wnt5a expressing cells. And we also showed that in Wnt5a expressing cells, there is a transcriptional repression of beta-catenin. Furthermore, we also showed that what Wnt5a can do, the inhibitor of beta-catenin also can do. That is XAV939. So both can actually inhibit beta-catenin as well as PFKP in the TNBC breast cancer cells. Furthermore, we showed that if you just treat beta-catenin inhibitor alone, there is a reduction in the lactate levels. To us, there was another question. If the lactate is outside in the tumor microenvironment, how it is affecting the cancer cells? So here we show that if parental cells of 468 cells, it's shown here that when they are given recombinant Wnt5a, migration goes down. But when these parental cells were given lactate, the migration goes up, but it was neutralized when recombinant Wnt5a was present. And this effect was more predominant in the overexpressing cells, where you can see that the lactate exposure actually increased the migration of the cell, but Wnt5a expressing cells somehow inhibited this increase. And we found that Wnt5a actually inhibits MCT1 in these cells. So if you compare the second and the fourth here, maybe the bar, So when you treat MDA468, empty vector transfected cells with lactate, the migration is high, the MCT1 levels are high, but in presence of Wnt5a, even if you give lactate, almost there is no change. So Wnt5a, what is doing? So Wnt5a, we postulated that it is inhibiting beta-catenin, which in turn inhibit PFKP, thereby reducing the lactate. So lactate production less, and Wnt5a is also inhibiting the influx of lactate by inhibiting the MCT1, thereby impeding the migration of triple-negative breast cancer cells. 2 years back, we also found that LDHA is actually a downstream to PFKP in triple-negative breast cancer cells that we published. So I'm not discussing that paper here. But this is all cell lines. And the question is it really happening in patients? This beta-catenin, PFKP, LDHA, will it make sense in patients? So what we did, we carried out immunohistochemistry. So this is all unpublished data, which we just recently have. So as you can appreciate, kidney are the control cells. The beta-catenin is actually cytoplasmic localized, as is in some cases, it is in the nuclei of triple-negative breast cancer cases. And if you look at the positivity of beta-catenin, it was around 64%. PFKP expression was predominantly localized in the cytoplasm, as we have postulated, in TNBC patients, and the positivity rate was around 54%. LDHA, yes, it is a cytoplasmic protein, but we found some of the protein in the nuclei also. I don't know its relevance, but we are working on that. And the LDHA positivity in triple-negative breast cancers are around 58%. And the MCTs, of course, it's a membrane channel, we all know. And we found it to be like 50% of the TNBC upregulated patients show the upregulation of MCT1. So this is just a statistical association between glycolytic markers. Unfortunately, we didn't find anything with either age, tumor grade, lymphatic involvement, or the tumor stage. But when we run the protein-protein association, we did find that beta-catenin is positively associated with PFKP as well as MCT expression in triple-negative breast cancer cells, although we are not able to find any correlation with LDHA. So we are still waiting for the prognosis data that we will compile in May, as we're going to censor the patients in April. Okay, so if there is access where beta-catenin is regulating PFKP, LDHA, MCT1, or else, can we inhibit it, and thereby inhibiting the lactate production, which is a readout of aerobic glycolysis or glycolytic phenotype? So first, what we did, we looked into like online tools and looked into what happens to these all 4 genes in ER-negative cells compared to ER-positive luminal cells. So we used a Xena functional genomic explorer, and we actually categorized 18 cell lines in luminal phenotype and 18 cell lines in basal A and B phenotype, which is actually which also comprise of TNBC cells. And we found that in estrogen negative cell lines, with basal A, B type, all the gene transcripts were upregulated. So now, if beta-catenin is regulating these markers, can we inhibit beta-catenin and thereby inhibiting the lactate production in these cells and thereby migration of these cells? So we did MTD assay, and with the optimal dose of 20 micromolar, we treated both 468 and 231 cells with XAV939, which is a time kinase inhibitor. So it basically modulates the beta-catenin destruction complex, thereby inhibiting beta-catenin. And yes, both the cell lines responded in a beautiful fashion. Although just to where we saw the decrease in beta-catenin, we saw the decrease in PFKP, LDHA, and MCT1. Though I have to tell you that in 231 cells, we don't find the expression of MCT1, although we are not the first one. There are earlier report also. When we look at the migration, yes, the migration was affected. even the lactate production was less, but it was not that much. So the question was, should we increase the dose? And then we were discussing, and then we looked at the literature that shows that tank arrays inhibitors has a lot of drug toxicity. Basically, it actually is involved into intestinal toxicity because beta-catenin has a role in the normal physiology of the intestine. So now the question was whether to go along with the drug XAV939 or we should look for other compounds also which offer less toxicity. We found one, although most of the beta-catenin inhibitors are in clinical trial and you can't get hold of that. And still, most of the clinical trial being stopped because of the toxicity. So we found a drug known as exitinib. We all know exitinib is a tyrosine kinase inhibitor, and it is FDA approved. It is, and our colleagues in the clinical side shows that it has a manageable toxicity. But in 2016 paper in PNAS, authors showed that exitinib blocks specifically Wnt beta-catenin signaling. And how it does that, exitinib actually stabilizes the E3 ligase known as SHPRH, thereby ubiquitinating beta-catenin, thereby putting it into the degradation. So we thought, let's check this compound, because if it performs in the same way as beta-catenin inhibitors, there might be a chance that we can take it to clinic. So we did MTT assay for the dose, and we first looked at the non-phosphobeta-catenin. Yes, exitinib, just like XAV, inhibited the active beta-catenin in both T and BC cell lines. Furthermore, it shows the same pattern, although blots are not good, but yes, it shows the same pattern. But what was the best thing which I liked about exitinib, the migration and lactate data was very clear. It was not like XAV939. And we looked also into the invasion of these cell lines, and the exitinib performed better. So where we are using dose of 20 micromolar of XAV in triple negative breast cancer cells, we are now using 100 to 150 nanomolar dose of exitinib. So we wanted to look into in vivo. So we took this 41 cells, and then we first profile that 41 cells also, when you treat with exitinib, actually shows reduction beta-catenin and all the downstream targets. And further, there is a relative impairment of lactate in 41 treated exitinib. And this is the initial data that has come up, that where we treated BALB/C mice, which it's orthotopic 41 model. And we can show that as compared to control, when we treated with 30 milligram per kg of exitinib, we found there's a reduction in tumor volume as well as weight. And at the same magnification, you can see the control cells and exitinib treated cells. The exitinib cells are totally regressed. And most of the cells, actually, in most of the mice, we couldn't find actually the tumors. So with this, I want to conclude. So till now, what we can see that there is significant upregulation of beta-catenin in T and BC patients in cell lines. We also found elevated level of glycolytic markers, PFKP, LHA, MCT1 in T and BC patients in cell line. Beta-catenin can positively regulate these glycolytic markers, PFKP, LHA, and MCT1, suggesting that its role, beta-catenin's role as a regulator of aerobic glycolysis in T and BC. Like XAV939, exitinib also inhibited the beta-catenin-induced glycolytic axis, PFKP, LHA, MCT1, thereby impairing the T and BC migration invasion, as shown by the in vivo data also. So when we showed this data to our clinical colleague, they are really interested to run a phase 2 clinical trial, although we need some more experiments to do whether exitinib is actually generating the specific effects on beta-catenin through SHPRH, but there is a limitation of this antibody because we are not finding it very specific. So we're looking for it. So I want to acknowledge my PhD student, Mr. Umar Sheikh, who is sitting here. Basically, he carried out all the work. And my collaborators from the Department of Surgical Oncology, Pathology, and Medical Oncology, and of course, the funders. Thank you.
Thank you very much for your presentation. Now it's open for discussion. Okay. Any question for
Yeah,
Dr.
thank you.
Prasad? Yes. Okay, a microphone. You can come here.
Very nice talk. I have just a question related to the primary cilium. The Wnt pathway is mainly regulated by the primary cilium. Did you get a chance to look at in your different cell line when you modulate the expression of Wnt5a, the expression or the absence of the primary cilium? Do you get? Yeah. The Wnt pathway is mainly regulated by the primary cilium. So I was just wondering if you get a chance to look at the expression of the primary cilium when you modulate the Wnt5a expression.
No, we didn't look that.
Okay, thank you.
I just had a quick question. I was just wondering, I'm not in the specific field, but how exactly lactate imbalance or MCT inhibition regulates migration pathway? Like what exact pathway does it affect?
Okay. We are just compiling a manuscript, okay? Where we are actually treating the cells with lactate and looking at the migration. So I'll not tell you the whole story, but there is a protein known as MPC1, mitochondrial pyruvate channel. We showed that its involvement in there, and its inhibitor 7-AAC2 actually inhibits the lactate-induced migration of the cells.
Dr. Chandra, I have a question. About 20 years ago, we analyzed 800 patients from the Cancer Center by immunochemistry using a beautiful anti-MCT1 and anti-MCT4. And we get out a very strong conclusion on all these 800 patients that is a very bad prognostic with the expression of MCT4, but not MCT1. Okay. And we had an interpretation on this, is that because that was also in conflict with another publication. Our interpretation on this, we went to see again the tissue from the patient that expressed the MCT1, and we find very strong immune infiltration. So here, our conclusion was really the MCT1 rare positive case of this patient were due to immune infiltration.
Thank you. Just to tell you that this immunohistochemistry is limited to not one PhD student. There are 3 PhD students they are working on the same cohort. So basically this 99 patients which I've shown you, basically we are doing 18 metabolic markers on these patients. So when the data will mature, of course, we're going to share the data with you. Yes, regarding MCT1, we will see how it fits with prognosis because we have not analyzed yet, but we will do.
Okay.
But thanks.
Thank you. Any other question for, yes? No, and what about online people for any questions? No? Okay, so if there is no question, thank you very much, Dr. Prasad.
Thank you, Dr. Prasad, for this wonderful talk. I now request the session chairs and Professor Vishwas to felicitate the speaker.
So now it's a present here. We are very happy to have the presentation online of Dr. Alessandro Carrer. Okay, it's Dr. Carrer.
Yes, I'm here.
Yes, you're there. So let me see if I have some information about you. Yes, Dr. Alessandro Carrer is assistant professor at the Veneto Institute of Molecular Medicine in Padova, Italy. His area of research is cancer metabolism and metabolic reprogramming in cell-fate decision. And he has several awards, the Marie Curie MCTO Fellowship in 2020 and the AICR Cancer Research in 2019. And also is the board of the director of the International Society for Cancer Metabolism that we know very well, ISCAM in Europe. And for this, it will be nice to start and to listen to your presentation. And the title of his presentation and the talk here is the reprogramming of acetyl-CoA metabolism supports pancreatic carcinogenesis.
Okay. Thank you very much for your kind introduction and good morning, everybody. Really sorry I cannot be there in person with you. This was a terrific meeting, but I really couldn't squeeze a long trip at this time. But in any case, I'm sure that after the talks yesterday and earlier today, you have really sense now that metabolites play multiple functions in during cell growth, and they certainly contribute to augment the mass of the cell by generating structural components. But more recent data indicated that metabolites also contribute to signaling events, largely because they serve as substrates or cofactors for protein post-translational modifications. And more on that, even complex products of metabolism, like lipid, for example, can also impact the signaling event. So to make a long story short, you can realize that this is obviously a strong intertwine between biomass generation and signal transduction. And this is important for the cells because it helps to coordinate nutritional inputs with cell proliferation. And this obviously, it's no surprise that this is hijacked in cancer. And talking about integration of different stimuli, so you can appreciate that acetyl-CoA is really uniquely positioned at the crossroad of multiple anabolic and pathologic pathways. So a quick look at what acetyl-CoA is and does. Acetyl-CoA is obviously present in the mitochondria as part of the TCA cycle, but it's also present in the nucleocytoplasmic compartment, where it's a building block for the synthesis of fatty acids, cholesterol, among other things, and is also served as the unique donor for every single acetylation reaction in the cell, including histoacetylation. That was the point of this slide. In any case, the nucleocytoplasmic acetyl-CoA is produced by different enzymes, but really the predominant role is played by this guy right here, ATP citrate lyase, or ACLY. And indeed, in genetic ablation or even pharmaceutical inhibition of ACLY is able to reduce both histone acetylation, as you can see in this plot, if you compare the first two lines or these two lines here, and he also reduces lipid synthesis, as you can appreciate in this staining. But and this correlates with the reduction in acetyl-CoA abundance within the cell. And while this is obvious in mouse adipocytes that synthesize lipids all the time. It has really been extensively demonstrated in different cells in different organisms by us as well as many other groups. But the genetic ablation of ACLY also decreases cholesterol level, and this was obtained in pancreatic. But in any case, this is probably less appreciated, but still bears a significant, remarkable clinical implications as ACLY inhibition, liver-specific ACLY inhibition are in the clinic for statin-resistant cases of hypercholesterolemia. So I think I made it clear that acetyl-CoA levels are able to impact both the production of structural components and signaling events. Over the years, we found that acetyl-CoA levels really fluctuate in response to a number of cell extrinsic stimuli. The question then was, this is important for tumor development and progression. So we decided to tackle this question in pancreatic ductal adenocarcinoma, which is a really deadly disease with an abysmal prognosis. Is projected to become the second leading cause of cancer-related death by 2030, and probably much earlier than that. And well, this is many reasons, of course, but ultimately it is because of the mortality rate, the life expectancy for this disease is really, really abysmal. It's basically a death sentence. So pancreatic ductal adenocarcinoma is a KRAS-driven cancer. As KRAS is mutated in more than 95% of the patients, and was according to a classical multi-step model where epithelial cells undergo a latent phase before transformation, okay, that you can see here, and then they develop preneoplastic lesions that are progressively more dysplastic until they manifest into the frank carcinoma. And pay attention that this model offers room for different therapeutic interventions at different stages. And while metabolic reprogramming, while metabolism is extensively reprogrammed throughout the whole process, metabolic aberrations are usually stage specific. And I will make this point clearer later in my presentation. So what happens to acetyl-CoA metabolism? Acetyl-CoA is reprogrammed early on, and in pancreatic epithelial cells before you can appreciate here, before the onset of any dysplastic lesion, and you can see that acinar cells in this case expressing oncogenic KRAS here in violet have augmented levels of acetyl-CoA compared to acinar cells expressing wild type KRAS. So we ask whether that could represent a targetable vulnerability, and we decided to create conditional knockout mice for ACLY, lacking these enzymes specifically in the pancreas. These mice are viable and do not exhibit any major abnormality. They are just fine as long as we know. So we crossed these mice with mice expressing oncogenic KRAS in the pancreas. These mice spontaneously develop lesions in the pancreas, as you can appreciate in this panel on the left, probably, and the blown out. And you can probably appreciate that the absence of ACLY also determines a reduction in tumor burden. And these mice lacking ACLY in the pancreas also live a little longer, but they do eventually get tumors eventually. So we decided to dig a little bit more into the mechanism to understand how how ACLY ablation leads to a reduction in tumor burden, and the mechanism by which tumors eventually find the way around it. And here, really, sorry to cut a very long story short, but in any case, we found that two acetyl-CoA-dependent processes play a critical role in different stages of the disease. I won't have time to talk about both, but I will focus my presentation on sterol synthesis. Some of the data are published, so you can check my publication if you're interested. So a couple of words on sterol synthesis is so the path that leads to the synthesis of sterol here in violet and isoprenoids here in orange is quite long and energy consuming. This is a very simplified slide diagram. The pathway is actually extremely branched out. There are a lot of branches. The upstream part is usually called the mevalonate pathway, and it contains the reactions that are usually targeted by cholesterol lowering agents like statins, for example. But the point that I want to make here is that because of this ramification and complexity, the sterol synthesis pathway produces a number of products beyond cholesterol. That is the most notable, of course, and these byproducts really impact a variety of cellular functions. So, spoiler alert, we found that this pathway is critical for tumor initiation. And to talk about pancreatic, PDA initiation, I have to take a few seconds. Pancreatic epithelial cells that constitute the vast majority of the parenchyma. They are probably boring cells. They're postmitotic, highly specialized. They secrete the digestive enzymes in the pancreatic duct to help digestion. So they don't really do much normally. But when KRAS is mutated, they undergo a dedifferentiation process and eventually acquire more ductal-like phenotype. This is usually a reversible process. It occurs also in physiology. For example, during tissue regeneration after injury, it's called acinar to ductal metaplasia. Again, first a physiological process. But when KRAS is mutated, they are not really able to go back to their original state. They are somehow locked in the ductal-like state, and they eventually expand and proliferate, expand, and give rise to prenoplastic lesions. So in this sense, acinar to ductal metaplasia, how we use the acronym IDM for simplicity, is really the initiation event in pancreatic carcinogenesis. And you can easily model that in vitro by plating primary acinar cell clusters in matrix or collagen in a 3D matrix. And you can appreciate that only after a couple of days, these clusters that, you can recognize the characteristic grape-like structure immediately after plating, they progressively lose this very neat morphology and start to acquire a more flattened circular structure characteristic of ducts. So you have a way to mimic in vitro acinar to ductal metaplasia. And when you treat these acinar cells with statin, you can see that they are not able to undergo ADM anymore, and they retain the acinar morphology throughout the whole experiment. ADM, however, is rescued by the addition of cholesterol. And we later found that the sole addition of cholesterol is able to promote ADM even in wild type acinar cells. We thought this was interesting because cholesterol has long been thought as a structural molecule with no business in cell function. But we now know that, of course, cholesterol derivatives participate in cell-cell communications, but also more recent data showed that the ability of cholesterol to impact signal transduction through non-canonical mechanisms. It impacts receptor activation, extracellular and intracellular signal transduction, and even mTOR recruitment and sensing at the lysosomes. So again, sorry to cut a long story short, but we found that these actually do not take place during ADM, but we found that cholesterol addition induces the generation of cyclic AMP, and this activates protein kinase A, as you can appreciate in this plot. For that the stains phosphorylated proteins, which are pKa substrates. And importantly, we found that pKa inhibition suppresses cholesterol-induced ADM ex vivo. So, to summarize this first part, we found that acetyl-CoA feeds cell synthesis during acinar to data metaplasia to produce more cholesterol that in turn activates pKa, that is a signaling event required for the metaplastic events. We think there is more to that, so pKa is not the only cholesterol-induced signal event, but for now, this is what we published. Another spoiler alert: we found that sterol synthesis is also critical at advanced stages, and we and many others really found that PDA cells are universally sensitive to statin treatment in vitro, as you can see in this small panel. But this is something that is really true across different cell lines. What is interesting is that in this case, the addition of cholesterol that you can see in this last bar of the plot on the left does not rescue cell growth in vitro, but upstream intermediates do, and this is really consistent across different cell lines. And to understand why, I need to go back to this slide and highlight the fact that many of the intermediates in the cell synthesis pathways are electron acceptors and are able to stabilize redox stress. That is something that, of course, cancer cells experience. And indeed, when we treated pancreatic cancer cell lines with statin, we found a dramatic increase in oxidative stress, and as you can appreciate in this staining, we used two different probes that measure whole cell and mitochondria specific reactive oxygen species, and this oxidative stress is quenched by the addition of mevalonate, that is an upstream metabolite intermediate in the sterol synthesis pathway. And here is a quantification for the cytoplasmic. Sorry, I messed with animations, but in any case, ROS production is important for cell viability because it determines sensitivity to ferroptosis. That is a non-canonical program cell death mediated by the overload of peroxidated lipids. We can actually measure peroxidated lipids in cells using a specific probe, the C11 body pi, that shifts fluorescence when it is oxidized. And you can see that simvastatin treatment really induced a massive accumulation of peroxidated lipids in these cells, which is again rescued by the addition of mevalonate. So this is consistent with the hypothesis that again acetyl-CoA feeds the cell synthesis pathways to produce a number of intermediates that stabilize redox stress in these cells, thus protecting these cells from ferroptosis. When cell synthesis is inhibited, ferroptosis is simply unleashed and these cells die. So again, I'll stop here. Of course, we will start from here to understand. We are now working on understanding additional signaling roles of cholesterol, and of course to understand the differences between in vitro and in vivo vulnerabilities, but most importantly to really understand why diet is so important to determine the prognosis of pancreatic cancer, especially high cholesterol diets are really really drive a worse phenotype. And thank you for your attention. Again, sorry for not being there in person with you, and be still happy to take any questions.
Thank you very much for this presentation. This is now open to questions. Yes, of course. Yes.
Yeah. Thank you for your very interesting talk. You mentioned a couple of effects of statins, and I was wondering which concentration you use, because statins are being used extensively by patients, and we have found that at a non-toxic concentration, some drugs are being activated, and resistance to some of these drugs is bypassed. But only this was found at concentrations which are achieved in patients. So I was wondering whether you, which concentration you use, whether this was this type of concentration, or did you use much higher, because we had to use much higher concentration to get cell death.
Yes, so in vitro we use concentration between 10 and 20 micromolar. For my cancer discovery, I did try in vivo administration. I honestly, that was long ago. I don't remember off the top of my head what concentration I used. In any case, in vivo, at least at the concentration I used, I couldn't really see a strong tumor inhibition. We used just xenografts, probably because of the compensatory mechanism in vivo that we are now trying to understand. But yeah, so I'm really sorry. I don't remember off the top of my head what concentration we use in vivo.
Above the level achieved in patient, but it depends a little bit on the statin which you use, because you use several statins.
Yeah. Well, in vitro we use like 10 to 20 micromolar in vitro.
Thank you. And it was
yeah,
very
hello.
nice story.
Myself is Dr. Manoj Garg. We work on the pancreatic cancer. So my first question is that when you knock out acetyl-CoA, do you see these mice have a lethal effect, or it is homozygous or heterozygous knockout?
Sorry. No, we do so. These are conditional, not to tissue-specific ACLY knockout mice. So the lack ACLY only in the pancreas. No, the mice are alive, and for what we tested, they're perfectly normal. They do not have like any histological or metabolic abnormalities. So, for example, insulin levels, glucose levels are just fine. When we bred them with the KC mice, and that's oncogenic KRAS, so we saw the strongest effect is on tumor initiation after the tumors form, and we reported that in the cancer discovery. After the tumor form, actually they upregulate other enzymes, and for example ACSS2, and they're equally able to produce acetyl-CoA through other routes. Okay. And they proliferate fine, so they don't have like major like.
Okay, so my next question is that as you know that KRAS is responsible for ROS and then ferroptosis and then this whole
mitochondrial reprogramming
pathway. Do you think it is mediated by KRAS because you are using KRAS as a mutant in your knockout model or that thing? Do you rule out it is because of acetyl-CoA or it is mediated through KRAS itself?
My first guess is that KRAS has a much stronger effect because it has been reported that it too strongly reprograms mitochondria, both in shape and function. If ACLY has an impact in that, I don't think so. ACLY is not even a mitochondrial enzyme, so I doubt. But yeah, I haven't directly checked that. Yes.
Okay, thank you. Thank you very much.
Another question here before we go to the online questions.
Hi, actually, this is HP here. I also work on a pancreatic cancer. So my team is more on the sensitizing the pancreatic cancer for the immune-mediated attack. Now, in your cartoon, what I want to ask you, whether blocking cholesterol pathway in these pancreatic cancer cells make them immunogenic for the immune cells, number one, and by blocking cholesterol synthesis, you see a significant drop in the survival of these pancreatic cells. So by blocking cholesterol, what all other lipids compensated in these tumor cells which withdraw them from the survival signal? These are the two questions.
Very interesting questions. I don't think I have an answer for either. We haven't directly checked that. Keep in mind that we are trying to block de novo synthesis of cholesterol, but cells are still able to uptake from the circulation, and of course pancreatic cancers are really starved and avascular, but there's still cholesterol around from dying cells, for example, so they can still uptake most of what they need from the microenvironment. So yeah, if it is if it can impact, if it's also true the other way around, if it can impact the microenvironment and inflammatory recruitment, I guess it's possible. We haven't directly checked.
Because why I'm asking this question, you see, you connected this to the cyclic AMP, and the cyclic AMP upregulation trigger the HO1 signaling in the immune cells. So this promote the immaturation of the DC.
Oh, I see. No, I didn't have the time to show that, but we reported in the metabolites paper. No, that sensing mechanism is actually lost in PDA cells. So it takes place during ADM in acinar cells, but for some reason, pancreatic cancer cells lose the sensitivity to cholesterol, to activate pKa in response to cholesterol.
Yeah. Okay, thanks. Okay.
no problem.
Thank you. So we have now a few questions from the online. Okay. Questions? If there is no question there, okay, then we go.
I think we have to read this question.
Yeah. Dr. Alessandro, are you still there?
I'm here. I'm here. I'm here.
We are still waiting for eventually the online. Yeah.
Sure, absolutely.
So, okay.
We have to read.
Okay.
We have to read that question.
Yeah. So,
Yes, second one.
no, which one? Garvey?
Did you check how? Oh, let me.
Yeah. What do you see? Where do you see?
Can I jump?
Yeah.
Hello,
Hello?
the question is, did you check how protein kinase A was getting activated by cholesterol? Which GPCRs were involved? That is the online question from Gargi Bakchi.
Yeah, good question. Yes, we checked through threat-based sensors. We actually use sensor for adenylate cyclase. And we saw that adenylate cyclase was were activated, specifically the soluble forms that associated with organelles. That implies that cholesterol needs to be internalized and travel within the cell. So we don't believe that any receptor is possibly involved, but rather than cholesterol is transported within the cells, gets to different cellular compartments, and there activates adenylate cyclase, probably, but we don't know, by anchoring the enzyme to the organelle, so some physical constriction. But yeah, we haven't really looked at the mechanism specifically.
So, Dr. Alessandro, we will have to stop here for the night. We'd like to thank you very much for your this online presentation. And now this is open for the following. Thank you.
Thank you.
Thank you, chairpersons. So now we have a interactive session. So here both online and offline participant can interact with the speakers. Already we have 3 speakers with us here. So I request IT to bring all online people on the screen. So you can interact, you can ask even more questions also. This is a special slot for the interactive session for both online and offline participants. So I request IT to bring all online people on the screen. Yeah, you can also ask any questions for these 3 speakers. Any queries from offline? Okay, I think there is no online questions. So we will have the tea break now. So then again, we will assemble at 11:15. Yeah, that's a PowerPoint, yes. This one, right? This is the next exam. Want to make it slide more? Okay. Okay. Now I request session chair, Dr. Andrew Westwell and Dr. Reshma Rani. Please come on the dais. Dr. Reshma. Yes, you can start soon.
Okay, good morning. Welcome back. My name is Andy Westwell from Cardiff University in the UK, and it's my pleasure to be chairing this session alongside Dr. Reshma Rani. The first talk of this session will be given by Elisa Giovanetti. Elisa is a professor in the Department of Medical Oncology, the Amsterdam University Medical Centers, BU University. Elisa is a specialist in cancer pharmacology with a special interest in pancreatic cancer and mesothelioma. She has been the recipient of a large number of awards and recognitions, including the EORTC Leadership Program and the Award of Excellence from the International Group of Endovascular Oncology, and many others, many other awards. She's very well published, 337 articles in referee journals, H-index of 62, a very impressive publication record, and one of the leading scientists in the field of cancer pharmacology. So it's a great pleasure to welcome Elisa to give her presentation. Thank you.
Thank you. Thank you very much for your kind word. And it's a pleasure to be here. I'm going to share now my screen. I hope everything is fine. Can you see my presentation? I don't hear for you.
Not yet. Yes.
And now, can you see it? Okay, just a moment. I will try again. Sorry. So, share screen. This one, share. Okay, and now can you see my presentation?
Yep.
Okay, perfect. So, thanks for the opportunity. Giving this presentation, I deeply apologize for not being able to be with you. And I would like to thank again the organizer for the excellent work and how they managed to organize this great workshop. And here in Europe, actually I'm in Amsterdam, it's pretty early in the morning, so I hope you will forgive me if I start with this slide, because we all need some energy, and we know, of course, that also cancer cells do. And since the second edition of the famous Hallmarks of Cancer, indeed, the regulating cellular energetic has entered among these features of cancer cells, which essentially we know differ from healthy cells because of several molecular changes that have been already presented many times in excellent presentation before this. And typically we call these metabolic reprogramming. And this stays also in the last edition of Hallmarks of Cancer published recently. And what is the clinical significance of such metabolic reprogramming? As reported in this review by Torresano and collaborators, there are many studies summarized here that essentially show that the regulating metabolic enzymes affect the prognosis, and in particular, in general, an upregulation of enzyme involved in glycolysis, the pentose phosphate pathway, lipogenesis, antioxidant, and glutaminolysis, as well as a downregulation of enzyme involved in oxidative phosphorylation, fatty acid oxidation, and pyruvate oxidation, which are depicted in the green and red line in this survival curve. They are related to a worse prognosis. Focusing more specifically on enzyme of glycolysis, a factor of glycolysis of the pentose phosphate pathway, they are typically considered potential cancer biomarkers, and there are a number of studies which shows that the protein levels that have been checked with, in most cases, immunohistochemistry, some cases also with Western blot, are upregulated in tumor tissue compared to normal tissue. And this is related to an unfavorable prognosis. And this is in particular what is happening, and I will discuss later, mostly, for instance, for the transporter GLUT1 and for the enzyme LDHA. So in my next slide, I'm going to introduce this main actor, which play a key role in the glycolytic switch. That has been already described several times. I thank Professor Minutolo, who will speak after me, who gave this light to me many years ago when we started our collaboration. The idea is that in normal cell, the glucose is transformed in phosphoenol pyruvate, and then the pyruvate from the cytosol goes to the mitochondria, and there it interacts with several enzymes, and it enters in the Krebs cycle, and then there is oxidative phosphorylation, bringing the production of a total number of 36 molecules of ATP. But in the cancer cells, the fate of this metabolite is different because of the Warburg effect that has been nicely described already in the previous talk. And LDH play a key role and essentially catalyze the synthesis of lactate starting from pyruvate. And this brings to the production of very small amount, let's say the truth, of ATP. But as underlined by Professor Poussigliere in the keynote lecture of yesterday, this is still, unfortunately, something that is very useful for cancer cells. There are indeed many reasons that make this metabolic reprogramming an advantage for more aggressive cancer cells, because ATP might seem less, but it's produced much more rapidly, and this favor also the increase in the biosynthesis of many fundamental molecules for cells which are actively proliferating. There is also a protection from the production of the reactive oxygen species in the mitochondria, and there are several important effects on tumor microenvironment, as also shown in this nice figure, which I take, by the way, from a review of the organizer of this meeting. And they clearly show that indeed the glycolytic metabolism in tumor cells assure that the ATP synthesis is going on also when there is hypoxia, which can happen when the tumor is growing. And it also somehow creates space because the cells nearby the tumor are starting to be starved. And somehow there is also a kind of sometimes metabolic symbiosis shown here between the core of the tumor, where there might be some hypoxic and glycolytic tumor cells, but this is a dynamic process, as underlined also yesterday by Dr. Kreuz. And so there are also normoxic cells, and there is a kind of symbiosis which is favored by the shift of lactate, as shown in this picture. And lactate for sure has a very important role in more aggressive tumor cells and also tumor microenvironment features, because it increases the angiogenesis and also the EMT process, essentially favoring the motility and migration and metastasis. And as already discussed this morning, of course, the role in the immune system is also very important because it favors essentially immune escape. So, as a summary, in this slide, we could say that lactate and the lactate dehydrogenase, which is the key enzyme in the production, have for sure an important role in metastasis. But as pharmacologist, I'm also very interested about the role in resistance, and this mostly because in this situation there is an increase in the activity of the system which are able to repair the DNA, and so the tumor cells, despite the mutation accumulating during carcinogenesis, essentially are actively proliferating, and they became more resistant to the drug. And it's not only against the, let's say, conventional chemotherapy. Indeed, here I am also showing a recent paper which was published in the European Journal of Cancer, which showed that among many factors that were studied in a study which is a multi-center study with more than 400 patients, what was standing out among the predictor of resistance to anti-PD-L1 immunotherapy, so a new therapeutic approach, is the presence of high levels of serum LDH and also a liver metastasis. So, focusing on the clinical significance of these features. As I said before, I will mostly discuss results on GLUT1 and LDH5 because there are many studies that show that these effectors of glycolysis are important in tumors which are more highly invasive and typically also hypoxic, such as pancreatic cancer, as you can see here in immunohistochemistry staining of LDH. It's obviously much more expressed in cancer compared to the relatively normal adjacent tissue. And it has been associated with poor prognosis, not only in pancreas, but in many tumor types. Moreover, we can check, in particular for LDH, also the levels in plasma. And these are very useful in the clinical set because they are typically used as biomarkers for malignancy because they have been associated with prognosis. So here I have a summary of the main feature of LDH. LDH is, as you know, tetrameric enzyme with two types of subunits, and essentially they are combined differently in these isoforms, which then are expressed in the different tissue. And we know that in particular LDH5 is more expressed in liver and muscle and LDH1 in heart. But essentially, when we look at the serum, what is important is that it's there at a certain level and is typically increased when, unfortunately, a patient is undergoing some severe disease, such as myocardial infection. This is mostly LDH1, and different types of tumor. And in general, it's measured as LDH. And the interest in the prognostic role of serum LDH is long-lasting, and we can see from the articles in PubMed just making a search for serum LDH cancer and prognosis in the last 40-50 years. And there are many papers essentially collecting data from different tumor types. Here I am reporting the largest meta-analysis. And so this one included data from more than 22,000 patients. And they were selected because they had advanced stage solid tumors. And this study included 73 clinical studies, and this is the flow chart showing how these studies were included in the quantitative analysis. And what I can already tell you, and it's going to be shown in the next slide, is that high serum LDH levels were very strongly, significantly associated with reduction in overall survival. Here, indeed, you have the forest plot. Of course, there are so many studies, it's really almost impossible to read in one slide, but what I want to underline is that it's obvious that the hazard ratio is above 1. And here in the funnel plot, the authors essentially summarized by tumor type, and the studies are represented by one circle. And here you can see that essentially, again, the hazard ratio, the average hazard ratio is definitely above 1. So it looks like that serum LDH is a very strong biomarker for prognosis, but still these studies have limitation because it's not always clear. It depends a lot also on the different laboratories, which is the threshold, the cut that we should take. Here in these studies, they made at the end a median, which is pretty similar to the one that is typically used, 245 units for liter, but it can change. And indeed, when they did the meta-regression analysis, it was not significant. So there are still some limitation and some standardization that should be improved even in this field, even it's for many years that it's studied. And then I would like to focus on the prognostic role of LDH when we are looking at this enzyme in tumor tissue. Again, I'm going to present some data of a meta-analysis. In this case, more studies, 86, but a little bit less patient, 12,000, still a pretty big number. And in this study, they evaluate LDH, but also other glycolytic related protein, but definitely LDH was the one who was emerging. And it was associated with, for sure, shorter survival in many GI tumor, gastrointestinal tumors, but also in breast cancer. So I was pleased to see the presentation of this morning, and also oral and nasopharyngeal cancer. And again, the range of hazard ratio is definitely above 1. And in the study, they also evaluate some feature of this tumor. And typically, LDH is more expressed when the tumor are poorly differentiated and they are bigger, so in advanced tumor stage, so typically metastatic, and indeed there is positive lymph node and also vascular invasion. So I told you many tumors have been collected. Our contribution to this study was the evaluation of LDH expression in a tumor which is not studied by, unfortunately, many authors because it's pretty rare, is mesothelioma. And here I am reporting the results of our analysis, which was performed on a tissue microarray, collecting essentially tissue from blocks of patient affected. In this case, you can see by diffuse malignant peritoneal mesothelioma. And we found that the high expression, unfortunately, was correlated with progression of the tumor, as well as with a significantly shorter survival. And we were able to validate this findings also in pleural mesothelioma. So we have indeed two main types of mesothelioma. Since this is not so common tumors, we were really interested in try to find a biomarker because it's a rare tumor, but unfortunately, because of the exposure to asbestos in many part of the world is still present, it's expected to peak in the next few years. And it's a tumor which has, unfortunately, a bad prognosis with limited treatment available, median survival of about 1 year, in particular for the pleural mesothelioma, and pretty severe symptoms. So we feel that we need for sure more effective treatment and hopefully also biomarkers that can help to select the patient. What was coming out almost 10 years ago was a drug called pemetrexed, which was associated with the previous already used therapy with platinum compound. And so we were interested in finding determinants of the activity of pemetrexed. As I told you, it's an antifolate, and these are the three main targets of pemetrexed. And in particular, the key enzyme here seems thymidylate synthase. And therefore, we decided to evaluate the mRNA expression of TS in a number, two cohorts of pleural mesothelioma patients treated with pemetrexed-based regimens. And we found a slight, significant correlation with survival, as you can see from the p-value, which are around 0.02. And then we wanted to improve that, and then we look at what is also important in the activity of pemetrexed. Well, we know that pemetrexed can be extruded by the cells, but this does not happen when it's polyglutamated, and this is thanks to the activity of FPGS. So initially, we look at some splicing variant of FPGS, but these were not so strongly correlated with outcome. And then our focus went on a dime pretty new transporter, which was discovered, proton coupled folate transporter, which is important, in particular in acidic environment, for the entrance of pemetrexed in the cells. Therefore, we investigate the expression of this transporter. And this time, we perform our analysis evaluating both the mRNA and the protein levels, again, using different cohort of patients and trying to also combine our data with the previous data of TS expression at mRNA level. And as you can see, when we get also more consistent numbers, so about 120 patients when we made the expression analysis at mRNA, we found that definitely the use of both markers was helping us to identify a subgroup of patients who unfortunately had a significantly shorter prognosis because they have high TS and low PCFT expression. And as I told you, we were able also to set up an immunohistochemistry here thanks to an antibody which was made at Karmanos Institute in US by Larry Matley. And essentially this, of course, is very useful because the pathologists would like to have this kind of tool to evaluate the expression in the tissue. This finding prompt us to make further studies to try to explain what is happening, why PCFT is so correlated somehow when it's poorly expressed with poor prognosis. And we found out that mesothelioma is a highly hypoxic malignancy. As you can see from pretty old study by the group of Dean Farrell, that show that there is an increase in the expression of IF1 alpha in nuclear localization in mesothelioma slides. And most recently, the group of Professor Carbone found also a very interesting correlation between IF1 alpha and BAP1. BAP1 is a tumor suppressor gene, which is pretty frequent in patients with mesothelioma. And the interesting aspect is that it's for sure important in carcinogenesis. It's a tumor suppressor, and there are inactivating mutations. But is also correlated with family cases and with a little bit better prognosis. Indeed, these studies show that this may be attributed to the fact that BAP1 mutation seems to reduce the IF1 alpha expression and then the aggressiveness of mesothelioma. What we observed in our tissue when we went back indeed to the same patient block where we performed the PCFT analysis, that we had low expression of PCFT, where in the tissue there was a high expression of carbonic anhydrase 9, so a marker of hypoxia. And this was essentially a kind of almost a reverse correlation. Then we decided to study in our preclinical models what could happen here. And we use a cabinet for hypoxia. We use spheroids with a core that show sign of hypoxia and also specific silencing for PCFT. And then we check the transcriptomics, what was happening. And definitely overexpression of LDHA was standing out, as you can see from this Vulcana plot. And then this was further validated, the transcriptomics data with PCR. And also, as you can see from the Western blot at the protein level, in particular here, I reported the data of the expression of LDH, which is clearly increased in the spheroids compared to the monolayer. So these are the tools that we use, the hypoxia cabinet, the spheroid, and the hypothetical model that we try to build was that essentially in hypoxia, as when we have the spheroids, essentially because of the epoxy core, we have an increase in IF1 alpha, which we know is an important transcriptive factor and promote the expression of LDH. But in the meantime, there is a downregulation of NRF1, which on the other hand, is most probably related to the downregulation of PCFT. And we think that there might be an hypothetical feedback that it's also playing a role, most probably, to control intracellular acidity. And we are performing further study for that. But what we also would like to see, because I told you we are pharmacologists, is if we can target some of these specific features, in particular in this group of patients where we have low PCFT and high LDH. And as I said, thanks to the collaboration with the group of Professor Minutolo, and I know also that Dr. Rani was working in the lab there, and we collaborate to some studies. We were able to use a series of these compounds, and in particular for these studies, we used the NH glucose 2, which essentially is conjugated with glucose, so it enters better in the cells through GLUT1. And we performed some studies, some initial studies in pancreatic cancer, but then also in our models of mesothelioma, both in the spheroids, where we definitely see a shrinkage, a reduction in the density of the spheroid after treatment. And also in our orthotopic model of peritoneal mesothelioma, where with some ultrasound we were able to detect a kind of hypoxic area, which was further confirmed after the end of the experiment in the tissue by the pathologist. And we observed definitely at least small reduction, not yet significant, but still there is a reduction of the tumor volume as measured by fluorosilferase in this tumor in the mice. And I can tell you that in the next presentation you will see the rest of the story, so how we decide then to combine with other drugs. But to finish my talk, I would like also to discuss a little bit the feature of GLUT1, because glucose transporter also are studied as potential biomarkers. We know that in general there are a wide group of membrane proteins, and each glucose transporter isoform plays a different role depending on the different tissue and the stage of life. And in particular, GLUT1, which is encoded by the CLC2 gene, is highly expressed in fetal tissue. In adult, it's expressed mostly in the erythrocyte and also in the endothelial cells of the blood-brain barrier, but it's also overexpressed in many tumors, as you can appreciate from this slide from the TCGA repository. So essentially, there is a pretty good number of tumors, which are depicted in red, where you can see a significant overexpression of GLUT1 compared to the normal counterpart. And as example, I reported here, for instance, what has been detected in esophageal cancer. And these are the graph, of course, of the TCGA is showing the expression at the mRNA level, but this has been also confirmed by studies with immunohistochemistry. So again, I'm showing another meta-analysis, but this time for GLUT1, and this time it includes 27 studies, so it's a little bit smaller compared to the previous meta-analysis for LDH, but still a good number of patients have been included. And the authors have checked carefully their characteristic, and they definitely observe a prognostic role, and they had both the data on survival and also on disease-free survival. So the expression of GLUT1 is prognostic, but also important to understand if the tumor, unfortunately, will progress. And they also check other clinical features, and as reported in this slide, in particular, they found that the overexpression of GLUT1 is also linked with the poor differentiation of the tumor. So tumor with a higher grade 2, grade 3, are the one which unfortunately have the highest expression of GLUT1. And again, with metastasis, this was checked looking at the number of positive lymph nodes, and with tumor which are bigger, so they have a larger tumor size. They also check for abnormal expression of P53, and here essentially, as you can also already observe from the forest plot, the difference seems a little bit less significant, let's say. So most probably these should be further investigated. It's not so strong as the other data. So GLUT1 seems also a good biomarker to be further validated, and immunohistochemistry again method should be standardized also for this kind of protein. Again, we tried to think about the potential use as a target, and this was a little bit a kind of study, which was coming out by serendipity, because the study started with the analysis of phospho-AKT, and we observe in this kind of pancreatic cancer tissue that high expression of phospho-AKT was correlated with worse prognosis. And from some of this tissue, we were able to obtain primary cell culture. And we had, in particular, two cell culture primary that somehow were representative of high and low expression level. And we observed that in the cells with low expression level of phospho-AKT, there was essentially lower sensitivity to gemcitabine, which is the standard drugs used in this tumor type, but also to an pro-apoptotic drug, periphosin, and also their combination was antagonistic and not synergistic as we observed in the cell line with high phospho-AKT level. When we check at the feature of these cells, one which was emerging was that the more resistant cells, let's say resistant to apoptosis induction, had a higher expression of GLUT1. And then again, thanks to the collaboration with Professor Minutolo, we had some compounds, and in particular this one, PGL13, inhibitors of GLUT1, which by themselves, they were not really active. Concentration inhibiting 50% of cell growth was pretty high in the level of 20-30 micromolar in these cells, which are definitely also very aggressive cells. But in the combination with gemcitabine and periphosin, we definitely see a reduction of cell growth and also in particular an increase in the induction of apoptosis. So we concluded our, we concluded our paper with this hypothetical model of the positive interaction between these two different drugs, gemcitabine and periphosin, with a compound which essentially inhibit the entrance of glucose through GLUT1 and then most probably inhibit the inhibition of apoptosis. So, in conclusion, in this talk, I wanted to highlight, and this is pretty evident in such a workshop, that metabolic reprogramming is a very important hallmark of cancer. And it plays a role in the clinic because there are key effectors, particularly focused on a glycolytic pathway, which have been associated with clinical outcome. There are many biological causes. I explained, as already reported by previous speakers, that these features are associated with essentially more aggressive phenotype, both considered in tumor cells and the tissue microenvironment, and also, unfortunately, resistance to different treatment. And in particular, I focus on LDH and GLUT1, which definitely resulted overexpressed, as I shown by the data from pretty extensive meta-analysis, associated with poor prognosis, and they might be further developed as potential biomarkers and hopefully also as therapeutic target, even if many more studies have for sure to be performed, also in particular to optimize a smarter combination. And with this, my last slide, I think that indeed, as you can also see most probably in the next lecture, we hope to get there. It's not easy to catch the tumors, to really be able to fight with this kind of aggressive tumor, but hopefully something is coming from therapies targeting anti-LDH and anti-GLUT1, particularly if we are able to play smart, to identify the group of patients that should be targeted, and also to identify novel combination that might be more effective. Thank you very much again for the kind invitation, the attention. These are the acknowledgement to many people I'm lucky to work with, and all your questions are welcome. Thanks.
Thank you very much, Elisa. Any questions, please?
So thank you for the wonderful talk, Elisa. I have a question that you have mentioned about the meta-analysis you have done to study the prognostic role of GLUT1 in tumor tissues. So when I saw your data, the number of normal patients were lesser compared to with the disease set. So does this statistically affected your downstream analysis?
Okay, yeah, this is always a problem. Indeed, essentially, most studies, also when I show the first table, the table of the review of Torresano, it's in most cases we have data on the expression on the tumor and not always on of the normal adjacent tissue, the counterpart that we should always use. But the meta-analysis were considered anyway also by our biostatistician pretty strong because at the end they were able to group good number of studies. You know, essentially the one with the lowest number was 27 studies. So essentially we still were able to reach a good level of let's say numbers that can give a statistical significance. But we are aware of this problem, and indeed we should always be very careful in collecting both the tumor tissue but also the information from the normal adjacent tissue when we want to evaluate a biomarker. And anyway, it was also interesting in this meta-analysis when it was possible to compare the data with other clinical features, such as the grading, the lymph node invasion. This really also makes the point that GLUT and also LDH are good biomarkers because they really seems more expressed in the most aggressive tumor.
Yeah, so because this I have seen in many of the cancer data set that this is a problem. So it would be good if I increase the number of studies whenever I'm doing such an analysis.
Yes, definitely.
Thank you so much.
Thank you.
There is one question online. Elisa?
Yes, thank you.
I will read it. Can lactate affect the epigenome by lactylation of histones?
Oh, this is an interesting question, but I don't know the answer. I mean, lactate has a lot of effect, but I should study about that. I don't know about the lactation of histones. I don't know if someone in the audience has some knowledge about that.
This is an anonymous attendee. Okay.
Okay. Thank you. I think it's something to check.
Yeah.
Thanks. Thanks.
Hi, Elisa. Myself, Dr. Reshma. I have one question, not a just question, it's like a query. So LDHA, the isoform, there are 5 isoform and all are almost similar in structure. So is really selectivity of LDHA matters?
Well, this, I think after the talk of yesterday of Dr. Posiguera about LDA, because when in particular we start our collaboration with Professor Minuto, that I'm also happy to see, and I start learning about this topic, the idea was to try to have indeed the more selective LDH inhibitors with the isoform A in order to have essentially also less toxic effect. But it's true that also many companies try to go in this direction, but as shown yesterday by Dr. Posiguer, most probably we should be more careful and also think about indeed targeting LDHB, and I think also other component of this glycolytic pattern. I told you in the next lecture for sure there will be indeed some more insight about how try to indeed develop better drugs and also check different component of the glycolytic pathway.
Okay, thank you. Thank you so much.
Thank you.
Okay, thank you very much. No further questions. So thank you again, Elisa. And we'll move on. So it's a real pleasure to welcome Professor Filippo Minutolo from the University of Pisa, Department of Pharmacy. Filippo is a professor of medicinal chemistry, full professor since 2016. He originally did his PhD in chemistry in Pisa, and including a year at the University of Groningen in the Netherlands, in the group of Ben Feringa. And then following that, he was carried out postdoctoral research at the University of Illinois, Urbana-Champaign in the USA, in the group of Professor John Katzenellenbogen. So he has a 30-year experience in the design and synthesis of bioactive molecules, and his work has resulted in 146 peer-reviewed articles with an H-index of 37, several international patents, and numerous Congress communications. In common with all of the speakers in this session, he's a member of the EORTC group, the PAM group. This is the theme of this session, the Pharmacology of Molecular Mechanisms group of PAM. And he served as a panel member in various national and international funding campaigns. So with that, I'll pass over to you, Filippo. Welcome again, and look forward to your presentation.
Okay, thank you very much, Andy. I'm really happy to be at least online with you. And it's a pleasure to see all those people that I've already met before, like Reshma, that she spent 2 years in my lab, so it's really nice to see her chairing the session together with you, Andy. Now I'll start to share my presentation. Please let me know if that works. Okay. Can you see my presentation?
Hello. Yes, absolutely.
Yeah, okay. Perfect. Okay, so I'll start with that. So I would like to thank the organizing committee for inviting me and giving this talk about some new prospective therapeutic and diagnostic agents that we're trying to develop that exploit tumor glycolysis. I'm really glad that Elisa basically introduced most of the concepts that are at the base of the research that we're doing about exploiting some of the targets that tumors overexpress to increase the glycolytic flux and survive in harsh conditions. I will briefly show you the way we want to exploit a few targets like the glucose transporters and the lactate dehydrogenase for both diagnostic and therapeutic purposes. And then later on, I will just briefly introduce a combination of drugs that we're currently experiencing in LDH and the PDK inhibitors. But this subject will be mainly discussed by the next speaker, Fritz Peters, that finally managed to reach you in Noida. And I'm really jealous because I really would have liked to be there with you, but unfortunately for some work purposes, I wasn't able to leave my institution. Now, let's start with the discussion of some novel diagnostic probes that we're trying to develop to highlight the overexpression of glucose transporters in cancer cells. Now, you're aware that the most widely diffused technology to scan tumor metastases and tumor sections in the body are based on the FDG PET scan technology. So the FDG is fluorodeoxyglucose labeled with the radioactive fluorine atom, which is shown here, which is a mimic of glucose. And this molecule is actively taken up by glycolytic cancer cells by means of the overexpression of GLUT1. And the FDG, once it is taken up by cells, then it becomes trapped inside the cells by the action of exokinase, which promotes the phosphorylation of the 6-OH group. So the FDG radiopharmaceutical agent is not able to exit the cell anymore. But at the same time, it doesn't get metabolized. So it accumulates and emits radiation so that the PET scan camera can get images. Now, this so widely diffused technology is all based on the fact that cancer cells have a higher uptake of glucose, of course. But it's based on radionuclide production. So it is associated with a series of problems due to radioactivity of the material produced, of the waste, and also of the exposure of patients to these radiations. Now, our idea was trying to find a possible novel technology that could avoid the use of radionuclides, if not for in vivo images, at least for, let's say, in vitro visualizing the glycolytic features of cancer cells and tissues. So we started to think about other technologies such as vibrational imaging, working in the infrared spectrum. Which are the advantages of infrared spectroscopy? Well, basically, it's a very cheap technology. It is non-hazardous. It doesn't require any radionuclide. Doesn't experience photobleaching, which is pretty common with the fluorescent probes. It's quite easy to implement and potentially can be extended from cells to also to solid and liquid specimens, if not to whole bodies, as we will see in a while. On the basis of this idea, I just had to consider that cells have some absorption in the IR spectrum. The visualization of glycolytic cancer cells by means of IR probes should avoid those regions of the spectrum that are highly absorbed in cells. You can see in this figure the infrared spectrum of a human breast ductal cancer carcinoma cells, the MCF7 cells. And you can see that many components of the cells have some strong bands of absorbance. For example, the lipid absorb above 2,800 with this stretching of the CH2 and CH3 bonds. Then the protein have very strong absorptions in the amide-1 and amide-2 bands around 1,600. Nucleic acid by means of the phosphate group absorbed in the 1,200 region, and also carbohydrates have strong absorption between 1,000 and 1,300 region. So we basically needed to exploit this portion of the spectrum, which is called mid-IR transparency window of the cells, which is basically around 2,000 and 2,400 wave number. So I started to think about which probe I could use for exploiting a strong absorption in this transparency window. And as many, let's say, scientific inspiration comes out from strange considerations. One Sunday afternoon, I was looking at my dining table. Actually, I had completed quite an extensive refurbishing of my apartment quite recently, and I got some new furniture, and this is a new dining table I was able to purchase. And actually, I wanted to buy 6 chairs because there was space enough for 6 chairs around this table. But my, let's say, family budget limitation imposed me to buy only 5 of these chairs. And this constraint actually turned out to be quite a fortunate event for the inspiration of what I'm going to show you in a while, because by combining the observation of these 5 chairs, this kind of flat top of the table and the 3 legs diverging from a central portion of the table actually reminded me about some complexes I was working on quite some time ago, which are the cyclopentadienyl rhenium tricarbonate complexes, whose X-ray structure is shown here. And you can see there are pretty high similarities with my dining tables, and you can see the flat top, which correspond to the aromatic portion of the CP ring, the 5 atoms, 5 chairs. The central crossing point is the rhenium metal atom, and the 3 legs of the table are the 3 carbon monoxide ligands shown here. Now, this complex are actually very stable. They do not undergo hydrolysis in aqueous environments. They are pretty lipophilic, so they do not have any specific strong interactions with polar portions of bioactive molecules. They are also actually rather small because they just occupy a limited space. It's a kind of, let's say, lipophilic sphere, which can be actually attached to biorelevant portions without substantially modifying their possible interactions with their targets if these complexes are placed in the proper position of these ligands. So I thought, well, maybe this can be a good IR probe, because the feature of this probe is that it is characterized by a strong IR stretching, so infrared absorption due to the stretching of these carbon monoxide ligands. And as a matter of fact, if you look at the typical band of absorption of these complexes, you would see that this is a kind of zoom expansion of the area, which is right in the middle of the transparency window. So you can see here wave number of 1950 here, and slightly above 2000 in this other peak. These 2 peaks are due to the symmetrical and asymmetrical stretching of these CO ligands, which are characterized by a higher energy for the symmetrical stretching, which is less populated, by the way, because there's only one possibility to have a symmetrical stretching, whereas the asymmetrical stretching can have 3 different possible population of stretching. So it's 3 times as much as intense, and it comes at a slightly lower energy of absorption. So these 2 peaks are placed right here around 2000. So it could be a good region of the spectrum to be exploited for IR imaging of cells. Now, I started to look at the literature, and actually I found out I was not the first one that had thought about utilizing CP CO3 probes for IR imaging. Actually, there was a publication where these CP complexes were placed inside proteins, and this study compared the use of these complexes with the more, let's say, diffused use of cyano groups, so this nitrile portion here, which were previously used as IR probes in proteins. They both fall within the transparency windows of cells, around 2000, 2200. But the cyano group has an excision coefficient which is much, much lower than that of the complexes included in this other probe. So the rhenium tricarbonate complexes have a much, much stronger absorption than the cyano group. So this means that these complexes might have a higher sensitivity when you try to use them as a perspective imaging probes. One thing to show here is that I thought in the previous slides you've seen that the isolated complex can give 2 different peaks of stretching in asymmetric and symmetrical mode. Actually, when these complexes are found within an aqueous environment with several different components, the different peaks are kind of merging into a wider peak, which is kind of sort of washed out because of the different situations of this CO depending on the environment. So you basically observe a single broad band, which where both the asymmetrical and symmetrical stretching will merge. And in this study published in 2014, these authors have shown also that by increasing the dielectric constant of the medium, the frequency of the stretching kind of shifts downwards at lower energies. But this was not a big deal with us. We just needed a broad and strong peak that could be placed in the strong transparency window in the transparency window of cells. So this was a good support, good literature support in favor of utilizing these probes for visualizing portions in cells. But instead of placing these complexes in protein, we decided to link these complexes to biorelevant portions, for example, to sugar moieties that could be actively taken up by glycolytic cancer cells. And this was the original thought that we had, so that we could bind an IR probe like this by means of a proper linker to a sugar portion, so that the cancer cells overexpressing GLUT transporters that have an enhanced uptake of glucose could selectively accumulate these probes, and then by means of an infrared instrument, we could visualize the cells that take up these probes at a microscope. So how we realize this aim? Actually, had been working more than 20 years ago on the synthesis of functionalized cyclopentadienyl tricarbonyl complexes of rhenium and technetium. Our original project that was developed in the US together with John Katzenellenbogen was devoted to the synthesis actually of radioactive technetium complexes, and we used rhenium as a cold model to optimize the reaction conditions and then apply them to technetium, which is always radioactive. So actually, I utilized these technologies that we had initially developed, and that employs diazocyclopentadiene to actually synthesize CP rhenium tricarbonyl complexes that have a substituent, which is characterized by being a nucleophile. Actually, if you look at the structure of diazocyclopentadiene, it can be considered as a sort of bifunctional synthon, where you have a precursor of the aromatic anionic cyclopentadienyl ring here with an empty exocycle orbital, which can be exploited by an external nucleophile, whereas the negative charge of the aromatic CP ring can complex a rhenium tricarbonyl portion. So we had developed this reaction, and initially we used diazocyclopentadiene, which, by the way, is a highly unstable reagent, which can also be explosive, so quite dangerous to be to handle. It has to be used in a solution of diethyl ether, cannot be dried, so it has some problems also of storage. Meanwhile, we had developed a stable analog of this diazocyclopentadiene, which is based on the polymer-supported phosphazane shown here, whose aspect is shown in this picture. So it's a nice material, orange, characterized by these small spherical portions, which are basically the backbone of the polystyrol divinylbenzene polymer, where we have functionalized the external portion of the triphenylphosphine with diazocyclopentadiene, and we got this very stable reagent that we can use by just weighting it up and placing it in the reaction to produce the CP rhenium tricarbonyl complexes, where we have properly studied suitable nucleophiles, so that this nucleophile includes a linker that can be attached, to a sugar portion, for example, to glucose. This way, we were able to obtain quite very nice complexes, like this one is just an example, which can be fully characterized. I'm going to show you just a single example of a bidimensional nuclear magnetic resonance spectrum of this glucose conjugated complex. This is a heteronuclear single quantum coherence spectroscopy image of this complex in deuterated methanol, and actually all the protons are easily identifiable, and the correspondence to the carbon atoms is also pretty nice, and all the signals are widely separated and can be perfectly characterized. These complexes were also, of course, characterized by mass spec, where you can see this single peak of the molecular weight of the complex protonated. This is an electrospray ionization mass spectroscopy. You can see also the relative abundance of the isotopic isotopes of rhenium, where rhenium 187 is 100% the nature composition, and the rhenium 185 is about half, less than half of that. So the mass spec confirmed the production of the proper structure of the complexes that we obtained. But what was most important to us was to verify that this complex is conjugated to the sugar, of course, still preserve a very strong absorption in the cell infrared transparency window. And as you can see here, the isolated complex have a very nice and sharp absorption at 2020 for the symmetrical stretching and 1910 for the asymmetrical stretching. Okay. Now we expected that these two bands would merge in a cellular environment due to the surrounding of this CO ligands. So the material was made, the infrared characteristics were verified, and we decided to use them to visualize at the microscopic level cells. Fortunately, in our department, we had purchased this Agilent Carry 620 FTIR microscope, which is a sort of combination of an IR spectrometer with a microscope that can visualize cells at very low sites. So just to make this story short, we started with some very preliminary imaging experiments of pancreatic cancer cells, PDAC cells, in collaboration with Nicola Funel and Elisa Giovannetti. And we treated these cells with some of our compounds. These are just very preliminary images that we got. So cells treated with this complex 61 were first analyzed for the spectroscopic properties. So you can see the spectrum over here, where there is a broad band around the 2000 here. Actually, it's 2100 mostly focused in this portion of the spectrum. You can see on the left very strong absorption of the water molecules of the medium. You can see also a very strong absorption of the protein bands at 1600 and 1500 of the cells, and some of the carbohydrate here around 1000. So we decided to compare images taken from all these relevant portions of cells, and you can see that there is quite a strong correspondence when we focus the spectrum around the carbohydrate absorption bands. You can see a PDAC cell here, which is very easily to be visualized. When we change the frequency to the protein absorption, the cells are still visible, and we were pleased to find out that when we switch the frequency to the 2100 wave number, where usually cells do not absorb infrared radiation, actually the cells treated with this complex started to respond as well, just as much as the carbohydrate and protein frequencies. So this was the first proof that we had that we could visualize glycolytic cancer cells after treating them with an IR probe, which is glycoconjugated. This is a different image of the same treatment, where we were able to visualize in 3D the absorption, so that we can have a better quantification of the absorption levels of the cells in the space. And you can see that there is a very nice correspondence of the peaks of absorptions when you switch from carbohydrates, proteins, and the complex frequencies. Okay, so there is a spatial correspondence, and also the intensity is quite near between these three frequencies. And so we started this project quite recently, and we wanted to develop that further in the future to find out if we can use these probes to visualize cells first in vitro, and then we will see if this technology can be also extended in vivo. Now, going back to the glycolytic targets, as I mentioned, with the few minutes that I have left in my talk, I'd just like to briefly talk about the therapeutic agents that we have developed towards these targets. First, we have developed, as Elisa has shown in her previous presentation, some auxin derivatives that are able to block GLUT1. We have developed this compound by means of computer-aided drug design approach, and there are three pockets in the glucose transporters. Basically, our auxin derivatives bind in the inward open conformation of the glucose transporter on the intracellular pockets, by establishing quite strong interactions with the protein. And these compounds proved to block the uptake of a glucose-labeled fluorescent marker, the 2N-BDG, in lung cancer cells. As you can see, the treatment of the cells with some of our GLUT inhibitors reduce considerably the fluorescence of this analog of glucose in cells treated with it. And so this proved to block glucose uptake, but we still were not sure that the glucose transporter 1 was involved. So we started a collaboration with the Karin Lindqvist in Lund, in Sweden, where they had developed some artificial vesicles where they could insert glucose transporters in say, phospholipids that were properly synthesized and assembled by them. So this is not a real cell. Is a double-layer membrane with glucose transporters and an enzymatic system in the inner part of the vesicles that could highlight the uptake of glucose. So basically the concept was that the vesicles without the glucose transporter do not absorb glucose, so they're not they do not emit light. When you place glucose transporters in these vesicles, the glucose is taken up by the vesicles and you have the light on. And when you use the inhibitors, the concept is that you block the uptake of glucose and the vesicles is off again. Okay, so this is a very specific way to show if the interactions of the molecules are due to GLUT1 inhibition and not to other side mechanisms. Now, we tested some of our compounds in this assay, and actually 4 out of 5 compounds that we tried proved to efficiently reduce the glucose uptake in these vesicles. Karin used cytochalasin as a positive control, which is a toxin that very strongly blocks glucose uptake in cells by inhibiting GLUT1. So these compounds were also utilized to see if they were able to block proliferation in leukemia cells. And Karin has done this very nice work by selecting the proper cell lines where the THP1 cells and the KG1 cells were analyzed by fluorescent microscopy to see which of these cells had a higher GLUT1 expression. And you can see here that the THP1 leukemia cells have a very high GLUT1 expression, as also shown here by this Western blot, whereas the KG1 cells have a much lower expression of GLUT1. So we wanted to see if there is a differential response to our compounds in these 2 cell lines, and we used 2 representative GLUT1 inhibitors, PGL13 and PGL14, both alone and in combination with RSE, which is a classical anti-cancer drug used in these leukemia cells. And you can see here that the GLUT1 expressing cells, the purple THP1 cells, have a stronger response to the combination of PGL13 and RSE than the KG1 cells that do not express too much the GLUT1 receptor. So this is a kind of proof of the mechanism that if you want to obtain a good results in combination in combining the GLUT1 inhibition with other chemotherapeutic agents, you have to be sure that the cells have a high GLUT1 expression so that the response is more pronounced. And the same happened for compound PGL14, although to a lower extent. So PGL13 proved to be the best in combination with RC in these leukemia cells. Now, other therapeutic agents that we have developed are ADH inhibitors, and I'm really glad that Reshma is over there because she was one of the postdoc that also participated to this project in developing an hydroxyindole carboxylate that could inhibit ADH. We started our molecular design by analyzing the putative binding process of this compound inside the lactate dehydrogenase active site. And as Elisa has shown, this enzyme works by reducing pyruvate to lactate by utilizing a reduced cofactor, which is NADH. And we found that basically this compound, both NH11 and its glucoconjugated compound, occupy the active site of the enzyme, especially the portion that is usually occupied by the cofactor NADH, and to a lesser extent the pyruvate substrate. Now, the idea was that we could utilize the glycoconjugate of NH1 so that we could exploit both the enhanced uptake of glucose by cancer cells and the LDH inhibitor properties of the indole portion. Okay. So after that, we have verified that, for example, this compound could occupy also the portion that usually, as you can see here, hosts the cofactor NADH, and this is an X-ray image, and you can see that the portion of this compound also is placed where the NADH is placed in the X-ray, and these binding poses were somehow confirmed by enzyme kinetic studies that we made to 3 of our representative compounds. You can see here the KI numbers of competition versus the cofactor NADH and the pyruvate. Now, these compounds are, well, they do compete with both cofactor and substrate, and the maximum potency is reached by this carboxylate, free carboxylate derivative, but the glucose conjugate seems to be a much weaker inhibitor of LDH in this in vitro isolated enzyme assays. Nevertheless, if you look at the activity of these compounds in cancer cells, this is a panel of cancer cells, and you can see that actually the glycoconjugated compound is the most potent antiproliferative agent. Part of this incongruence was explained by the fact that, based on our rationale, this compound could be internalized much more than the other 2 derivatives by means of the active uptake of glucose portion by cancer cells. And you can see here in the bars that shows you the intracellular concentration of this compound, which are normalized on NH1, the first compound here. You can see that the glucoconjugated compound has an accumulation which is about 25 times higher than that of the original compound. So this could explain why in cells the glycoconjugated compound is more potent than the other 2s, in spite of the fact that on the isolated enzyme, this compound is not as potent as the other 2 compounds. Okay, so this is a graphic showing the reduction of the lactate production induced by these compounds. And again, the glucoconjugated compound is the most active inhibitor of lactate production in lung cancer cells. Elisa has already told you about the combination of some of our LDH inhibitors with gemcitabine in mesothelioma cells. So this is just to show you that when combined, this compound can have a pretty potent results in mesothelioma cells. I'm not talking about this because Marika has presented a poster at the conference about the combination of our LDH inhibitor in the mesocells, which reduce both the content of ATP, ADP-MP, and also of the cofactor in cancer cells. And the combination is made by mixing LDH inhibitors and GLUT1 inhibitors like EGL14. And I'm sure that Fritz is going to talk about that later on. So I'll switch to the next content, which is allows me to introduce a very recent study that we have started in collaboration with the group of Kin Tam in Macau, where he is now combining LDH inhibitors with PDK1 inhibitors developed in his lab, because they are at the crossing point of this glycolytic pathway, so that they could synergize in blocking the use of glucose in cancer cells. But here again, Fritz Peters, after me, is going to talk about this combination in detail. So I will leave the discussion to his talk. I just wanted to show you that these compounds were also combined in vivo, and we have a pretty strong antiproliferative effect in this xenograft model of mouse without significant toxicity on the mice body weight. Okay, so I guess I'll stop here, and I would like to thank all the people that contributed to all these works, especially Giulia Bononi and Sebastiano Di Pietro, who are here in Pisa, and they contributed to developing the compounds by molecular design and synthesis. I'm really grateful to Reshma Rani and Sarabindu Roy. They were both 2 very brilliant postdocs from India that visited my labs for a couple of years. each, and then Valeria Di Bussola and Carlotta Granchi for helping me in coordinating this project in Pisa, and the computational analysis were carried out by Giulio Poli and Tiziano Tuccinati. Then the biological assays were done by Nicola Funel here in Pisa, and of course Elisa Giovannetti and Fritz Peters in Amsterdam, Marika Franzek and Tom Smoleski by the Medical University of Dansk, King Tam and Yan Zhu in Macau, Paul Ergenrotter at Illinois, the research group of Karin Lindqvist in Lund, and Xiaozhuo Chen in Ohio for the GLUT1 experiments. And finally, we just got some funds from the European Union, the Next Generation EU projects that is funding essentially the precision medicine exploiting tumor glycolysis for both therapeutic and diagnostic purposes. I'm still grateful to the European Union for the 2 grants given to Sarabindu Roy and Reshma Rani for their Marie Curie postdocs in my lab. We got a grant for LDH inhibitors together with Paul Ergenrotter by the NIH in the US, and finally an internal project called Glinfrared by the University of Pisa is still supporting the development of the infrared probes. I should not forget my unintentional designer, the late Kazuhide Takahama, who designed the chairs, and still alive and kicking Mario Bellini, who designed the table, for their inspiration to my complexes, and all of you for your kind attention. Thank you very much.
Thank you, Filippo. I think in view of time, maybe just one question. If there's a question from the floor, please. Oh, yes.
Thank you very much for presenting the nice work. My question is that when you are glycosylated the molecules, it increasing the uptake into the cancer cell. Does it do the same in the normal cells?
Okay, that's a very good point. Actually, also normal cells, of course, use glucose for their physiological activities. The problem is that we have to set the proper concentration so that we could exploit the overexpression of glucose transporter in cancer cells. And this sometimes is kind of like, let's say, tens to hundreds times higher in cancer cell than in normal cells, which means that you could hope to have a selective accumulation of these molecules in cancer cells, just due to the fact that there are order of magnitudes of difference in glucose uptake. Basically, this is the same principle that is exploited by the FDG PET scan, as you can see. So also normal cells take up glucose, but not as much as cancer cells. So you can see that difference when you use FDG PET scan, and we hope we can see the same difference when we use our diagnostic and therapeutic probes, which are glycoconjugated. So it's a kind of, let's say, therapeutic window that is in favor of cancer cells. Thank you for your question.
Okay, thanks again, Filippo.
Thanks, Andy, and thank you very much for your attention.
Hi, Professor. Myself, Reshma, this side. Hi, how are
you?
you?
Hi, Reshma. Hi, very nice to see you again. Very well.
Good to see you after a long time.
Yeah.
Good to see you here, but next time we will meet again.
Okay. Yeah, I'm really sorry I cannot be there, but it's a real pleasure to see you all there. Thanks.
Yeah, and thank you for the appreciation you showed me the slide here. So I have a quick question for the IR spectroscopy here. The molecule are synthesized and you are using for the IR concept for the diagnosis.
Right.
Is that you showed that you did the NMR in MEOD, right? Methanol. You did the NMR in MEOD. So it's a quite soluble in methanol. It's soluble. There is no issue for the solubility. So that molecules are quite stable in stable also. Stable. Is there any issue for the stability?
Yeah, that's a good point. Yeah, actually, we have verified that these compounds could survive conditions that are utilized in vitro visualized in cellular environments. The molecule is stable in methanol, deuterated methanol. We use the deuterated methanol because, of course, that molecule was not soluble in chloroform, of course, but it's soluble in methanol, it's soluble in DMSO, it's soluble in mixture that you could use like aqueous solution to visualize cells. So, yeah, these compounds are soluble and stable in those media, and so there was no issue, no particular issue about that. Yeah.
And thank you very much for the wonderful talk and the bring the new concept for IR for in this tumor metabolism. Thank you.
Thank you very much.
Okay, we move on to the final presentation for this session, and it's a great pleasure to welcome Professor Fritz Peters to the stage. Fritz is an internationally reputed scientist in the field of pharmacology of anti-cancer drugs, pharmacokinetics, pharmacodynamics, pharmacogenomics. It's difficult with a long career, Fritz. It's difficult to know what exactly. You seem to have worked on all kinds of cancer drugs and all kinds of mechanisms over a long career. So, without going into too much detail, certainly you specialize in preclinical pharmacology of anti-cancer drugs and their translation into the clinic. I've known Fritz for many years through his extensive work through the EORTC PAM group, the European Organizations for Research and Treatment of Cancer Pharmacology and Molecular Mechanisms group, and Fritz has been secretary and chair of that group, and has done lots of other things as well. He's supervised more than 30 PhDs, more than 100 master students, more than 600 refereed research papers, H-index 96, so pretty remarkable. It's a great pleasure, Fritz. I'm glad you made it. I know the journey wasn't straightforward, but we welcome you to give your presentation. Thank you.
You can start, yeah. Yeah, this one, yeah. Yeah, Peters. Yeah. I would first like to mention that I'm pleased to be here. I had some difficulties to come, but thanks to Vinit, who helped me to solve all the problems, and my friend Professor Mayur, I'm here. And although I arrived late last night, 1-2 o'clock, but I can usually deal with a little bit of sleep. I'm very pleased that Elisa and Filippo have already given some of the background on LDH and GLUT. One, Elisa on the, I would say, the clinical background on patients, and Filippo on the medical or the medicinal chemistry background. And I will focus on how to use this type of information for developing combinations. Let's take a look. What is just this way? This is the outline of my presentation. But the pointer does not work, or whatever it works. Anyway, I will give you a little brief introduction on the history of targeting metabolism, because this is not a new novel idea. And related to that, I will also talk about a molecular correlation concept, which fits with what Elisa told about the imbalance. And then subsequently, I will show a number of combinations based on these principles. How do you overcome chemoresistance by targeting metabolism? There are a couple of different ways to do this. Use the difference between tumor cells and normal cells, and Filippo already mentions the high expression of GLUT, and also Elisa the high expression of LDH. And how can we do that and evaluate this? You can also try to correct the aberrant metabolism. I will give you some examples, and also bypass aberrant metabolism. A brief history going back to targeting metabolism. This started in the 40s by Sidney Farber and George Hitchings. Sidney Farber was a clinician in Boston, and Hitchings was at that time working at Wellcome. And they had, especially Hitchings, had the idea that we have to hit cells on where they are sensitive. So you should realize there were no cancer cells at that time. There was no DNA structure was not known. but he thought that when he would hit the metabolism of the cells, then the cells would die. And he realized that the cells would need folates. So there was a design of two compounds, aminopterin and methotrexate. Aminopterin was tested in patients, and it was the first time that complete remissions in leukemia were shown by aminopterin and later by methotrexate, which is an inhibitor of dihydrofolate reductase. This has led to a successful treatment of acute leukemia in children where 95% are cured, and to the development of the compound which Lisa already mentioned, pemetrexed, and also later pralatrexed. There are a lot of antimetabolites which are antimetabolites first already tells you it interferes with metabolism. And I'm not going to discuss all of them, but you see that all of them, also methotrexate, needs ATP for activation. 6-maltodextrin and so cytarabine, which already mentioned, and they all affect DNA synthesis. Deoxyconformation doesn't need activation, and I have been so fortunate to work on the development of both gemcitabine, pemetrexed, and more recently also this last compound, TAS102, which I helped to get registration, at least. The molecular correlation concept was introduced by George Weber, and at that time I was a PhD student and was really intrigued by his ideas. And what he actually postulated that the molecular imbalance in cancer was associated with neoplastic transformation. There's usually an increase in essential enzymes, and we already heard about that, GLUT1 and its transport, but also LDH. The degradation enzyme are often decreased, and that will lead to a change in metabolic patterns, and that supports the growth of tumor. The metabolic imbalance also is controlled by key enzymes, which can be considered as an Achilles heel of a cancer cell. And there's a lot of resemblance to the current hypothesis for the addiction of cancer cells to growth factors. A little bit more on the key enzymes. They usually are important in normal cells. They control the metabolic flux. They are one way. They are first or last in a pathway and have a tight feedback control and controlled by isosign patterns and also controlled by alternative splicing. They are often increased in cancer cells to sustain growth, and the gene is often mutated. And therefore, we consider this as an ideal target. Non-key enzymes actually are not considered as an ideal target, and because they're usually not limiting a metabolic flux. We have already seen a couple of pictures on this the glucose metabolism to pyruvate. And what is shown in this slide is different from what the previous slide is, that the essential enzymes in this pathway, the first one, the glucose to glucose-6-phosphate, fructose-6-phosphate to fructose-diphosphate, and PEP to pyruvate, they are all increased. They were indicated by the increased size of the arrows. What is important in this slide as well is that there is two side pathways, the pentose phosphate pathway, which is important in the synthesis of ribose 5-phosphate, and also both the oxidative pathway and non-oxidative pathway are increased. Why is this important? Ribose 5-phosphate is the precursor for phosphorylated pyrophosphate, PRPP, which is essential for the synthesis of nucleotides and of NAD+, which are both compounds which I'm going to discuss further. As I mentioned, PRPP is a substrate for pure de novo nucleotide synthesis, pyrimidines, several enzymes, the OPRT and alterate phosphorybosol transferase, which were mentioned in the previous slides, but also NAD+. As mentioned, NAD+ is important for glycolytic and mitochondrial enzymes, but also for synthesis of indirectly of the deoxynucleotides. They are all increased in cancer cells because cancer cells have an increased demand for these deoxynucleotides for the synthesis of DNA. On the other hand, Lisa already mentioned a few points about hypoxia, and usually nucleotides are decreased on hypoxia, and the cells are, which limits their growth. So I mentioned the deoxynucleotides are increased, and this was one of the first papers, as far as I know of, that was shown that these deoxynucleotides were increased. What is shown here from the right, from the left to the right, is the difference in deoxynucleotide triphosphate pools of deoxy-CTP, ATP, deoxy-CTP, and D-ATP in tumors which have an increasing growth rate. So the fast-growing tumors have a much higher deoxynucleotide pools than the normal cells and the low non-growing, the slowly growing cells. As mentioned, this is essential for having the deoxynucleotide triphosphate pools for DNA synthesis. In hypoxic 3D cultures, we have measured the nucleotide content. What you see here is ADP, UTP, CTP, ATP, and GTP, and the closed bars are the monolayers, and the 3D hypoxic cultures are the stripe bars. And you see each of these nucleotides is much lower, which is limiting, for instance, for the activation of the compounds which I just mentioned, gemcitabine, methotrexate, and so on. They need ATP to be activated. So these cells are subsequently less sensitive to these compounds. This is gemcitabine, which was already mentioned. It needs ATP to be activated. And on the left side, you see the monolayers, which are quite sensitive in the nanomolar range to gemcitabine. And the 3D cultures, they do not really reach cell kill. You see that almost all the cell lines, they are still above the 50% growth, which is not sufficient to kill the tumor cells. They are not really killed. Actually, they're still growing at 50% of their rate. So that is not what you want when you want to treat the tumor cells. How to improve this? That's what I'm going to talk about in the next slides. As I mentioned, ATP is essential for activation of gemcitabine. And on this slide, in the close bar, you see activation of gemcitabine to the active metabolite difluorodeoxy-CTP, which is higher than in the 3D cultures, which is in the other bars, the non-closed bar. So this is really reflected in a poor metabolism of gemcitabine in the 3D cultures. On the other hand, that's quite interesting. In these cultures, you see a change in ATP when you treat the cells with gemcitabine. And that was quite surprising because what we saw is that gemcitabine increased the concentration of ATP. And not only of ATP, but also GTP, which is a little bit surprising. And that's why I don't like the growth inhibition assays, which are based on ATP, because they could believe that ATP is always going down in the tumor cells. And you see, it's not happening depending on the drug. So it cannot be used as a universal assay. On the other hand, we see that CTP is going down. And that can be explained because gemcitabine inhibits the conversion of UTP to CTP. So UTP is going up and CTP is going down. So how can we use this information to increase the sensitivity to gemcitabine and some antifolates? As Elisa already mentioned, to some extent, she introduced antifolates. Just go back to how is gemcitabine metabolized. Gemcitabine is difluorodeoxycytidine, abbreviated to DFDC. It has been to be taken up by a facilitated diffusion that doesn't use ATP for this. And finally, it will be activated to DFDCTP, which can be incorporated into DNA, but also to RNA, which was a surprise. We were the first to show this, and it was the first time for the deoxynucleotide that was really found. What DFDCTP does, it is, and also DFDCDP, it inhibits ribonucleotide reductase, which is essential for the synthesis of deoxynucleotide triphosphate. So DNTPs, including DCTP, is going down, which leads to an increase in incorporation of DFDCTP in DNA. On the other hand, DFDCTP actually activates deoxycytidine kinase in contrast to deoxy-CTP, so you can have an increased metabolism. So if you modify the ATP concentration some way, you can activate the synthesis of DFCTP more. You should also realize that ATP is essential for ribonucleotide reductase as a not as a cofactor, but as a catalytic factor. Elisa showed this already briefly. Gemcitabine and this LDH inhibitor, which was synthesized by Filippo, was synergistic with gemcitabine, which is shown on the right slide by the combination index, and the combination index lower than 0.9 is indicating synergism. And you can see that in the two hypoxic cells, both hypoxic LPC 006 and the PANC1 06 was also introduced earlier. Especially in the hypoxic cells, we see a very pronounced synergism. Also the normoxic cells, but hypoxia is really most prominent. So that's really because of the ATP, which is going up. These are data which were found in monolayer cells, but it was also introduced into, I'm sorry, also investigated in spheroids, which are three-dimensional cells which have a hypoxic core. And what you see here again is that LPC and these two inhibitors reduce the size of the spheroid, which really means that the spheroid is not growing, and not only not growing anymore, but is also decreased in size. And decrease in size means that the cells are killed. And that's what we want with tumor cells. We want them to be killed. How can we explain this? There's actually an additional factor which was changed in these cells, and there are 2 points you see in the Western blot. That E2F1 is increased, and E2F1 is a modulator of the oxycytidine kinase. On the y-axis, you see the expression of the oxycytidine kinase, and what you can see here is that the oxycytidine kinase is also increased by gemcitabine, which was found earlier years as well. There's also some control on the phosphorylation of the oxycytidine kinase. But the two LDH inhibitors also increase the activity of the oxycytidine kinase, and together even more. So you get a potentiation of each other. So this the combination of the two drugs increases the oxycytidine kinase, which leads to an increased formation of the DFTCTP. This is associated with an increased formation of DFTCTP, because I just mentioned that the oxycytidine kinase is increased. And you see that on hypoxia, the black bar, the concentration of the gemcitabine deoxynucleotides is low, which is higher, much lower than under normoxia. But you can see that the hypoxic concentration of DFTCTP is increased by combination with the LDH inhibitors. So this is the final proof that indeed with the modulation you can increase the synthesis of these deoxynucleotides. This also leads to an increase in apoptosis. Gemcitabine by itself causes apoptosis. The LDH inhibitors actually not, but the combination of gemcitabine and the H1 they really increase the apoptosis, so the cells are really going to be killed. Previously, this NH1 GLUT2 inhibitor was also called PFLY, and that also shows an synergy with gemcitabine, which is also shown here under the hypoxic conditions. Again, at the lower part, the combination index is 0.2, which is really very good. This was also found under in the spheroids, and here again you see that the spheroid aggregation here is the default increase or decrease in the aggregation is increased by the combination. Finally, this was also found in the vivo model system, which is we actually see that gemcitabine and the LDH inhibitor have some anti-tumor effect, but the combination is really much better, and it leads to a decrease in the size of the tumor. So the really tumor cells are being killed, which is what we aim to find. With another compound, pemetrexed, we also have been looking at the combination of which you see that here in this number of mesothelioma cells, the pemetrexed is active only at very high concentrations. Should realize that under normox conditions, the concentration is in the nanomolar range, but here we need micromolar range. But when we combine pemetrexed with the LDH inhibitors, you see here from the left to the right, the LDH inhibitors, pemetrexed, but the combination is again leading to a larger fold decrease in the tumor size. So that is what we would like to see. And here what you see it a little bit differently. You see that the bars in the combination are going down, and that's actually what we aim to see with this type of combinations. These are combinations, I discussed a couple of combinations with normal drugs which are being used in treatment of cancer. But what about combinations of compounds which target glycolytic pathway or the glucose transport simultaneously? This slide was already shown shortly by Filippo, and what it shows here is that the glucose is uptake is mediated by GLUT1, which can be inhibited by this PGL13 and 14, and LDA and NEH1 is the inhibitor of LDH. And my PhD student, Elisa joint PhD student from Gdansk and Amsterdam, Marika has tested these cells for mesothelioma cells for LDH inhibitor. And what you can see here is, and the GLUT inhibitor, they are quite sensitive in the micromolar range, and especially the PGL13, the GLUT inhibitor, is quite active. When we tested these cells under hypoxia, we see that the closed markers are on normoxia, the open markers are on hypoxia, that the LDH inhibitor has more activity under hypoxic conditions. And when we combine this with this case, these two inhibitors, the LDH inhibitor and the GLUT inhibitor, you see on the right side the combination index again, and you see that in the meso cell line 2052, that there's a very nice synergism. In the other cell line, there's also synergism, but not so pronounced as in the meso. So showing that combination of the transporter and the LDH inhibitor is synergistic. This was also seen in the nucleotide compounds. You see that the PGL1, the GLUT inhibitor, hardly affects nucleotides, ATP, and AMP, and the LDH inhibitor to a little bit. But when you combine it, you really see that the ATP is going down, especially with the PGL13 and the NH2. In the right part, you see the effect on the NAD, and we see a similar effect that the combination really reduces the concentration of NAD plus and NAD. In a normal cell, which was a question in the previous talk, what is happening in normal cells? This is a vascular endothelial cell, and we see that the similar combination, even when using very high concentrations, does not really affect the concentration. We see a little bit of the change in the energy charge when we look at ATP and ATP, but again, we have to use really, really high concentrations for that. So that is a selective concentration. Going back to this, we also have this compound NH-GLUT1 with the glucose part, and the guys and the lady in Macau synthesized the compound which would inhibit PDK1, and that's would be also inhibit the enzyme which inhibits PDH, so that the mitochondrial nucleotide energy source would be increased. That was done by this compound was made by putting this in the active site of the enzyme. I'm not going to talk about this because I'm getting at the end of my time, I believe. But what has been done by our Macau colleagues is that they combined the two compounds, the LDH inhibitor and the PDK inhibitors. And what you see here is again the combination index on the left side of the H9070 non-small cell lung cancer cells was always below 0.4 at a high part of the fraction effect, and that's what we consider as important. When we look at the number of cell lines which they have been testing, they have been tested 6 cell lines, and only the H228 is not synergistic, and the other cell lines are also very nice synergism between these two compounds. They did some mechanistic analysis, and what you can see here again, the apoptosis was increased on the left side by the combination, and this was also associated with a decrease of BCL2, which BCL2 is an anti-apoptotic modulators, and that was decreased, but the PARP, for instance, was increased, which is also a marker for apoptosis. What we also see is that the ROS level was increased in the combination, which is actually good for killing cancer cells. The other things which have been investigated by them is looking at the phosphorylation of PDH, which was decreased by compound 64, as would be expected, and was also found in the combination. The other good point was a decrease in RAS and the phosphor-ERK pathway, which was especially in the combination decreased. Also, the phosphor-AKT pathway was decreased, as what can be seen in decrease of p-AKT, and the downstream pathway for mTOR was also increased. So there's a number of changes in cells which are really being affected by this combination. This also led to a normalization of the mitochondrial respiration in these cells. We have done this also for this LDH and GLUT inhibitor, but I prefer to show this one. What you see, the lowest part is in the middle. At around 40 minutes, you see the mitochondrial respiration. The lowest is the control, and the highest is the combination. And that's what you expect in a normal cell. And what you can conclude from this is that the mitochondrial respiration is normalized by the combination of these two compounds. Finally, as Philippe already mentioned shortly, the combination was also synergistic in vivo. You see this on the left side, you see the time course of the size of the tumor, and the combination was really controlling the tumor growth, which can be seen on the right side by the decrease in tumor weight measured at the last time point. So what can we conclude from this? Cancer cells have an altered energy metabolism, both glycolytic and mitochondria. So it's not only the Warburg effect, which is focusing on the glycolytic pathway, but also mitochondria are changed. Hypoxia will decrease nucleotides, which can be to some extent normalized by the LDH inhibition. So we get some sort of normalization. And inhibition of LDH will increase the effect of gemcitabine in pancreatic cancer cells and mesothelioma. And one of the explanations for this is the increased concentration of ATP, but also the activating enzymes are increased, leading to an increase of the active metabolites. Also, an antifolate can be increased, and you should realize, I didn't show this, pemetrexed has to be polyglutaminated to be active, and for this pathway you need ATP. The inhibition of the glycolytic pathways also increase the growth inhibition in vivo, and the nucleotides and the NAD and NADH are affected as well. The perspective is that the molecular correlation concept, which was developed in the 70s, can still be applied, and that's quite amazing from my point of view. We can target metabolism that is really not. We can do this with a novel knowledge that we can do this more but more effective than we did in the past. And I believe we should select patients on their metabolomic alterations to increase selective kill of tumor cells, but also prevented effect on normal cells. I want to thank in Amsterdam, of course, Elisa and some PhD students, Amir and Mina. They're back to Iran, and they do very well. I put myself both in Amsterdam and Gdansk. Of course, some of the work was not be possible without Marika and Tom Smolensky, for which they got a grant from the Polish grant organization. Elisa is also in Pisa, and Filippo and Carlotta provided the compounds, and Kintam and Jiangsu in Macau provided the S. In the previous slides, you saw that I was not present. That was because I made this picture, and then I moved to the group, and I'm also on the picture as well. And a number of the PhD students and so on, which are depicted on this slide, and I'm very grateful for their support. And I thank you for your attention.
Thank you very much, Fritz. Okay, time for one question before we break for lunch.
I believe the audience needs some glucose and other energy.
I think so. Before we have to do the presentation, so stay there, Fritz. We'll do a presentation, and then we'll break for lunch.
Thank you, Professor Peters, for such a wonderful talk. Now I invite session chair, Dr. Andrew Westwell and Dr. Reshma to please present memento to Professor Peters.
Yeah, I will answer. Yeah, I will answer. So there is a interactive session again, so you can interact while you are having the lunch. Right now, lunch is the same place, that is Wall Street. That yesterday we had it. The same place we arranged the lunch. Thank you. Right? And we will assemble here at 3:30. Probably within, because poster session is also going on. So
Yeah.
it will take 5 minutes more. So,
Okay.
So after 5 minutes, we'll start.
Yeah, yeah, I'll stay here. No, no worry.
Session, and those who are in poster session, they can come in the time. Okay, so let's start. So now I will invite our session chairs, Professor Flavio Rizzolio. He's professor in Ca' Foscari. It's a university in Venice, and he's also professor in National Cancer Institute, Aviano, Italy. And he's also my kind of collaborator, mentor, and postdoc supervisor. So, okay. And another session chair is Professor Manoj Garg. He is professor and assistant director of our department, and he works in pancreatic and blood cancers. So, over to you, session chairs, please.
Yes. Thank you very much, Dr. Vineet. So, the speaker of this session is Dr. Johanna Cheche. She is a well-known figure in the field of cellular metabolism and immune responses in human cancer. She got various prestigious awards from France Lymphoma Espoir. She also received Helen Stark Prize from ARC Foundation. She has published more than 100 manuscripts in well-reputed journal in the field. Her H index is 27 and she has a 3 patent. Now I invite her to give a talk on lymphoma and cellular metabolism and therapeutic. You are welcome, please.
Thank you very much. So I'm very happy to be present here in India and to share the work I do on diffuse large B-cell lymphoma and cell metabolism. And for the rest of my talk, I will talk about DLBCL for diffuse large B-cell lymphoma. Okay, here we are. Thank you. So diffuse large B-cell lymphoma are the most common type of non-Hodgkin B-cell lymphoma. They are high-grade tumors composed of malignant mature B cells that proliferate in the germinal center of secondary lymphoid organs such as the lymph node and the spleen. And DLBCL are highly heterogeneous at the molecular level because they may arise from different cell origin. Based on specific gene signature, they can either be classified as germinal center B-cell DLBCL, meaning that they express genes like that found in normal B-cell in the germinal center, or they can resemble activated B-cells. And this molecular heterogeneity has been shown to influence tumor sensitivity to first-line therapy, with ABC-DLBCL being associated with a bad outcome. So what first-line therapy for this disease? It consists in an immunotherapy targeting CD20 expressed on the cell surface of pre-B, immature and mature B cells, thus leading to cell death by different mechanisms. An anti-CD20 named rituximab is administered in combination with a polychemotherapy named CHOP. And this combination significantly improved the overall survival of patients as compared to previous standard of care that was CHOP treatment alone. Nevertheless, 40% of patients still experience therapeutic failure. And nowadays in 2023, patients with low comorbidity factor will either receive high-dose chemotherapy followed by autologous stem cell transplantation, or they will benefit from chimeric antigen receptor T-cell therapy that has been recently approved in the clinic in Europe and in the US to treat patients with DLBCL. Following CAR T-cell infusion, however, about half of the patients still present a disease progression, and there is a need here to improve chemotherapeutic strategy for bridging and lymphodepletion as they may influence CAR T-cell expansion and efficacy. There is also another type of tumor heterogeneity that has been first discovered in DLBCL, is the metabolic heterogeneity. So by genomic clustering, the lab of Margaret Sheep identified two DLBCL subtypes, the B-cell receptor proliferation subtype that is characterized by the upregulation of genes encoding B-cell receptor signaling component and also cell cycle regulatory genes, and that was later being shown to rely on glycolysis, and the oxidative phosphorylation cluster that is enriched in genes involved in electron transport chain complexes. Importantly, there is no link between the molecular and the metabolic heterogeneity. That is why we wonder what are the clinical consequences of DLBCL metabolic heterogeneity in the context of patients that are treated with R-CHOP. So to answer this question, we perform an unbiased bioinformatic analysis on available transcriptomic data, and we identified about 500 genes capable of predicting patient outcome upon R-CHOP treatment. Functional analysis revealed oxidative metabolism on the top of the list to predict a bad outcome of patient. And when among those genes, we analyze metabolic enzyme deeper. And interestingly, we found only one gene encoding for a glycolytic enzyme, the GAPDH, the glyceraldehyde 3-phosphoglycerate, sorry. And it seems that GAPDH has moved in my slide. It's still on the cytosol, but it has moved. So I'm sorry for that. So GAPDH catalyzed the downstream reaction that convert glyceraldehyde 3-phosphate and 3-phosphoglycerate, and this contribute to NADH recycling, a step that is essential for the cells to maintain ATP production from glycolysis. Interestingly, in our context, GAPDH high expression in DLBCL is associated with a good prognosis. So to confirm those results, we set up an automated immunohistochemical staining of GAPDH and a scoring to quantify its protein expression in newly diagnosed DLBCL. And this allows us to subgroup DLBCL according to GAPDH low or GAPDH high levels, first in a training cohort of 43 patients, and later in a larger validation cohort. And according to the clinical data, we could indeed confirm that a low level of GAPDH are associated with a poor outcome of patient upon R-CHOP treatment, and GAPDH high DLBCL are associated with a better outcome. So to make a long story short, we also demonstrated that GAPDH biomarker is independent of bad prognosis factor already used in the clinic, such as the International Prognosis Index, which take into consideration the age, the number of extranodal sites, but also the LDH level in the serum, and also the cell of origin, MYC, BCL2, BCL6, or P53 expression, as well as translocation, single MYC translocation, double hit MYC BCL2, or triple hit MYC BCL2, BCL6. So identification of a novel biomarker of R-CHOP response is only of interest if we can propose new therapeutic options to patients that are R-CHOP refractory relapsed patients, here the patient that suffer from DLBCL GAPDH low. So we wondered whether we can identify metabolic vulnerabilities in those DLBCL. So for that matter, we went back to a more fundamental research, and we used the immunic mice model in which MYC is overexpressed in B-cell lineage under the control of the immunoglobulin mu enhancer. Transgenic mice develop spontaneous B-cell lymphoma with features of human non-Hodgkin B-cell lymphoma, such as the molecular and metabolic diversity. While we observe distinct expression level of GAPDH in lymphoma collected from different transgenic mice, we were unable to see changes in the expression of other glycolytic enzyme analyzed. And now we observe that immunic GAPDH low cells secrete less lactate and produce less glycolytic ATP, while total ATP levels are conserved, but they consume more oxygen, and consistent with an oxidative metabolism, they are sensitive to electron transport chain complex 1 inhibitor fenformin. Moreover, immunic GAPDH low cells increase glutamine transport rate and glutaminolysis, as shown by the level of glutamate and alpha ketoglutarate, and this is to favor mitochondrial respiration and ATP coming from mitochondria. In line with this, the immunic GAPDH low cells are also characterized by a high mTORC1 activity that is also implicated in mitochondrial respiration, as we could observe a decrease in basal oxygen consumption when we treated immunic GAPDH low cells with rapamycin. GAPDH overexpression in immunic GAPDH low cells reduces mTORC1 activity, as you can see by the reduced expression of phospho-P70 cis kinase, and induces a metabolic shift toward glycolysis. So really, GAPDH expression level influences the metabolism of the B-cell lymphoma, immunic B-cell lymphomas. So we wonder next, which chemotherapeutic agent ready to use, that's an important point, might collapse the metabolism of the LBCL GAPDH low. And unfortunately, targeting metabolism in the clinic, as we heard those last days, has very few successes in the clinic, but there is one drug clinically approved that seems to be of high interest in our context, and is the L-asparaginase. So the anti-cancer property of L-asparaginase was first discovered in lymphoma model in vivo, and it's later in 1997 that it has been clinically approved and used in the routine to treat patients with acute lymphoblastic leukemia, because in most of the cases, those leukemia are asparagine oxotrophic, and we'll come back to that later. So what asparaginase does, it induces the depletion of asparagine in the bloodstream, thus leading to cell death of asparagine oxotrophic cancer cells, but it also holds a glutaminase activity, leading to the depletion of circulating asparagine. However, it's of course evident that the affinity for glutamine is much lower than for asparagine. Nevertheless, by preventing glutamine availability in the media and glutamine utilization by the tumor cells, the asparaginase was shown to inhibit mTORC1 activity in glutamine-addicted cancer cells. So we thought it would be a good candidate to test in the context of B-cell lymphoma expressing low levels of GAPDH. But we first uncovered the respective contribution of asparaginase activity in our model, and for that we have used the wild-type form asparaginase that depletes asparagine in media, but also glutamine over time, as you can see at high concentration, 0.3 unit per ml. And we compared that to a mutant form of asparaginase, the Q59L, that has lost its glutaminase activity. And in that way, we could show that indeed, when we reach both the asparaginase and glutaminase activity, is where you see we can improve cancer cell death as compared to results obtained when only the asparaginase activity is reached with the wild type form at low concentration or with the mutant forms. We also titrate asparaginase in vivo to know more about the kinetic of asparagine and glutamine depletion in the plasma. So here what you can see is wild type animals that have been injected once with 500 units per kilo asparaginase. We could observe a drop, very rapid drop of asparagine in the plasma that remains under the limit of detection from 3 hours and up to 48 hours without no significant changes in glutamine concentration in the plasma. But now we injected once 5 times more asparaginase. You have the same kinetic of asparagine depletion in the plasma. However, you start to see a rapid and transient decrease in plasma glutamine that is back to normal levels 12 hours after the injection. And you can appreciate the mirror effect on aspartate and glutamate as asparaginase catalyze the hydrolysis of asparagine into aspartate and glutamine into glutamate. So then we also show that we need both activity to reach an optimal outcome for mice bearing immunic lymphoma, because you can see that the survival of the mice is improved when we use asparaginase at 2,500 units per kilo. We also verify that the asparaginase treatment has no impact on mice body weight. And it's when we challenge glutamine concentration in the plasma that we could indeed observe at high asparaginase concentration inhibition of mTORC1 activity in lymphomas in vivo. So now, if we analyze what's happened with in mice in vivo with mice bearing immunic GAPDH high cells or immunic GAPDH low cells that have two distinct metabolism, as I explained before, you can see that in mice bearing immunic GAPDH high cells, asparaginase has no impact on the survival of the mice, in contrast to the results and the improvement of overall survival we have in mice bearing immunic GAPDH low cells. And by overexpressing GAPDH, we could reduce tumor sensitivity to asparaginase treatment. So just to summarize this part, we have shown that the LBCL GAPDH low are associated with a poor outcome upon R-CHOP treatment. B-cell lymphoma with GAPDH low levels use glutaminolysis, mTORC1, and OXPHOS metabolic axis. They are also sensitive to L-asparaginase in vitro and in vivo. And then we wonder if we can translate our findings into the clinic. So we had the chance to meet a fantastic clinician, Professor Catherine Thieblemont, head of the hemato-oncology department in Hôpital Saint-Louis in Paris. She designed a compassionate protocol to interfere with tumor metabolism in MYC-positive DLBCL that are refractory to all anti-CD20-based therapy. And at that time, CAR T-cell therapy was not yet available for that disease, and patients were left with no other therapeutic options. So here is patient 1 with a cervical bulky mass 1 day before he received asparaginase every 2 days during 13 days and temsirolimus mTOR inhibitor on day 1 and day 7. 1 week after the beginning of the treatment, he had a spectacular reduction of his tumor mass. And then to limit the toxicity that is linked to long-term asparaginase treatment, the clinician decided to maintain the patient with metformin for 14 days. And here is what the complete response we could observe after 1 cycle of treatment, because his PET scan was negative. This patient had a normal life for 4 months and then died upon local and central nervous system relapse of the lymphoma. This is not a case report. 3 other patients with same inclusion criteria were treated, received this anti-metabolic strategy. 2 patients completed 4 cycles of treatment. Unfortunately, 1 patient presented adverse events very quickly during the process, and we had to discontinue treatment. 3 patients out of 4 were in complete response, and the median duration of the response was 6 months. Interestingly, retrospective analysis of GAPDH in their biopsies at diagnosis revealed a GAPDH low status of their DLBCL, consistent with our results. So just to summarize this part, we have established the proof of concept that patients with MYC positive DLBCL GAPDH low that are R-sharp poor responders, but they can show complete response to disruptors of their metabolism. So as I said before, L-asparaginase is highly efficient in the context of acute lymphoblastic leukemia because they lack this enzyme, the asparagine synthetase, which catalyzes de novo asparagine synthesis, and they lack this enzyme due to hypermethylation of high-sense promoter in 70% of the cases. So they are asparaginoxotrophic, and then they are asparaginase in that context is really efficient because they have no way to synthesize or to uptake asparagine, and they die from that. But so considering these results on ILL, it was long been considered that SNS is a marker of asparaginase response. So now with the renewed interest of asparaginase in other hematological cancer and solid cancer that all express heterogeneous level of SNS, we decided to clarify whether SNS can be used as a biomarker of tumor sensitivity to asparaginase treatment. So here on this slide, you can appreciate the heterogeneous expression of SNS protein in our immunic lymphomas, from SNS null due to hypermethylation of SNS promoter to low and high levels. And our model is as valid as a solid cancer cell line in which SNS was shown to be rate limiting for tumorigenesis. So we've verified that, and we propose to aspartate activity using stable isotope tracing. So briefly, aspartate plus 4, labeled on 4 carbon, comes from the oxidative metabolism of glutamine, and it can be converted into asparagine plus 4 through the reaction catalyzed by SNS, or into N-carbamyl aspartate plus 4 from the reaction catalyzed by CAD, or it can be converted into argininosuccinate plus 4 through the reaction catalyzed by the urea cycle enzyme ASS1, argininosuccinate synthase. So we decided to analyze the isotopologic distribution of those metabolites to really know more about SNS activity in our model. So when cells are cultivated in the presence of supraphysiological concentration of asparagine, aspartate plus 4 can generate N-carbamyl aspartate plus 4 and argininosuccinate plus 4 in immunic cells lacking SNS or expressing high level of SNS. But we didn't detect asparagine plus 4 regardless of SNS expression. So this suggests that SNS is inactive in B-cell lymphoma, regardless of basal SNS expression. However, when cells, and specifically SNS high cells, are cultivated in asparagine-free media, glutamine indeed contributes to de novo asparagine biosynthesis, as shown by about 40% of asparagine plus 4 detectable. So it is important that the composition of commercially available media greatly differs in asparagine concentration. And this is important to consider not only because cells that are grown in RPMI are unlikely to synthesize asparagine de novo, while cells cultivated in DMEM will rely on ISNS level to synthesize asparagine, but also it has consequences on the global metabolism of the cells. So now, is de novo asparagine synthesis active in physiological and pathological conditions? So as you can see here, the plasma concentration of asparagine in immunocompetent mice is about 90 micromolar, and we mimic this concentration in vitro, and we could only detect less than 3% of asparagine plus 4 coming from oxidative metabolism of glutamine in physiological conditions, suggesting that glutamine minimally contributes to de novo asparagine biosynthesis in physiological conditions, even in cells expressing high level of ISNS. We also perform 13C carbon label glutamine tracing in vivo to determine the extent of asparagine biosynthesis in lymphoma expressing high level of ISNS, but also in the pancreas of wild-type animals, as it is the organ expressing the highest level of ISNS in mice and in humans. So in the pancreas, what you can see here is a detection of asparagine plus 4 and asparagine plus 3 in red, coming from the oxidative and reductive metabolism of glutamine. But in lymphoma expressing high level of ISNS, we were unable to detect any traces of carbon labeled asparagine, while indeed metabolism of glutamine indeed occur because we could detect substantial levels of aspartate plus 4 and argininosuccinate plus 4 in the lymphomas. So it's clear that there is no correlation between basal ISNS expression and its activity, thus questioning the relevance of ISNS expression as a marker of tumor sensitivity to asparaginase treatment, especially in lymphoma that express different level of ISNS. So to summarize this part, we have shown that supraphysiological and physiological concentration of extracellular asparagine prevents its de novo biosynthesis by ISNS. Basal ISNS protein expression in chemo-naive B-cell lymphoma does not correlate to its activity under physiopathological condition. And asparagine concentration in commercially available media matters, so please pay attention to that. And I would like to thank the team of Jean-Erland Ricci, head of the laboratory, as well as all team members. And especially I would like to thank Professor Catherine Thieblemont, but also Ivan Nemanazi, in charge of the metabolomic platform in Hôpital Necker, and all our collaborators and facility, as well as our founders. And I thank you very much for your attention.
Thank you very much for wonderful talk. May I request Dr. Flavio Rizzelio to take over the question and the interaction part, and I request IT team to put all the online participant on the screen, please. Thank you.
Okay, thank you. So, there are questions? I have one question, if I can. Okay.
Yeah, please go ahead.
I see one guy. Okay, please.
Yes, please.
Okay, so just for Joanna, one question maybe I miss online. When you start to talk and treat about asparaginase on your animal model, I was wondering about toxicity, and then this happened in the clinical trials, So
Sorry, can you repeat because I couldn't hear very well?
can you hear me?
Yeah, better. Thank you.
Okay, Joanna. No, just when you start to treat your animal model with asparaginase, I was wondering about toxicity, and this is what maybe you see in the clinical trial that this treatment can have some toxicity side effects.
Yeah.
And my question is, since you show some relation with the mTOR pathway, are you planning to even to use some other inhibitors of this pathway to improve the chemotherapy treatment?
So concerning mTOR pathway, we have just looked at rapamycin. We analyzed it with rapamycin or in the clinic with temsirolimus. We didn't went deeper into that because it has mainly cytostatic effect on B-cell lymphoma, at least in our immunic models. So we didn't go deeper. And concerning toxicity due to asparaginase treatment in vivo, we didn't, even at high dose, we didn't observe any toxicity, but it's true in human it has some, especially in adults, while it's very well tolerated in pediatric cancer and pediatric leukemia. Toxicities are about thrombosis, coagulation troubles, hepatotoxicity. In mice, we didn't see any problems. And we even, because one also problem of asparaginase treatment, because it's bacterial enzyme, it can induce an immune response and the production of anti-asparaginase antibody, neutralizing the enzyme. We even, at the end of each in vivo experiment, did the last bolus of treatment and analyze a plasma composition of the plasma in terms of amino acids. And we always have the efficacy. So there is even no immune response after long-term treatment in our immunic model. I don't know if I replied correctly.
Thank you very much.
Dr. presentation. Jonah, actually Rizzolio already asked about the animal models. So I am asking about the human sample. Yes. So you have shown one patient is treated with the L-asparagines followed by the metformin after 14 days, right? So that patient is a GAPDH low or high? That is not shown in your slide, I guess. Maybe
so the compassionate protocol was done in blind without knowing GAPDH levels. It was based on criteria such as MYC positive, refractory to all on immunochemotherapy. But retrospectively, we could indeed see low level of GAPDH in their initial biopsies at diagnosis. Yes, it's true.
Okay, it's a low, right? And
yes.
If it is a follow-up patient, so what is your expectation? So after metformin treatment?
Yeah,
when the tumor is already spectacularly reduced.
Is reduced. Yes. So the clinician did cycle of treatment as long as she can, and as the patient can bear. So actually there was a complete response for 3 out of 4. We have no idea whether metformin was efficient in that clinical trial. We think most of the benefit comes from L-asparaginase, as we can in 1 week see the benefit, and what's given at the beginning is mainly L-asparaginase. But we have no, and in our immunic model is the only drug that leads to cytotoxic effect.
Right.
So, but unfortunately in adults, we cannot maintain asparaginase more than 2 weeks.
Yes.
So to continue inhibiting mitochondrial metabolism, because those lymphoma rely on mitochondrial metabolism to produce ATP, she decided to try metformin for a while. And then she repeated cycles of treatment as long as the patient can bear.
Yeah, we know there is a lot of question mark about metformin still. Thank you. Thank you.
Any other question from participant? So I have a last question. As we know that CD20 antibody is the kind of a standard treatment. Do you think the cytoplasmic level of GAPDH or the mRNA level of the GAPDH affect the CD20 treatment, or it's vice versa?
That's a very good point. And yeah, we have shown that DLBCL GAPDH high are more sensitive to anti-CD20. And it's true that indeed when we manipulate GAPDH, we overexpress, we do see more CD20 mRNA and protein expressed on the cell surface. That's why they are more susceptible to anti-CD20. And when we downregulate by short terpene RNA, we downregulate GAPDH, we see the opposite. Yeah, it's true. And now we are analyzing the mechanism of action, how GAPDH regulates CD20 expression. We don't know yet, but we work on that. Yeah. Yes.
Thank you so much.
Thank you.
So, okay, thank you, Dr. Flavio, for accepting our invitation to chair the session and the speaker. So now I will request Professor Lowry and Professor Prakash to felicitate, and Professor Manoj to felicitate our speaker. Thank you.
Dr. Jonah, please come on the stage. Yeah, please.
Professor. Okay, so now will have coffee break, and after that we will continue with the oral poster session. So please, let's move for the coffee, and after that we will continue with the oral poster session. Thank you.
So, good afternoon, everyone. So, myself, Priyanka Jain. So, we'll start the oral presentation with the selected candidates here. So, the first candidate selected for the oral presentation for day 2 is Dr. Dipali Gupta, who is from AIMS, New Delhi. Okay.
So this is the time for quick coffee break. Then we will back in 10 minutes and start the oral presentation. So please, thank you.
A very good afternoon to all of you. First of all, I would like to thank organizers to provide me this opportunity to present my work. I am working on aurora kinase proteins. Aurora kinase are the serine 3 protein kinases that regulate the different stages of the cell cycle. Basically, aurora kinase family of protein consists of 3 members: aurora kinase A, aurora kinase B, and aurora kinase C. Aurora kinase A, B, and C are homologous to each other, but they have difference in their function and localization. Aurora kinase A is located in the centrosomes in S phase and localized to spindle poles during mitosis, where the aurora kinase B is present at the inner centromeric regions and migrate to spindle zone and midrib during the anaphase and telophase. Aurora kinase A and B is present in the somatic cells, while the aurora kinase C is present in the germinal cells. Here you can see the domain organization of aurora kinases. Aurora kinase consists of 2 domains, N-terminal domain and the C-terminal domain. In between this, there is a kinase domain. Kinase domain is responsible for the phosphorylation activity. These domains also has the activation loop. Activation of the activation loop further enhance the activate the downstream processing for cell division. Aurora kinase domain is composed of 3 parts. It consists of N-terminal lobe region and the C-terminal lobe. In between the N-terminal lobe and C-terminal lobe, there is the active site. The active site residues of aurora kinase consist of leucine, threonine, and arginine, but it differs in these all 3 aurora kinase A, B, and C. Aurora kinase has been found to be involved in their overexpression was found in many cancers like colorectal carcinoma, glioma, breast cancer, ovarian, and pancreatic cells. However, a limited studies has been found in aurora kinase C. The aurora kinase C overexpression was reported in the breast and prostate cancer cell lines. As I told you, the aurora kinase is involved in wide range of cancers, so they can be used as potential drug target for the development of the new anti-cancer therapy. So several inhibitors of aurora kinase has also been identified in the last decades, such as barasertib, tozasertib, alisertib, etc. They target the different phases of the cell cycle, but due to their organ toxicity, altered drug response, and decreased efficacy, there is a need to develop the new inhibitors which can act as a therapeutic agents. So to identify new inhibitors against aurora kinases, we took the small molecule database and we took the protein structures from the RCSP. RCSP is a depository of the X-ray crystal structures. X-ray crystal structure of aurora kinase A, B, and C are available. So we took the X-ray crystal PDBs of these structures and we docked the small compounds against these proteins. High throughput screening was performed and the docking was performed using the GLIDE software of the Schrödinger tool. Now, to identify where does the ligand bind to my receptor and how strongly it binds, we did the molecular dynamic simulation and MMGBSA calculations. Molecular dynamic simulations were performed to know the stability of the drug ligand protein complex molecule and to calculate the free binding energy of the ligand to protein. MMGBSA was performed using the Schrödinger tool. So here you can see the active binding sites of aurora kinase A, B, and C, and the virtual screening was performed and the molecules were docked at these binding sites. Here you can see the difference in the binding cavities of aurora kinase A, B, and C. So on the basis of GLIDE score, We obtained a glide score for many small molecules. Here I am representing only the 4 or 5 molecules. So VX680, which is a pan inhibitor against aurora kinase, was used as the positive control, and the values and the energy is greater than -6.26, -7.3, and 5.23 was used as a standard, and above these values, compounds having above these values were taken for the further studies. Here you can see the binding pattern of these molecules. The dotted line represent the hydrogen bond interactions. You can see the maximum hydrogen interactions in compound 5326 that make maximum hydrogen bond interactions with all 3 aurora kinases. Then further, this represents the hydrogen bond interaction and hydrophobic interactions of these small molecules with aurora kinase proteins. Here you can see in compound 5326, the compound binds to the alanine and glutamate moieties of the aurora kinase proteins. To further determine the stability of these drug ligand protein complexes, molecular dynamic simulations were performed for 100 nanoseconds. The compound 5326 was found to have stable trajectories in all 3 aurora kinases. So we further taken these compounds for the in vitro analysis. RMSF values was also observed for these inhibitor and the protein molecules. RMSF value indicate the amino acid fluctuation during the simulation times. So we procured the compound 5326 and we do the in vitro analysis. For this, first we cloned, express and purify these 3 aurora kinase A, B and C. And we did the protein binding study experiment, ligand compound and protein binding studies using the BLI. BLI is a biolayer interferometry, which is related to the change in interference pattern. As the drug molecule or the small molecule binds to the protein, the interferometry pattern changes. The aurora kinase proteins were immobilized on the nickel anti-sensors, and the compound in different concentration was tested. For aurora kinase A, we obtained KD value of 5.57 micromolar, while for aurora kinase B, we obtained the lowest value of 10.5 micromolar, and while for aurora kinase C, we obtained the value of 30 nanomolars. Then to check the inhibitory potential of this compound, inhibitory assay was performed using the ADP glow assay. In this, the phosphorylation activity was found to be inhibited against this compound 54326. Minimum IC50 was found to be 10.02 micromolar for aurora kinase B protein, but the already existed inhibitors has much lower IC50 values than our reported inhibitors. So we are consulting with medicinal chemistry person to further modify these structures and further modify these compounds to have a better inhibitory potentials. So I want to thank Department of Health Research, Ministry of Health and Science for providing me the grant to do this research, and my guide, Dr. Puneet Kaur, for guiding me to do this work. Thank you.
Thank you, Dipali. So any more questions for a talk? There is one question.
So, hi, indeed, this is very interesting talk. So you have designed this type of molecule. So what is the rationale behind to design this type of molecule?
Yes, we want to design this molecule. So we want to have a new inhibitors for this against aurora kinase. So we tried the kinase library, as this is a kinase inhibitor library. This is a kinase protein. Just to have a new inhibitor, we are searching more number of inhibitors. So this was just one work only. We are dealing with more of the inhibitors and all. We procure these inhibitors and do the
Okay.
activity.
So you selected a library and then you did the docking
Yes,
study,
yes,
right?
yes, yes.
So based on this score, you checked the
ADME
molecule,
properties.
yeah, the molecule and docking score.
Yes,
How
yes.
you'll consider this the binding cavity? You define this.
Yes, yes. In this study, VX680 was used as a positive control, which binds to all three aurora kinases. So this was the binding site. So we superimpose these structures with the already observed inhibitors, and we found that it is the active site at which our molecules are binding to, our inhibitor molecules are binding at the same site as the site we want to target.
Okay. Can you give some input? What are the main functional group required for the activity or binding
Pyrazole
affinity?
group, pyrazole.
Pyrazole. Okay.
Yes,
Okay.
these all are the ATP analogs, but the compound group which was responsible for activity was for the pyrazole group.
Pyrazole scaffold. Okay. Thank you so much.
Okay. Thank you.
So thank you, Dipali. So I would like to call our next presenter, Professor Anna Preto. She's from University of Menho. So welcome you. Yeah.
Just in the okay. Yeah. Yeah, that's fine. So, good afternoon to all. First of all, I would like to thank the organization. It's a pleasure to be here. And I would like also to thank mainly Professor Vinet and all the students that have been amazing and all the organization for this. So, I work in Braga, beautiful Braga, in the University of Minho, and in CBMA, that is the institution where my group is working. And so we've been working for a long time in a model, colorectal cancer, and the reason we work on colorectal cancer is because it's the second leading cause of cancer worldwide, and so far there are a few therapeutic approaches. And there are several causes and several risks of colorectal cancer. And one of the risk of colorectal cancer is bad diet. And bad diet we can easily substitute by good diet. And this good diet, I'm talking about some of the good diet, namely dairy products. And why dairy products? Dairy products, they mainly those from very smelly cheeses that normally we don't like Camembert, Roquefort, and so on. They are very rich on some microbiota, namely propionibacteria. And these propionibacteria, they produce very interesting metabolites. So the propionibacteria produce a high level of short-chain fatty acids, namely acetate, butyrate, and propionate. And these short-chain fatty acids, they are in a ratio in the intestine, so in the colon, and they normally have a protective role. So what is known is that in colorectal cancer, the microbiota is mainly altered and there are a decreased production of these short chain fatty acids. And my group for a long time have been interested in understand a little bit the mechanism of action of some of these metabolites, namely acetate. So we've been working mainly on acetate. And other groups already show that not only acetate but butyrate and propionate individually, so in different works, they regulate important issues on colorectal cancer cells. But nobody had already proven and showed that what happened when we mix all these three together in the physiological ratio. Do they behave as the separate as they are individually? Are they potentiated? So this was the question that Sarah, that is my one of my PhD students, asked and went to understand. So what Sarah has been doing has been studying the combination of the short chain fatty acids in the physiological ratio and understand a little bit if this could be important implications for the therapeutic or the implications for the therapy of colorectal cancer. So Sarah we choose a non-cancerous cell line or a cell line derived from normal adjacent to tumor cells and two colorectal cancer cell lines to understand a little bit what's happening with these combined short chain fatty acid produced by the microbiota at the intestine. And what Sarah saw and we've been seeing is that these short chain fatty acids growth in a dose dependent manner and seems to be very high specific to colorectal cancer cells. So we saw that the selectivity index is higher than 1. So this is a good thing. So it means that they seem to kill more specifically the cancer cells and not causing so much damage to the normal cells. And then what we did, what Sarah did, we went to study what happened to the effect of the mixture here in gray, comparing with the same concentration of the short chain fatty acids individually in the mixture. And as you can see here, the mixture has a higher effect than the fatty acids separated. But interestingly, the most potent, and this is true for all the studies that Sarah did, the most potent short chain fatty acids seem to be butyrate. As a control, we will also study them individually in their IC50 doses, just to understand a little bit or control what was already known. So the first result she got is actually that the mixture of the short chain fatty acids produced by the microbiota, they inhibit colony formation assay. Then what she saw is that they induce cell cycle arrest, and this is the same that they do when they are separate. But again, the mixture has a higher effect on the colon cell lines. They induce apoptosis, mainly early and late apoptosis, and again, looking at the mixture, the effect was really higher when compared with them individually. And then we went to see whether there could be any change on the metabolism of the cells, because we previously have shown that acetate lead, at least acetate, lead to the increase of monocarboxylate transporters. We publish that also acetate induce lysosomal membrane permeabilization, induces acidification of the cytoplasm, induce production of lactate, but we didn't know nothing about the mixture. So what Sarah went to study was exactly to prove whether or not the mixture that we have in the intestine could have an effect on lactate production, and she saw that it increases lactate production. She also saw that lysosome permeabilization was promoted also by this mixture, and very and a higher proportion when comparing with them individually. Also, it leads to the acidification of colorectal cancer cytoplasm, and again, very in a higher proportion comparing with them individually. And this was really very important to show that our intestine, if we increase the microbiota, the good microbiota, and we increase the levels of this mixture of short chain fatty acids, we can kill cancer cells. So the question we asked further was, okay, so could these short chain fatty acids potentiate the chemotherapy drug? Because the colorectal cancers, they are resistant even to 5-FU. So these cells are very resistant to the main chemotherapy, and there are not a lot of therapeutic approaches unfortunately for this type of carcinoma. So what we did is we study the combination of the short chain fatty acids with the 5-FU using 3 models. So we use cell lines, it will be model, we use spheroids, and we use also the in vivo assay to understand a little bit what was going on, and if indeed if we increase the levels of short chain fatty acids in the intestine, we really could increase the sensitivity of the patients to 5-FU. And what we saw was that actually there was really a high increase when we mixed the low levels of 5-FU with the mixture of the short chain fatty acids. We decrease in a high level the cell growth. This was true also in the 3D model. And then when we moved to the in vivo assay, what we saw is that the mixture of the 5-FU with the short chain fatty acid reduced a lot the tumor area, and there was a very high potentiation of the effect of 5-FU. And of course, we went to see whether the proliferation in this tumor was reduced, and as you can see, as you can see here, also again, the mixture reduced a lot the proliferation of these tumors. So in conclusion, what Sarah showed was that the mixture of these 3 short chain fatty acids are very produced by the microbiota, namely the propionibacteria of the intestine, is very important in the intestine, and the increased can induce decrease of cell growth in colorectal cancer by inducing cell cycle arrest, activating apoptosis, inducing lysosomal membrane permeabilization, decreasing the acidification of the cytosol. Moreover, when we tested the effect of the potential, the possible potentiation of this short chain fatty acid with the more used chemotherapeutic drug, that is 5-FU, what we could see is that indeed the short chain fatty acids increase the effect of 5-FU in tumors, and we are really very excited about these results. So the take-home message, and this has been published already, but the first part, not the second part. We are still finishing some results concerning the potentiation of the 5FU, but we can say that we showed for the first time that the combination, the physiological combination of all these metabolites of the microbiota from the intestine, they are specific for colorectal cancers, and they have a potent anti-cancer effect, being able to potentiate at least 5FU. We are testing other drugs. And of course, we can think that this could be a new complementary therapeutic approach for CRC. And as you know, fecal microbiota transplantation now is something that is already in the clinic, and also changing the diet. And we are also submitting a project on producing a symbiotic for increasing the levels of these short-chain fatty acids in the intestine. So this will be the next steps. And concerning the basic science, the next step is this. Actually, we got the cover issue in International Journal of Molecular Science. We are now interested in understanding the effect of the modulation of the short-chain fatty acid in the tumor microenvironment, and how this can impact cancer therapy. So we're just starting this new project now. And these are the teams. So Sarah, me, of course, Elizabeth, that is from the University of Brunel, and Fatima Baltazar, of course, I collaborate with her for a long time in metabolism. She's the metabolism girl, I'm not. So, and I have to thank my team. This was last week. So we normally have fun also, because science is also fun. So we go to lunch several times. So only girls, one boy. He just finished the PhD, so now just girls on the lab. And this actually was the last meeting we went together before COVID. So and they are the 5 PhD students, and those are the new master students. So and thank you very much for your attention. So now.
So thank you, Anna, for the wonderful presentation. So we are open for the questions here.
I was interested in the effect of lactate production after adding the short chain fatty acids. That you showed it very quickly, but it seemed like you had no effect at 24 hours, but then you had the effect at 48 hours.
Is it on? Because I cannot hear. Yes.
Yeah, I was asking about the effect of adding the fatty acids over 24 hours. It looked like there was no effect, but you saw the effect at 48 hours. Seems like kind of a slow-ish response when considering the kind of pathways that are involved. So I'm wondering if you think that the induction of the lactate is sort of a secondary response to the addition of the fatty acids, and maybe what was happening in between there. Was it somehow regulating the expression of the enzyme, or what's the mechanism? Because I would have thought if it was going to have the effect, it would have it within minutes, hours, but it took 48 hours.
Mm-hmm. This is a very interesting question. Maybe I'm not the best person to answer that, but I will try to. The thing is, we don't know much about the regulation of short-chain fatty acid in metabolism. What we know from the lessons and what we know from ASTATE is that ASTATE seem to regulate the MCTs, also the aquaporins. We know that at least ASTATE lead to an increase of CD 147. So the chaperone of the MCTs. We know that it leads to a decrease on glucose consumption and high increase of lactate. We haven't explored yet what is the mechanism. I think it's very interesting. I think we might go for that, and is something that I think we should look in the future. So thank you.
Perhaps it's a little interesting in that typically cells with higher lactate production are growing faster, but but your response in the end is the cells grow slower or not at all, or they die off.
And the thing is, we have acidification. We have cytosolic acidification with these molecules, and what is also interesting is that, of course, lactate should be also. The cells kind of have to get rid of lactate because they are already so acidificated. That might be a way of feeding back the acidification of the cytoplasm. We don't know. We have to check for it. But thank you for that.
Thanks.
Yes, I'm here.
Yeah.
Now here.
Yes.
There was interesting question of interesting results on the combination of 5FU with these propionates. I actually want to refer to the previous answer. Having studied 5FU for decades and looking at different exposure times for 5FU, the effect of 5FU is very minimal when you expose for 1 hour, a little bit better than 24 hours, but you usually need really 48 or 72 hours exposure. And that's why the clinical FOX regimen is for this time period, 48 hours. You need this time to inhibit thymidine synthase, the enzyme, sufficiently to get this anti-tumor effect. However, my question is that you didn't see much effect with the 2D and the 3D, and that was but nicely in the CAM, which was really nice. So
sorry, I didn't say you haven't seen.
You studied the combination in 2D systems and 3-dimensional systems, and in the CAM assay. Yes.
I can't understand the question. Sorry. What is the question? Sorry.
The question is, why was the effect in the 2D systems and the 3D systems much less than in the CAM assay?
Oh, okay. Okay.
And I also would like to know whether you investigate oxaliplatin or combination with 5FU.
Okay, they are, I think we cannot compare, right? So in the CAM assay, we induce a tumor. We inject the short chain fatty acids actually in a similar dose, but the dose is different. And comparing with the 2D is, I think we cannot compare in terms of time and in terms because we are talking about a tumor or a 3D model because the spheroids you have the availability of the cells to the short chain fatty acids is also different. So I would expect that the time point to have an effect will be different in the 3 models. So I don't know if I answer your question but it is an interesting and an important question. And of course, this is just a very, very preliminary and preclinical proof of concept. And of course, we really have to understand a little bit more, maybe also using in vivo xenografts, mice xenografts, and to confirm. But actually, the CAM assay is being very well accepted by the scientific community as a good in vivo model for start. Of course, not then to of course we will have to run other things.
So thanks, Senna. So any more question can be asked in the break because we are running out of time.
No? Okay.
Thank you. So the next presenter is Miss Nusrat Nabi from University of Kashmir.
Hello, everybody. I'm Nusrat Nabi, working in Department of Biochemistry, University of Kashmir. Today, I'll be talking about FAC and CDK16, novel druggable mitotic kinases providing resistance against chemotherapeutic agents. So the development of a cancer, as all of us know, that is a very intricate process in which a healthy cell undergoes an oncogenic event. If the DNA damage is unfixable, cells undergo apoptosis. But in case of cancer, cells keep on dividing and gain further mutations, which leads to the formation of malignant tumor or cancer. Now, to reduce this tumor mass, a number of chemotherapeutic drugs have been developed which kill these cancer cells, but still a small proportion of cells elude this death and continue to grow and survive, which leads to the formation of resistant cells which have stem cell-like properties. These cells are basically responsible for chemotherapeutic resistance, which is the major problem all the cancer biologists are facing all over the globe. Now, the human kinome, that is the complete set of protein kinases, is composed of 600 kinases, which in turn are composed of 400 serine threonine kinases and 200 tyrosine kinases. In our lab, we had purchased a library of 200 serine threonine kinases and screened it against polyoma small t-induced cell death. So what is this polyoma small t? Polyoma small t is basically one of the early set proteins of polyoma virus, which when overexpressed in mammalian cells cause overwhelming mitotic arrest. As you can see these small tiny spheres in the microscopy pictures, these are basically the cells which have been arrested in mitosis. But there's a twist in the story. When this polyoma small t is overexpressed for a prolonged time, it causes mitotic arrest induced apoptosis. The death is built almost of the same manner as caused by different chemotherapeutic agents like alkaloids and taxanes. This is basically a crystal violet image where you can see like after the expression of polyoma small t for 48 hours, all the cells have died. So, using this library screening strategy, we almost shortlist 7 candidates which were able to overcome this polyoma small t-induced cell death, as you can see from this crystal violet picture, where polyoma small t has caused death of almost all the cells, but when overexpressed with certain kinases, cells were able to elude this arrest and continue to grow and divide in the same way as shown by cancer cells against different chemotherapeutic agents. So the shortlist kinases that we shortlist after library screening were PICTER1, FAC, TAUK3, GRK5, MAP kinase 12, and PLK1. I was primarily working on PLK1, PICTER, and FAC during my PhD. As already mentioned, both PICTER and FAC were able to overcome the polyoma small t-induced cell death. This is a representative picture of the crystal violet picture of plate, where you can see that PICTER1 and FAC have basically were able to provide substantial resistance against polyoma small t-induced cell death, and polyst has caused death of majority of cells. But polyoma small t is not a very relevant thing, so we wanted to see if this resistance is provided against different chemotherapeutic agents as well. So we treated the cells with paclitaxel and nocordazole, 2 well-known chemotherapeutic inhibitors. Interestingly, we found that the resistance shown by FAC and PICTER was also against both paclitaxel and nocordazole. I am not very big fan of overexpression studies, so we did knockdown of PICTER and FAC and again treated the cells with paclitaxel, and we found that once the cells are knocked out of PICTER and FAC, cells become more sensitive towards paclitaxel-induced cell death. So we concluded that PICTER and FAC provide substantial resistance not only against polyoma small t, but against different chemotherapeutic agents as well. So we used an inhibitor of PICTER1, that's CDK16, interleukin. We again, screened the cells for polyoma small t-induced cell death, but now cells were not able to resist polyst induced cell death in presence of PICTER1 inhibitor, that is interleukin E804. We performed live cell imaging of polyst and polyst-PICTER cell lines, and we found that cells overexpressing PICTER1 were able to exhibit normal mitosis as compared to the plain polyoma small t cells. Furthermore, we performed a double thymidine block for 16 hours where we, arrest the cells in S phase of cell cycle and then allow them to proceed to different phase of cell cycle. And we found that once cells were entering into mitosis, as you can see from the, increasing the cyclin B1 levels and phospho-CDK CDK substrate phosphorylations, the expression of FAC and PICTER concomitantly increased. It gave us an idea that probably these proteins are having some role in mitosis. We checked the effect of different inhibitors on FAC and PICTER1, and surprisingly, we found that different inhibitors like paclitaxel, nocortisol, hydroxyurea, etc., are increasing the expression of FAC and PICTER1. So we assume that cells are somehow, trying very hard to fight with these inhibitors by increasing the basal levels or endogenous levels of FAC and PICTER. Till now, we had concluded that these two proteins are having a pretty good role in mitosis. So we wanted to see its localization in different phases of cell cycle. And interestingly, we found that PICTER1 is localized on centrosomes of the, here you can see on the centrosomes of the spindle. Not only on the centrosomes, but PICTER1 is localized on the mid-body of the central spindle as well. So we concluded that PICTER1 co-localized with gamma-tubulin and is present at the spindle poles and at the mid-body of the mitotic spindle. Furthermore, now we had concluded that PICTER is localized on spindles, but does it have any role in mitosis? So we did a knockdown of PICTER1 and checked its effect on spindle microtubules. And we found that once PICTER is knocked out of the cells, cells are not able to form proper spindles and are not able to exhibit normal mitosis. So we concluded that PICTER knockdown causes various mitotic abnormalities within the cells. Next, we did some bioinformatic analysis on PICTER1 and found that PICTER is having a very potent site of for tyrosine kinases, and FAC, that was already a library shortlist candidate, is also a tyrosine kinase. And we did a transient transfection of FAC in PICTER1. We found that with increasing the amounts of FAC, PICTER1 protein levels also increase. We confirmed our results by overexpressing FAC and knockdown in the FAC knockdown cells, and we found that PICTER protein levels are high in FAC overexpression cells and low in FAC knockdown cells. Whether PICTER is stabilizing FAC by directly interacting with it or just using some adapter protein, we did an immunoprecipitation assay and we found that PICTER and FAC are directly interacting with it and somehow are helping cells together to, fight the arrest caused by different chemotherapeutic agents. So once cells are arrested in mitosis, PICTER and FAC form a complex and help the cell to come out of mitosis and exhibit a successful mitosis. This is an interesting slide. We checked the expression of PICTER and FAC in different ovarian cancer samples, where we found that the expression of PICTER and FAC is high in tumors. But we were very fortunate enough that we got a recurrent sample of the same patient. We found that in the recurrent tumor samples of the same patient, where the cancer is more aggressive with more stemness, PICTER and FAC levels are increasing to a larger extent. So we concluded that PICTER and FAC are overexpressed in ovarian cancers. I'll summarize my results. PICTER and FAC are novel mitotic kinases. They are able to overcome mitotic arrest that is caused by different chemotherapeutic agents as well as polyoma small t. PICTER1 particularly is extremely important to exhibit normal mitosis, and it therefore makes it a druggable target for cancer. Furthermore, PICTER is regulated by FAC within the cell. Being a serine threonine kinase, it's regulated by tyrosine kinase. And PICTER and FAC are upregulated in ovarian cancer, especially recurrent tumors. I would like to thank my supervisor, Dr. Shahida Andrabi, organizing committee of IELC 2023 for allowing me to present my work here. Our collaborators, patients for giving samples, my faculty members and fellow scholars of Department of Biochemistry, University of Kashmir. I would also like to thank our funding agencies for providing grants to Dr. Andrabi. Thank you very much.
Thank you, Nusrat. So any questions, please? Quick.
In your introduction slides, you mentioned about the MAP kinase 12, right? This is like delta MAP P38, right? P38 delta.
Yeah, P38 gamma.
Yeah,
MAP
P38
kinase
gamma.
12. Yeah, yes, sir.
So, I mean, 'cause seeing all of your program of blocking the FAC and IR protein, so does blocking these protein give these tumor cells some advantage of survival, or does it really, or does it uh, uh, control or inhibit some kind of their differentiation and metastasis index?
Right now, we are assuming that because FAC and PICTER are localized on centrosomes and on mid-bodies, and formation of mid-body is basically destines the cell to exit the mitosis. So somehow these proteins are helping the cells to exhibit normal mitosis, either by correcting the spindle abnormalities caused by drugs, or probably by forming a proper central spindle or mid-body, and then pushing them out of the mitosis. Cells are not able to correct those abnormalities; they just divide with those abnormalities already caused by drugs. Therefore, once the abnormalities accumulate within the cells, obviously tumor cells become more aggressive and they have zero effect of chemotherapeutic agents. So
it
yeah.
means it's you have not checked about their sensitivity for the first line, second line TKI inhibitors.
Not yet. We are still doing the experiments, but yeah, till now we have just concluded, although we have a lot of other data as well, but we are yet to go towards xenograft models and organoids basically. But till now we are we are concluding that absence of these two proteins definitely causes abnormalities within the cells, and if these proteins are knocked out of the cells, cells are not able to exhibit proper mitosis.
Okay, nice.
So thank you, Nusrat.
Thank you,
Thank you, please. So our next speaker is Dr. Lakshmi from KLEF, is a deemed to be university, is our next speaker.
Good evening to everyone. Myself Lakshmi Vinila. I'm here to present a topic of my PhD research entitled as empagliflozin mediated microRNA 128-3P upregulation promotes differentiation of hypoxic cancer stem-like cells in breast cancer. So breast cancer is the second leading cause of deaths in the women worldwide. And if we see 1 in 8 women, US women were found to have the invasive breast cancer in their course of lifetime. So from the clinical breast cancer patients, we have seen a critical observations that 87% of the advanced breast cancer patients are having a loss of heterozygosity at chromosome 3P. So due to the loss of this heterozygosity at chromosome 3P, there is a loss or the amplification of few microRNAs. So from these 16 microRNAs, it was found that microRNA 128-3P was the most commonly deleted microRNA. And due to the deletion of this microRNA, it is correlated with poor survival, recurrence, and chemoresistance of the advanced breast cancer patients. So, however, the relation between these microRNA 128-3P with the stemness has not been clear. Generally, these poor survival, recurrence, and chemoresistance are the characteristics for the cancer stem cells. So, if there are more of cancer stem cells, we generally observe the recurrence and chemoresistance. So, now we would like to know what is the relation between the microRNA 128-3P with the stemness. And in our hypothesis, we have a second target, that is PKM2. So PKM2 is a glycolytic protein which is involved in the glycolysis, involved in the rate limiting step of the glycolysis. And this exists in the form of dimers and tetramers in the cancer cells. So this dimeric form of PKM2, generally it translocate to nucleus and acts in the protein kinase action. So as a protein kinase, it generally activates various other several proteins which are present in the nucleus and helps in the progression of cancer. So, and further it also has a role in the hypoxic environment where PKM2 is a direct target for the HIF target gene, And also PKM2 helps in the tumorigenesis in the hypoxic conditions. And even it is also found that hypoxia upregulates PKM2 and also promotes metastasis. Also, it is also found from few studies that it is also involved with the stemness. And the unknown facts are like, we would like to know whether microRNA 128-3P regulation will it have any down regulation with the PKM2 levels in the hypoxic breast cancer cells. We would like to look into this approach because from a paper we have found that hypoxia induces PKM2, it increases the PKM2 levels, and also it is found that microRNA 128-3P like it directly inhibits SP1, which is a transcription factor. And from papers, we have seen that SP1 acts on the promoter region of the PKM2 and increases the PKM2 transcription level. So thereby, we would like to see whether microRNA 128-3P mimics can downregulate the PKM2 in the hypoxic breast cancer. And we would like to see the mechanism of PKM2 inhibition and also what is the effect of stemness in the hypoxic cells. So we have seen from our study that hypoxia increases SP1 and PKM2 levels. And we also found that microRNA 123P levels were significantly reduced in the hypoxic breast cancer cells. Further, we checked the levels of microRNA 128-3P and we found that the microRNA 128-3P levels were significantly reduced in the hypoxic breast cancer cells. And with the treatment of mimics and siRNA PKM2, we have seen an increase in the levels of microRNA 128-3P. And with inhibitors, we have seen the opposite level. And then we checked the effect of microRNA 128-3P transfection and also SIPKM2. And we have seen the levels of SP1 and PKM2. And we found a significant reduction in the SP1 and PKM2 levels in the siRNA treated PKM2 and also mimics treated. Whereas we have found a reversal effect with the inhibitor treatments. Then we have seen the stemness markers where SOX2, OCT4 and NANOG levels were checked. And we found a significant reduction with the siRNA of PKM2 and mimics. And whereas with the inhibitor treatment, we found a significant increase in the stemness markers. Then we looked into the differentiation of cells, like generally CD24 minus and CD44 positive cells were having a stemness markers in breast cancer, whereas the CD24 positive and CD44 positive cells were thought to have like differentiated phenotype. So we looked into the population of CD24 positive and CD44 positive in our treatments, and we found that the ratio of CD24 positive and CD44 positive were significantly increased with the SIPKM2 and mimics treatment, whereas with the inhibitors, we found a decrease with this population. And we also found the same type of phenotype in the MDMB 231 cells. So from these results, we can conclude that microRNA 128-3P has a potential role in inhibition of cancer stem cells. So generally, there is a limitation for the microRNA therapeutics to reach the clinical trials. Generally, this is due to the microRNA therapeutics, they are highly costly, and we need high quantity of these microRNAs for the preclinical and clinical studies, and also product development is difficult, and even it is patient, like it has lower patient accessibility and also has stability concerns in serum. So therefore, we thought of finding a molecule or a drug which is already FDA approved drug, which can mimic the functionality of microRNA 128-3P. And we have screened for drug molecules, and we found that empagliflozin, which is an anti-diabetic drug and an approved FDA molecule, which is in the market, was found to significantly upregulate this microRNA 128-3P. So then we started our treatments with the empagliflozin, and we found that empagliflozin treatment has significantly increased the levels of microRNA 128-3P in both MCF7 and MDMB 231 cells. And later we have seen the levels of PKM2 and SP1, and we have seen that there is a decrease in the levels of SP1 and PKM2 with the treatment of empagliflozin. Then we have seen, we have checked the population of differentiated cells, and we have seen that with the treatment of empagliflozin, there is a significant reduction in the population of differentiated cells, increase in the population of differentiated cells. Then we have carried forward our in vivo experiment with the orthotopic mouse breast cancer model, where we have injected the 41 cells, and we have divided the group number of animals into 3 groups with a control, empagliflozin 10 mg and 25 mg per kg. And then after 4 weeks of treatment, we have performed the various analysis. And we have seen that the body, there is a negligible change with the body weight, whereas we have seen that there is a significant reduction in the luciferase expression after the treatment of empagliflozin for 4 days. And we also found that there is a significant increase in the microRNA 128-3P expression in the isolated tumors after the sacrifice. Then we have checked the H&E staining of the isolated tumors, and we have found that in control and low dose of empagliflozin, there is a lung metastasis, whereas the high dose of empagliflozin has shown no metastasis in all the animals. And then we performed the ICC for SP1 and PKM2, and we have seen that there is a significant reduction in the SP1 and PKM2 levels in the empagliflozin treated animals. Then we have further checked with the CD24 and CD44 positive cells and also Ki67 proliferation marker. And we have seen that there is a significant reduction, a significant increase in the CD44 and CD24 positive cells, whereas there is a significant reduction in the Ki67 proliferation marker. So this is a summary of our results, where we have seen that empagliflozin, by upregulating the microRNA 128-3P, it can significantly reduce the CD44 positive and CD24 negative cells, and also it reduces the SP1 and PKM2 levels, and further it also decreases the stemness markers in the breast cancer cell. So I would like to acknowledge the Department of Pharmaceutical, Ministry of Chemical and Fertilizers, Government of India, Naipur, Ahmedabad, and also Koneru Lakshmaya Educational Foundation, KL University, and also my mentor, Dr. Amit Kaina. Thank you.
So thank you, Dr. Lakshmi. So due to lack of time, we can have questions in the break session. Thank you. So our next speaker is Ms. Swarnali Kar. Yeah.
Good afternoon, everyone. I'm Swarnali. I'm working as an ICMR SRF at Bose Institute, Kolkata. So I'll be talking about aerobic glycolysis today and how differently it can promote cancer progression. So, oh, ma'am, what is the point? Okay, and what is the pointer? Okay. Okay, thank you. So here the cancer cells are basically, they are asking for more lactate here. As we all know that rather than acting like a waste material, lactate has different roles in cancer progression. So I'll be starting with a brief, very brief introduction. So since it was established that cancer cells rely largely on aerobic glycolysis, it has been considered to be a great target for cancer therapy. And in the past many years, different small molecule based anti-cancer drugs have been designed targeting this pathway. But unfortunately, their clinical translation is still very limited. So I think that a better mechanistic understanding is required. So if we look at tumor microenvironment and actually glycolysis within tumor microenvironment, we can assume that there are several factors. Those can actually activate this glycolytic switch. On the other hand, glycolysis is related with different cellular processes, thus actually facilitating cancer progression. Now, these factors, the factors within tumor microenvironment, they actually activate glycolysis by modulating a few glycolytic enzymes, and they do so by upregulating different sets of glycolytic enzymes. So it's basically a multifarious event, and I will be addressing two issues here, that how diverse this glycolytic process is in the context of cancer progression, and if we are if we are going to target this cancer metabolism, should we modify our strategy, and if yes, then how should we plan this? So in order to address both these issues, I would like to share two studies here where two oncogenic yet different, functionally different proteins are associated. The first one is a growth factor, specifically 5 with FGF receptor inhibitor, the reversal was not at all significant. So we then measured the intracellular concentration of a few glycolytic intermediates, and we observed that glucose 6-phosphate, 2-PG, and lactate were significantly increased in presence of FGF16. And now lactate is considered as the glycolytic end product for aerobic glycolysis, and lactate is secreted out of the cells. So we measured the extracellular lactate concentration, and we observed that in presence of FGF16, the lactate secretion is also significantly higher. So it was a general conclusion that FGF16 can induce glycolysis and lactate production as well as lactate secretion. Now the most interesting part of this study was that FGF16 can significantly upregulate the regulatory protein PFK-FB4. Now PFK-FB4 is a member of PFK2 group of genes that catalyzes the conversion of fructose 6-phosphate to fructose 2,6-bisphosphate. Now, fructose 2,6-bisphosphate, on the other hand, can activate PFK1. Then we tried to validate the data by means of qPCR and Western blot assay, and we observed that indeed in presence of FGF16, PFK-FB4 was significantly upregulated. So now our question was that whether in presence of FGF16, indeed PFK-FB4 was regulating this whole aerobic glycolytic pathway or not. So we measured the extracellular lactate concentration again, and we observed that when we silenced PFK-FB4 in presence of FGF16, the extracellular lactate concentration was significantly lower. So now, long story short, we then extended our study at phenotypic level and performed an invasion assay and observed that here also when we silenced PFK-FB4 in presence of FGF16, the number of invaded cells, those actually invade the membrane used in during the invasion assay that actually resembles the extracellular matrix, the number of invaded cells are significantly lower. So this was our conclusion that PFK-FB4 is crucial for FGF16 mediated invasion in MCF10A cells. So maybe if we can target this regulatory protein, we may be able to reduce the invasiveness in breast cancer cells. So for the immediate future plan, we are trying to deduce whether this PFK-FB4 induced invasion is solely lactate based or there are some other molecular machinery involved. So now I would like to move to the second part of my presentation. Here I have worked with a transcription factor, that is PITX2. It is implicated to be oncogenic in ovarian cancer cells. Now our question was whether PITX2 utilizes cell metabolic pathways as a tool to promote cancer progression or not. So in our preliminary studies, we observed that PITX2 can augment cell proliferation in ovarian cancer cells, and it also can differentially alter expression of a few glycolytic genes. So then we carried out our metabolomics experiment and quantified a few glycolytic as well as TCA cycle intermediates, and we observed that here lactate was significantly increased, whereas for succinate fumarate, the TCA cycle intermediates, the changes were not very significant. Then now PITX2 itself is a transcription factor. To understand whether it can directly induce the expression of glycolytic genes or not, we performed chip qPCR assay, and we observed that HK2, INO1, and LDHA were directly induced by PITX2. For MCT1 and MCT4, they are not directly regulated by PITX2, but in case of PITX2 overexpression, they are significantly upregulated. Then we were interested about the crosstalk between metabolism and epigenetics. So we chose the three most popular epigenetic modification: DNA methylation, histone methylation, and histone acetylation. I'm not going to talk about these two here, but for histone acetylation, we observed that in case of PITX2 overexpression, there is a global hyperacetylation of H3 molecules, histone 3 molecules. So we thought maybe PITX2 is involved in any pathway leading to acetyl-CoA production. So we noted down every pathway leading to acetyl-CoA production, and we tried to block one pathway at a time to find out which pathway is used by PITX2. But unfortunately, we are very disappointed, and we found no significant link between PITX2 and acetyl-CoA production. So we were left with the next option, that is change in deacetylation event. So if we have to create a pool of some molecule, we can increase the synthesis, or on the other hand, we can decrease its degradation pathway. So when we measured the HDC activity, we observed that in case of PITX2 overexpression, the HDC activity is significantly lower than the untreated cells. And the observation was same as when cells were treated with external lactate. And this observation was verified with the Western blot analysis. Now, in the meantime, we performed another round of CHIP qPCR to understand whether this relative hyperacetylation can influence the proliferation pathway, proliferation process or not. So we chose two proliferation marker genes, PCNA and Ki67. For both the genes, we observed that there is significant upregulation or significant overexpression. Their expression is facilitated by this hyperacetylation. Then we tried to restore the HDAC activity, but it was very interesting that when we blocked the glycolytic pathway by 2DG treatment, there was a mild increase in HDAC activity. But when we tried PITX2 2DG along with the MCT1 inhibitor, there was satisfactory restoration of HDAC activity. So we could conclude that maybe MCT1 inhibitor along with 2DG can restore the HDAC activity to full extent. So then we concluded this study with the expression analysis of proliferation genes, and here also we can observe then that in presence of PITX2, when we treated the cells with 2DG and MCT1 inhibitor, the expression of proliferation genes were significantly lower in comparison with the PITX2 overexpressed cells. So it was a general conclusion that PITX2 can induce aerobic glycolysis, and on the other hand, lactate, the aerobic glycolytic end product, it can inhibit the HDAC activity, and by this it allows the H3 molecules to remain in acetylated form for a longer period of time. So maybe MCT inhibitors in combination with glycolytic blocker may provide us with a good result to block the glycolytic pathway in terms of cancer progression. So we are trying to find out whether this lactate mediated HDAC inhibition is solely pH mediated or there are some other molecular machinery. So now I sum up my study. So these two proteins are very different in terms of their function, yet they can activate the aerobic glycolytic pathway, but by modulating different sets of glycolytic genes. On the other hand, lactate can induce different hallmarks of cancer, invasion and proliferation. So if we are trying to target this aerobic glycolysis along with looking into the mainstream glycolytic enzymes, we may look into the regulatory pathway like PFK, FB4, and also look into some combination therapy like glycolytic inhibitor and MCT inhibitor. These are my references. I would like to thank my I'd like to thank my lab members, my supervisor, and also my funding agency, ICMR, and finally this EMBO and Amity University to let us present our work here. Thank you.
Yeah, thank you, Swarnali. So due to loss of internet connectivities, so this will be our last talk, and Dr. Saran Kumar from IIT Delhi. So please stick to 8 minutes. And you can.
Okay. Thank you all, firstly, for the organizing a wonderful conference, Vinith and colleagues. And I understand I have 8 minutes, so I'll be rushing through. But I'm basically Saran Kumar, new faculty at IIT Delhi, Kusma School of Biological, and this is my lab, which is just less than a year old. And we work on mainly different aspects of how vasculature is important in different diseases, specifically also cancer. That is what I would be discussing here. And today's title would be Vascular Influence on Glioblastoma stemness. So just a very brief introduction on the blood vessels. It's constitutes the first organ in the embryo and forms the largest network in our body. And when dysregulated, there are multiple diseases which are associated with this, which, even if it is upregulated or downregulated, both is a problem. And tumors, as we all know, is like an organ anew, so it requires blood vessel supply. And there is this famous Jutta Folkman's term, angiogenic switch, which was already established like half a century ago. But still, there are very challenging questions which are yet unanswered. So what is the outline of my talk? So in the next 400 seconds, I will at least make you interested in two of my stories. One is about how blood vessels dictate metabolic heterogeneity in cancer, which we published in Cell Metabolism a couple of years back, or maybe more than that now. And also the second one is how microenvironment is very important for cancer stem cells, specifically in the case of glioblastoma, which we published just last year. So tumor heterogeneity is one of the major challenges, which also includes the metabolic heterogeneity, right? And the model which we are using is basically the glioblastoma, where there is also necrotic regions on one end, and there is regions which are with perfused blood vessels. And what happens is there is a perfusion gradient, right? There are regions which are very well perfused, there are regions which are not so perfused, but this is a gradient which is not hypoxic or normoxic, but this is a gradient. So how do we study this? So that was the question which we took up on. So what is can we design a tool to study this, where I can label or identify a cell based on how closer am I to a blood vessel in the tumor context? So that was my task. And what we did is basically, very briefly, we designed a perfusion-based cell labeling technology where we could isolate or identify cells even after dissociation where they are present exactly with respect to their vasculature. So if they are closer to the vasculature, they will take the dye higher intensity, so you can easily sort them, and because they have a higher intensity. If they are farther, a little less, and you can fractionate them basically to a different number of layers as much as you want. So that is the PFDLC technique in short. And again, GBM and immunocompromised mice, because we are using human GBM lines here, which are GFP tagged. So there are no stromal lines. And please remember, these are all identical clonal population. So the only difference or the only variability is that where is it present positionally? So what do we see? So this is what I try to explain. I hope the picture represents much better. So it's a thick 50 micron thick section where you can see green is basically the whole tumor's xenograft, and the blue is the perfused dye, and the blood vessels are stained in red. As you could see that there's regions which are very hyperperfused and there are regions which are hypoperfused. And by playing with the methodology with respect to time and the concentration, we can identify different regions. So what does it help us to do? This helps us to do a spatial omics. We could do, of course, metabolomics, tumor metabolism conference, right? And also transcriptomics and other omics. So as a proof of concept, we tried two omics, which is first is the transcriptomics, and also we did metabolic tumor zonation. So again, sorry for being redundant, but yes, these are clonally identical cells. Only difference is where they are present positionally, right? So we tried to do with three different regions: perivascular, intermediate, and hypoxic niches. So you can see a clear-cut graded differences. There are genes which are upregulated and downregulated. Same is the case for the metabolomics also. We had some metabolites which are up in the perivascular region, not so much, and then at least in the hypoxic. And there's also some metabolites which are vice versa, right? And we did multiple other studies, which I don't want to go in details, but please do discuss with me or come to me for any more discussion. Because of the lack of time, I won't go into it. And I request the next time the organizer to give me more time. So the next story quickly is basically the perivascular niche for CSCs in glioblastoma. So we have now a tool to identify basically perivascular regions or hypoxic regions, right? So can we push this further to identify so-called cancer stem cells? One of the major properties of cancer stem cells is quiescence. So that is what we try to merge with my earlier technology, that is of the perfusion-based labeling. What is quiescence? Basically, the stem cells are known to be less dividing. So basically, these will be the cells which will retain a dye. If you stain a particular dye and let the cells divide, there will be some cells which will retain the dye because they are not dividing so actively. And these are putative stem cells or quiescent cells. And what we did is we basically used this in vivo approach. We combined the PFDLC with the perfusion dye and the PKH26, which is a label retention dye, and we found where these CSCs are residing. And are these CSEs? Yes, we did conclude. We have the whole details validation, which has been published in this particular paper. And if you see this particular beautiful picture, you can see the white are the CSEs and the red is, of course, the blood vessels. It reminds me of an epiphyte, which is sticking to a tree and trying to take out the nutrients or metabolites from that. Okay. So very quickly, what did we do further from this? We also try to dissociate or understand how metabolic reprogramming is happening with the endothelial cells, because these endothelial cells are not just, or the blood vessels are not just a passive conduit for nutrients and oxygen, but it does much more than that. It regulates cancer stem cells also. So we use simple co-culture techniques where we again did some tricks where we could really by fluorescent staining as well as by transcriptomically differentiating glioma and endothelial cells. And we could also segregate between perfusion dependent and perfusion independent vascular cues, because we could do in vitro as well as in vivo comparison. And not just that, even juxtacrine signaling or the paracline signaling we could dissociate by using a not specific inhibitor, which basically inhibits the juxtacrine signaling, that is the GSI. So using all these tools, very briefly, we did understand or found out that there are some set of genes which are important in the specifically in the glioma transcriptome. When you co-culture with endothelial cells, they overexpress the cancer stem cell marker, and some of them are immune to any kind of such insults, and some of them only change when you put them with such an inhibitor. So, whether there is perfusion or not, no perfusion or contact dependence, all these three we try to segregate with this. So, cutting the long story short, basically we also try to understand how the notch-dependent metabolic reprogramming is happening. And if you see, there is a big, again, lot of heat maps, sorry, no time to explain, but you could see that there are multiple transporters as well as, glycolysis or PCA cycle genes. And so that is what is the left heat map, and the right heat map is basically how the glioma induced reprogramming is happening. And it's not one direction, it is bidirectional. So basically the glioma is also influencing the endothelial cells and the vice versa. The endothelial cells are also influencing the glioma. So reprogramming is happening bidirectionally, and that is what our study shows. So I hope I was within my time, and thank you so much. And this work was mainly done in Israel as a postdoc, and it's being continued my young lab. And I would like to specifically thank my professor Eli Keshet, who is in Yellow Jacket, and ERC and ISF for funding. And now the baton has been taken over by the SERB funding and the DBT funding, and I would be happy to take questions later, I understand, or for any prospective collaborations or interactions, please. I'll be there for 2 days and I'll be happy to interact. Thank you so much.
Thank you, Dr. Saran, for such a wonderful talk. So he has completed on time, so we'll go with the next session. So I would like Dr. Prakash for the next session.
So for the next session, I would like to call chairperson on the dais, Professor Frederick Boost and Dr. Chandra Prakash Prasad. No? Yeah. Yeah, Dr. Professor Fatima will chair the session. Please. Presenter, no? Stay here. Presenter. So I guess Professor Hidar is already online. So I request Dr. Fatima to take over. So Professor Idar, are you there?
Yes, I'm here. Hi.
Okay, good to see you. Okay, so Dr. Fatima, please.
So it's on. So good afternoon. It is my pleasure to introduce our keynote speaker from this afternoon, Dr. Heather Christoph, who's the Associate Professor of the Department of Biological Chemistry at the David Geffen School of Medicine, UCLA, US. Her area of research is cancer metabolism, and she recently discovered that asparagine is a key signal of mitochondrial respiration that can be targeted to reduce tumor growth. She has received several awards and recognitions, for example, the Scholar, Damon Runyon Rushleff Innovation Award, and NIH Director's New Innovation Award. She has 54 peer-reviewed research publications, each index of 39, and she has also multiple international patents. She also serves as editorial advisory board of many journals, important journals, including Cell Metabolism, and Cancer Discovery. And she also serves as associate director of the UCLA Johnson Comprehensive Cancer Center. So thank you very much, Dr. Heather, and the stage is yours.
Great. Thank you so much for the wonderful introduction, and for to Dr. Vineet Kumar for the kind opportunity to share some of my lab's recent work here at this excellent meeting. My lab at UCLA studies the bidirectional relationship between metabolism and cell state. And we study this in several different contexts that exhibit metabolic transitions, such as development, cancer formation, and virus infection. And some of the questions that we've been interested in lately include: how does maternal metabolism, including prevalent conditions such as diabetic pregnancy, shape fetal development? And also, can we target metabolic dependencies driven by specific cancer mutations or viruses? And related to the second question here, especially since this is a tumor metabolism meeting, I'd like to share with you one unpublished story from my lab on our efforts to identify a fixed metabolic dependency driven by a specific tumor mutation that we think we can now target for therapeutic benefit. And that is a mutation in the gene fumarate hydratase. Many of you likely know that there are a couple of TCA cycle genes, fumarate hydratase and succinate dehydrogenase, that when lost can actually drive tumorigenesis in certain tissues. Fumarate hydratase loss leads to accumulation of the oncometabolite fumarate that, when accumulated in tissues, can drive tumorigenesis in certain contexts. Fumarate hydratase loss and fumarate accumulation drives formation of these adrenal gland tumors, pheochromocytomas and paragangliomas, also a very aggressive form of kidney cancer, and also these benign cutaneous and uterine leiomyomas. On the other hand, succinate dehydrogenase loss leads to accumulation of a different oncometabolite, succinate, which, when accumulated in certain tissues, can drive different forms of tumors, pituitary adenomas, GIST, and then also adrenal gland tumors and kidney cancers. And we, again, are interested in whether accumulation of these oncometabolites, fumarate or succinate, can drive a fixed metabolic dependency that we can target for therapeutic benefit in these tumors. Now, really nice work from others in the field have studied how these oncometabolites have these transforming effects. For instance, the best studied mechanism now is that fumarate and succinate accumulation can inhibit the activity of a big class of enzymes called alpha-ketoglutarate dependent dioxygenases. And this can have big effects on the cells. It can lead to aberrant HIF stabilization and a transcriptional profile that is similar to what we see during hypoxia that leads to angiogenesis and metabolic reprogramming and cell growth and migration. But also, some of these alpha-ketoglutarate dependent dioxygenases are involved in methylation reactions. And so inhibition of these enzymes by fumarate and succinate can lead to DNA and histone hypermethylation, which can affect the epigenetic state of the cell in a way that can transform the cell. A fumarate also can react non-enzymatically with cysteine residues on proteins. And so can succinate proteins through this mechanism. And one example by which fumarate succination on a protein can likely contribute to its transforming effects is that it succinates KEAP1, the tumor suppressor, and dysregulates KEAP1's ability to inhibit NRF2, a transcription factor involved in antioxidant dysregulation. And so that's another mechanism by which fumarate can transform cells. Even earlier this month, 2 papers were published in Nature showing another effect of fumarate accumulation on cells, and that is to promote inflammation. These 2 different nice papers in Nature showed that fumarate accumulation leads to release of mitochondrial RNA or DNA that can drive interferon production and inflammation. And so this is another way that fumarate is affecting a tissue microenvironment and likely contributing to transformation. But this is not the focus of my research. My research is focused on a different question, and that is, does fumarate or succinate accumulation, regardless of how it transforms the tumor, does it drive a fixed metabolic dependency in these tumors, which you might imagine because these metabolites are accumulating to millimolar amounts. And so one could imagine that accumulation of a particular metabolite to that amount in a tumor would affect the tumor metabolism in a way that might make a dependency in the tumor that we can target. So this was really the question that we were after. And with this in mind, we decided to focus on the case of fumarate hydratase loss for several reasons. One is that this fumarate hydratase loss is associated with a genetic disorder, a familial cancer syndrome called hereditary leiomyomatosis with renal cell cancer, or HLRCC. This is an autosomal dominant genetic disorder caused by inactivating mutations in fumarate hydratase. The parents inherit one bad copy of fumarate hydratase, and then over time, they get loss of heterozygosity in certain tissues, which drives tumor formation in certain tissues. The patients get very painful cutaneous leiomyomas. They get really large uterine fibroids. And unfortunately, most problematic for these patients, they get a very lethal form of kidney cancer for which there are no treatments. And these tumors typically form somewhat early in life, in their teens, 20s, 30s, 40s. Now, while fumarate hydratase variants were once thought to be quite rare, now that we do so much germline testing of patients, it's now appreciated that fumarate hydratase variants are actually much more common than we originally thought. They're detected in 1.3% of individuals on germline testing, and actually 0.6% of individuals have a fumarate hydratase variant of uncertain significance, which is a bit of a problem. Because if you have a fumarate hydratase variant, you would want to know if it is a loss of function variant or not. Because if it is a loss of function variant, you should probably be screened for kidney cancer more often and start your screening early, because this form of kidney cancer is very aggressive and right now not treatable. So in all of these fumarate hydratase variants, almost half of them, or over half of them, are variants of uncertain significance or are a variant of conflicting interpretations. And of these variants of uncertain significance, the vast majority are missense mutations, which is just interesting for us to note. But this is actually a problem clinically, like I mentioned, because patients with an FH variant of uncertain significance need clarity on the pathogenicity of their variant. Let me give you an example of a patient at UCLA who is seen by my collaborator, Dr. Brian Shuck, a urologist and director of the kidney cancer program at UCLA. This patient had a family history of kidney cancer. He was 45 when he came to UCLA, and he presented with a large right renal mass that was FDG PET positive. This patient, at the time of that he came to UCLA, his brother had passed away from metastatic renal cell cancer at age 35. His sister had passed away from metastatic renal cell cancer at age 21. At the time that he came to UCLA, his mom had had a hysterectomy, potentially due to uterine fibroids. So Brian realized this looks like maybe this patient has HLRCC. It's a familial cancer syndrome. His mom had a hysterectomy. His siblings, unfortunately, already passed away of kidney cancer. So, and when they looked at the tumor by histology, the histology was suspicious of HLRCC because there was papillary eosinophilic architecture in the kidney tumor. However, somewhat confusingly, the tumor stained positive for fumarate hydratase. However, Brian did see that the tumor also stained positive for 2-succinylcysteine. Remember, I mentioned that fumarate can react non-enzymatically to cysteine residues. And so often this is used as a stain, as a diagnostic stain for HLRCC tumors. So this was a bit of a confusing case. Brian went ahead and had this patient sequenced, and this patient has a germline heterozygous for a variant of uncertain significance in fumarate hydratase. And this variant has conflicting interpretations of pathogenicity in the literature, which means that this variant is not supposed to be is not classified as HLRCC. And people with this variant are not supposed to have their clinical guidelines are not supposed to be dictated by presence of this variant, which is really unfortunate because this patient's daughter and niece also carry this variant and at young ages already have had fibroids. So when Brian was telling us about this patient and how frustrating it is that there are all these variants of uncertain significance, and it's a really big problem in the clinic because patients with variants of uncertain significance, even when they present clinically in a way that seems like they have HRCC, they can't be called HRCC without additional evidences of pathogenicity for the variants. And that's a big problem because insurance companies won't reimburse screening, regular kidney cancer screening for people who have these variants of uncertain significance. So this seemed like a problem that my lab could help with. This just seemed kind of obvious to me. We do biochemistry in the lab, and so a really great postdoc in my lab, Blake Wild, took this upon himself to start his project by classifying fumarate hydratase variants of uncertain significance. He designed an enzymatic assay to measure fumarate hydratase activity. It's based on the absorbance of fumarate at 250 nanometers. And he he designed an activity assay to assess the activity. You can see fumarate hydratase catalyzes the reversible hydration of fumarate to malate, or dehydration of malate to fumarate. And Blake started by just classifying the activity of a handful of variants. Shown here in orange are known pathogenic variants of fumarate hydratase. And as expected in these Michaelis-Menten plots, where the wild type enzyme shows nice activity, each of these pathogenic variants have no activity. They're completely inactive. And then when we looked at a handful of variants of uncertain significance, just 5 of the 5 that we randomly chose, 3 were completely enzyme inactive. Completely inactive, did not fit in the Michaelis-Menten curves. 1 actually had a higher KM for fumarate and lower catalytic efficiencies, and then 1 was not enzyme inactive, was completely fine. And so this gave us encouragement that yes, there are a bunch of these variants of uncertain significance that are in fact completely inactive, and we set out to characterize these. We collaborated with a clinical genetic testing company called Invitae to help us prioritize which variants we would go after. And I'm just showing you a subset of the data that we obtained. This was a really large effort by Blake. What you're seeing here is the catalytic efficiency for fumarate hydratase and all the variants that we tested in the forward direction. The wild type enzyme activity is here in the dotted line, and each different dot is a different variant that we tested. And what you can see here is a bunch of variants that had 0 activity. Among these are known pathogenic variants shown in orange that we use as controls. And then what I'd like you to note is that several of the variants that we tested, each of these was found in a patient, are enzyme inactive. And so of the 74 that we tested, almost half were inactive, which is strong evidence of pathogenicity. And sure enough, the patient's mutation that I showed you as an example a few slides ago, this M151K mutation also was completely an inactive variant. Somewhat confusingly, and I should note that unfortunately this patient has since passed away from metastatic kidney cancer at the age of 47. But now we are hoping that our additional characterization of this variant will help classify it as a pathogenic variant and enable people with this variant, including his family members, to get regularly screened for kidney cancer. One thing that we noted is we assessed the tetramerization status of the different inactive variants of fumarate hydratase. Fumarate hydratase is only active as a tetramer. And one thing to note is that some of the inactive variants formed stable tetramers. So while tetramerization is required for enzymatic activity, tetramerization status is not a good measure of activity. You can see that this variant found in this patient that's completely inactive has tetramerization similar to wild type. Okay, so what we did next is we decided to use this catalog of fumarate hydratase variants that we had assessed for different, assessed biochemically to understand how fumarate accumulation impacts cancer cell metabolism. Remember, we're really interested in whether fumarate accumulation can drive a fixed metabolic dependency that we can target. And so we used this catalog of variants with different levels of catalytic activity to correlate how increasing levels of fumarate in cells causes changes in metabolism, perhaps a fixed metabolic dependency. And so how we did this experimentally is we took our HL-RCC cells that lack fumarate hydratase and so have high fumarate levels, and we made a a cell line catalog in which we've either put in a vector just with GFP, so those cells retain high fumarate levels, or a vector that has wild-type fumarate hydratase, so it rescues the fumarate levels, puts them back to normal, or variants with differing levels of catalytic activity. So then we could correlate metabolite abundance, or different abundances in metabolites, with the fumarate malate ratio. And here's just an example of our catalog of cell lines that we generated with varying levels of fumarate to malate ratio. And what we found here is what I think something quite interesting. So to begin with, I'm showing you here, let me orient you first. I'm going to show you the metabolite levels across this catalog of cell lines with varying levels of fumarate hydratase activity. And to begin with, the cells that lack fumarate hydratase or just got the GFP vector are here, and the ones that got the wild type fumarate hydratase, so rescued the fumarate levels to normal, are here. And red is high, green is low. So you can see, as expected, there's high fumarate to malate ratio and fumarate levels in the cells that lack fumarate hydratase. And also there's high levels of fumarate-driven metabolites that we look at all the time, such as fumarate and arginosuccinate. Fumarate can actually succinate arginine through, it's a catalyzed reaction by arginosuccinate lyase to cause arginosuccinate, which is something known, and it was published previously by Christian Freza and Patrick Pollard's groups. Fumarate can also react non-enzymatically to the cysteine residue on glutathione and cause succinate glutathione, which we see elevated in these cells that lack fumarate hydratase. And it can also react with cysteine, so we see 2-succinylcysteine. So these were kind of a sanity check in our data, showing that these fumarate-driven metabolites are correlating with high fumarate to malate ratios. But what was new and completely unexpected is that 8 out of the 10 most highly correlated metabolites with high fumarate levels in our data set are metabolites involved in nucleotide metabolism. And this was not something known before or something that we would have predicted. And you can see it's very, very strong correlation. There's an elevation in nucleotide metabolism intermediates, in particular purine metabolism intermediates, correlating strongly with elevation in fumarate levels. And so we decided to look at this more closely. In another HLRCC cell line, we looked in an unbiased manner. What are the biggest differences in metabolite levels in these cells when we put back in fumarate hydratase? And again, another huge difference was in purine biosynthesis. When we put back in fumarate hydratase in these cells, we see an accumulation of a certain purine metabolite intermediate, ICAR, whereas we see, on the other hand, accumulation of different purine metabolite intermediates, SICAR and adenylosuccinate, which, by the way, in our previous cell line were the 2 most highly accumulated metabolites in the correlating with fumarate accumulation. And so, again, this made us look more closely at purine metabolism. And to our surprise, something I didn't know before, fumarate is actually a byproduct of purine biosynthesis. Fumarate is actually generated by 2 different reactions in purine biosynthesis catalyzed by the same enzyme, adenylosuccinate lyase, which catalyzes the desuccination of SICAR to ICAR to make fumarate, or the desuccinylation of adenylosuccinate to make fumarate and AMP. And so this reaction in the textbooks is thought to only be unidirectional from SICAR to ICAR or adenylosuccinate to AMP. However, our data showing that SICAR and adenylosuccinate accumulate in cells in which we have accumulated fumarate made us wonder if actually it can be reversed, that fumarate accumulation might drive reversal of this reaction. And for simplicity's sake, I'm going to focus on just the AMP biosynthetic pathway, not AMP also, but everything I show you also is true for AMP biosynthesis. And so our model here is that maybe fumarate accumulation in HLRCC cells or tumors that lack fumarate hydratase might cause reversal of the adenylosuccinate lyase reaction towards SICAR generation, thus blocking de novo IMP biosynthesis. And so in support of this model, we did glutamine tracing in our HLRCC cells. Glutamine can be used to label up different metabolites of the TCA cycle. Glutamine can label fumarate in the M+4 position shown here in yellow. And glutamine can label aspartate in the M+3 position shown here in orange. And what you can see here, we looked at SICAR in particular to see the labeling pattern of SICAR to see whether it can be labeled from fumarate. And sure enough, we do see M+4 labeled SICAR, suggesting that fumarate can contribute to SICAR generation, really proving that this reaction can be reversed in cells when fumarate accumulates. The other important thing that we noticed here is that no label gets through to ICAR. This reaction is pretty much not happening in the cells. It's not going this direction. It's going this direction as you would expect with accumulation of fumarate. So all of this data I just showed you, this labeling data is suggestive that fumarate can drive reversal of the ADSL reaction towards SICAR generation. But really to prove this model, we need to put fumarate hydratase back into the cells and see if this rescues the directionality of the reaction and rescues IMP biosynthesis, ICAR generation and IMP biosynthesis in the cells. And so to assess this, we now use glucose labeling. And what we see here is that when we label cells with glucose, we now see and compare glucose labeling of purine intermediates in cells that, in HLRCC cells, lacking fumarate hydratase or rescued for fumarate hydratase. We see that putting back in fumarate hydratase actually depletes overall levels of these purine intermediates that had accumulated, like SICAR, and now rescues levels of ICAR, and also now the label gets into ICAR. So it really does prove that fumarate accumulation causes reversal of the ADSL reaction and blocks purine biosynthesis, which is really intriguing to us. If the cells have a block in purine biosynthesis, does that mean they're growing? These tumors are growing even though they lack fumarate hydratase, and fumarate's accumulated at really high levels, but they cannot make purines. So how are they growing? They must be getting purines from somewhere, right? And so this made us wonder if these cells rely on purine salvage. Purines are salvaged at the base level. They take in nucleobases and then use the purine salvage pathway. And interestingly, this is the purine salvage pathway. It's catalyzed by two main reactions, adenine phosphoribosyltransferase, APRT, and hypoxanthine guanine phosphoribosyltransferase, which catalyze the reaction between guanine, the nucleobases guanine, hypoxanthine, and PRPP to make nucleotides, or adenine and PRPP. And interestingly, these two enzymes at the gene level are elevated in patient HLRCC primary tumors and metastases relative to the adjacent normal tissues, which is suggestive that even in these tumors, they are relying more on purine salvage pathway. However, to prove this, or to examine this more closely, we decided to knock out these enzymes in HLRCC cells. We actually weren't even able to get full knockouts of both enzymes. There was strong selection against cell lines that grew out having both knockouts. But you can see here, we were able to get a clean knockout of APRT and a knockdown of HGPRT in one cell line. In the other cell line we tried, we actually weren't even able to get any clones that came out that had HGPRT knockout. However, we went ahead and assessed these cells with genetic knockout or genetic loss of function of these purine salvage pathway enzymes. And we found, sure enough, knockout of these enzymes was able to decrease purine salvage contribution to nucleotide pools, as you would guess. And importantly, knockout of these purine salvage enzymes really plummeted proliferation of these cells in vitro. These cells just really didn't grow with knockout of these enzymes. And the same was true in vivo. Tumor xenografts formed from cells that have knockdown of these enzymes grew much more slowly than cells that did not have knockdown of these purine salvage pathway enzymes. And importantly, the tumors that did form were escapers. They had regained expression of these enzymes. So this data suggested to us that indeed HLRCC tumors, or just tumors that lack fumarate hydratase, cannot do purine biosynthesis because fumarate is reversing direction of a key step in purine biosynthesis. And they really rely on purine salvage for growth. So what can we do about this? Is this a targetable metabolic liability for these cells? Luckily it is. There is an old drug called 6-mercaptopurine. It's a hypoxanthine analog. It blocks both of these key enzymes in purine salvage pathway. And sure enough, HLRCC cells are exquisitely sensitive to 6-MP treatment in vitro in an FH-dependent manner. FH introduction into these cells shifts the EC50 by over an order of magnitude. And in vivo, tumors are extremely sensitive to 6-MP treatment at clinically relevant doses, and even in a dose-dependent manner in the xenograft model that we tried. So, to summarize, I've shown you the biochemical characterization of 74 patient-associated fumarate hydratase variants of uncertain significance, which revealed nearly half as enzymatically inactive, strong evidence of pathogenicity. Hopefully, our data will contribute to reclassification of some of these variants as pathogenic and will help these patients who have these variants get regular kidney cancer screening. I've also shown you that fumarate accumulation in FH mutant renal cell cancer cells blocks de novo purine synthesis through forcing reversal of the ADSL reaction. And I've shown you that FH loss of function sensitizes renal cell cancer cells and tumors to purine salvage pathway inhibition. These results are described in a manuscript that we've placed on bioRxiv and that is under a second round of review at Cancer Discovery, so hopefully will be accepted and published soon. This was really I see a beginning. Our next questions are, what about patients? Can patients benefit from 6-MP treatment? These patients really have no treatments that are effective. And so people who run clinical trials for HLRCC patients are motivated to try new approaches. And we've had some conversations with scientists at the NCI who run trials on HLRCC patients and are hoping to start a clinical trial soon. Another question we have is regarding our FH variants characterization. Based on our biochemical characterization, we really only made claims about those variants that had complete loss of function. But there were several variants that had an intermediate level of activity, and it made us wonder, is there a threshold of fumarate accumulation that is sufficient for tumorigenesis, or is it more of a linear relationship between fumarate accumulation and tumorigenesis potential? In vitro, surprisingly, the proliferation rate of the cells in which we put back in variants with varying levels of activity showed more of a linear relationship with proliferation rate and activity, which was surprising to me. However, there's a very well-known variant. It's very, actually, prevalent, this K477 duplication variant, it's actually as high as 1 in 195 in some populations. So it's a really, really frequent variant. This variant is a loss of function variant. It's not complete loss of function. I'm showing you the activity in the forward direction and reverse direction here. This variant is not associated with increased risk of kidney cancer. So we reasoned that perhaps we could use this variant, the level of activity of this variant, as a benchmark to understand the pathogenicity of some of our other variants, thinking that any variants that we found that have activity above this variant are likely benign, not pathogenic in terms of kidney cancer. And so that's one step we've gone in that direction to kind of better understand which variants that have some activity might be pathogenic. The other question we have is, I've only focused so far on fumarate hydratase loss of function. What about succinate dehydrogenase loss of function? This happens in tumors as well. Does succinate accumulation drive a fixed metabolic dependency that we can target? And this is something we're very interested in examining. What I will say is I think it's going to be harder to figure out because succinate is involved in hundreds of different reactions, whereas fumarate is really only involved in a handful. But we're very keen to study this and hopefully come up with a strategy to treat patients with SDH deficient cancers as well. So let me end by thanking the people who've contributed to this work. This was really spearheaded by an excellent postdoc in my lab, Blake Wild, shown here. That's not what he always looks like. It was done in collaboration with our urology collaborator, Brian Shuck, and some folks at Invitae. And it was funded mainly by our Kidney Cancer Association Foundation grant and a small foundation grant from Driven to Cure, which is focused on treatments for HLRCC patients. Thank you.
Thank you very much, Professor Heather, for the great talk. We have some questions here from the audience.
Thank you very much for this very nice presentation. It was possibly too early for you this morning because I already mentioned that 6MP is an important anti-cancer drug dependent on PRPP. And actually, I was wondering how this, or actually don't how, I guess that 6-mercaptopurine will not really inhibit HPRT and not cause deficiency, but compete with hyperxanthine and guanine. And then 6MP, as has been demonstrated in hundreds or thousands of patients, will accumulate as thioguanine nucleotides, which will subsequently have an anti-inflammatory effect, for instance, in IBD patients and in leukemia patients, it will inhibit the growth. So that, I believe it is not really the inhibition of HPRT, but a competition with hyperxanthine, which is driving this. And actually, it would be a bad thing when you would have an HPRT complete inhibition then, because that would lead to an HPRC deficiency, and you possibly know that this is associated with the Last-Nyhan syndrome, leading to gout and severe neurotoxicity in these patients, for which we still don't have any treatment. So maybe you might have to, at least that I wonder whether you might have to change your hypothesis on inhibition somewhat, because you will have an increased salvage, which will increase the metabolism of 6-MP to the 6-thioguanine nucleotides, which might be an explanation for this. And 6-MP has been tested in the past for solid tumors. I don't believe renal cell cancer, but it is. And what about 6-thioguanine, which is an even much better substrate?
Oh, that's a good idea. We haven't tried that one. However, what we have found is that these tumors are sensitive to 6-MP at much lower doses. fumarate hydratase loss sensitizes these tumors to 6-MP. And so, yes, 6-MP is toxic. And yes, 6-MP can get metabolized and then inhibit even purine biosynthesis. But we think that given these tumors' reliance on nucleoside salvage, they are probably taking up more 6-MP because they have higher nucleoside salvage, which we've shown. And we think their increased sensitivity to the drug might mitigate some of these problematic effects. The other thing I'd like to highlight is these patients have there's no treatments for them. They die of this aggressive renal cancer within a few years. And so even though 6-MP does have toxic effects, I think that they would welcome this drug. And maybe there is a lower dose that would work in them than has been used in other cancer types. And to my knowledge, I have checked this in the literature and also with some people at the NCI. 6-MP has not been tried for this cancer type yet.
No, I agree with this latter one. And the solid tumors which have been tested with 6-MP in the past were you treat it with a much higher dose than was usually given to IBD patients, which is a relatively low dose, but you still have a high accumulation. And by the way, when you have accumulation of 6-dihydroguanidinoids, you get an additional inhibition of the pure de novo synthesis as well.
Yes.
By the metabolites or by the methylated metabolites.
Yes. Yes, I'm aware. And we have detected the methylated 6-MP, the TIMP, in our tumors that have been treated with this. So we do know it's getting metabolized in the tumors. But I'm not worried about that.
I'm not either.
Yeah. Okay, good. Well, thank you. Thank you for your suggestion in trying the other salvage pathway inhibitors.
Yeah, Heather, it's very nice talk. Basically, I have just one general question because you are using the HLR-CC cell line, that is a kidney cell lines, and your colleague already shown about the brand that is a kidney fibroid, and the patient history about these fumarate hydratase variants. And
yes.
Pathogenic variants is AM15K is very prominent. You showed that. So whatever your angle of work, I can find out based on the kidney. But if this variant is also can cause other cancer, and I don't know in the vice versa, happen it is other cancer also there is some kind of the variants maybe available for the FH, I don't know. So you can answer.
You know, that's a really good question. If I understood it correctly, why do we only get fumarate hydratase loss in certain cancer types, right? I mean, the most problematic cancer type that shows FH loss is the kidney cancer. The other tumors that the patients get are benign. The cutaneous leiomyomas and the uterine fibroids, they're benign, which is interesting. And I've thought about this. I don't have a good answer. One idea that I have is that maybe those tissues can tolerate fumarate hydratase loss. So the patients they have a germline mutation in one copy. And then over time they get LOH, and that can lead to tumors in certain tissues. But maybe in the tumors, in the other tissues that get LOH, that cell that doesn't make it, right? So that's one thought that I have. We would really like to model this in a GEM model. So we are crossing FH floxed mice with different crees to try to model this. People have done this already in the kidney, and the mice get cysts, but they don't get full on tumors just with FH loss by itself. So we're going to knock out another tumor suppressor gene that is the most common tumor suppressor gene found in patient tumors that have FH loss, and that is NF2, to try to better model the renal cell cancer in mice. But I also am really interested in modeling the other benign tumor types. And we're generating those mice now, and I'm happy to share them with the community, with anybody who's interested in them once they're made.
Hi, Heather.
Hey.
Hi. I'm struck by the fact that I have been thinking about this for a long time, but why does the loss of just this one metabolic enzyme cause this cancer when typically you need all these accumulation of hits? And do you know what happens acutely when you just silence FH in cells? Do they start growing? Or is this a long-term chronic response to the loss of this activity through the mechanism that you're talking about? Because I think then about newer data talking about how tumors are really shutting down the TCA cycle because they're de-differentiating. Essentially, they don't need to do the work of a normal cell, so they don't need so much TCA. Could be that what's happening here is that the cells shut down, aspects of the TCA cycle, and that forces them over time to de-differentiate because they can't do the work of a cell, and so instead they just adopt this proliferative.
I do think that de-differentiation is involved. I think that's a really good point, and I don't think it's been studied very carefully in this case. But given all the methylation changes that happen in these tumors, due to the fumarate accumulation and inhibition of the DNA and histone demethylase enzymes, I wouldn't be surprised if there's some sort of de-differentiation that happens. So regarding your question about acute, we have done that. Most tumors do not want fumarate hydratase to get lost. If you acutely inhibit or knock out FH in most tumors, they won't like it right away.
Cells, just primary cells or whatever.
Oh, primary cells. We haven't done that. We haven't done that experiment. It would be interesting to do. My hunch is that this is an adaptation over time. That the fumarate accumulation, and the reason why I think this is a guess, but the reason why I think that these tumors really don't have a strong signature of many other mutations is that the effects of this oncometabolite are pretty pleiotropic, right? Fumarate is having effects on all sorts of things, and so it gets the job done with one punch.
I think we had some questions from online. I saw. Can we see them? I guess I saw 2 questions. Yeah. No. Okay, so it seems. Oh yeah. Should be there, no? No. Okay, so we'll finish now. So thanks very much again, Professor Heather. Nice to see you. Thank you.
Okay, thank you, Dr. Fatima. So I think we reached end of the session, today's sessions. So I would like to thank all the chairs who have conducted very nicely all the sessions. And I would like to thank the speakers who have talked very excellent. And I hope all of you enjoyed the talk. So I also thank all the oral and poster presenter also. Very nicely they have presented. And now we will move to the dinner. So dinner is at the Holiday Inn. It is besides the Crown Plaza. And before going to there, we will have one photo group session. So here only. So then we will move to the hotel. Hi Flavio, thank you for chairing the session, and it's good to see you after such a long time.
Hopefully, at some point we'll meet in place. Maybe
Yeah.
in Italy.
Yeah, sure. Thank you.
Thank you all of you to invite me. Thank you.
Hi Flavio, how are you?
Hi, very good.
So how's your kids?
growing. And become old, they are growing. So thank you very much, and hopefully to see you and all the family together.
Yeah, together. I want to see you too here in India again.
Yeah, sure.
Thank you so much.
Thank you.
Thank you. All of you, please gather here so that we will have a group photos. So I request all the speakers and participants, please come over so we can have one photo. I know you are all tired, but let's have this photo and then we will proceed for line. Yeah. So please come over the dais. So let's have a photo. Yes, everyone. So all the participants, please come forward. Let's have a photo.
Linoleic acid or olic acid? Sorry? Linoleic acid and olic acid, fatty acid. 18 carbon chain.
No, I haven't tried because it's a really, I tell you the story. It's a really story. This is a nice story. We just have 8 minutes, so but we have if you want we have yes. Yeah, absolutely.
I think it's
You have to go to the front.
usually on the front.